Good morning, everyone. Welcome to the 12th Annual Student Research Conference. This is a joint conference between Mott Community College, Kettering University, and the University of Michigan-Flint. It's a great venue for students to present on their research. My name is Ken Sylvester. I serve as the director of the U of M Flint Office of Research. In normal times, we'd be doing this meeting in person, but this year's event is virtual. Follows a very successful effort last year to move the event online. And I want to thank Andre Lewis and Mary Dybus for all of their hard work in organizing this year's SRC, for putting out the call for proposals, receiving the submissions, and then organizing them into sessions today. Andre will join us in a few minutes to explain the organization of the program today and how the breakouts will work. And thanks to all of you students who submitted presentations and papers, and to the faculty mentors who support them, and the faculty and staff who have agreed to help moderate the presentations today. So now I'd like to turn things over to Heather Dawson, who is the Executive Director of the Office of Research and Economic Development at the University of Michigan-Flint. Heather is a professor of biology who leads this new unit on our campus. And Heather will offer some introductory remarks about the impact of student research. Heather?
Hey, thank you, Ken. I appreciate it. So I'll just say a few things and then of course yield the floor to the main presenters today, which are the students. So I just wanted to mention that this Flint Student Research Conference has been going on since 2011. We have about an average of 50 to 60 students from U of M Flint, Mott Community College, and Kettering University that present their research and creative activities each year. And then of course we have annual conference proceedings that students can actually publish their research into. So just to say a few words about the different universities and sort of the support they provide for undergraduate research. So since 2005, U of M Flint has provided over $850,000 in student stipends through UROP. We also just recently started a summer undergraduate research opportunity experience. This provides a more intensive experience in the summer and support of up to $5,000 to support sort of that full-time summer research opportunity. And then U of M Flint also provides graduate student research support. So there's some graduate students here, and the GSRA assistantship program began in 2006, and there's been over 1,000 appointments since then, and about $3.5 million distributed to graduate students. And Mott Community College, they pride themselves on offering experiential learning through service learning projects in conjunction with community partners, so injected right into the community. So they have students volunteering, completing projects like at Autism Support and Resource Center, the Whaley's Children's Center, McFarland Home Residential Care Facility, the Genesis Program of All-Inclusive Care for the Elderly, just to name a few, and provided lots of support for undergraduate research, which students can then move on to potentially a university after that. Kettering University supports a lot of different research opportunities for undergraduates. You know, they may be sponsored a student to work in a project in a research lab during the school term or the work term. And then the Center for Culminating Undergraduate Experiences provides students with opportunities to conduct research work for completion of a thesis. And Kettering University was awarded a $270,000 grant recently from the National Science Foundation to provide research experience for undergraduates, right? And so we know that there's a lot of learning gains for students that get involved in research. You know, not only do you get to work with a professor who can also provide you with recommendations, help you mentor yourself on a career path, but it also provides learning experience. We did a recent survey, and students that have been involved in a research experience, they have more confidence in their own understanding, more comfort in their subject matter, comfort in collaborating with individuals, and understanding theory and concepts and connections to other concepts. So And that's from the voice of the students that have told us that. Okay. So, and the students here are not doing this for a grade for the most part, right? So you find that students tend to challenge themselves even more in those situations, really. And I know that I have a couple of students presenting here today, and I'm very proud to see them present their work. I have a graduate student and 2 undergraduate students presenting. And they've been working on this for months. They worked every week for months on this presentation. And if you've ever been to a professional conference, you know that the student presentations are often better because they spend a lot more time in preparation and preparing themselves for this opportunity. And last thing I'll say is that, if you are a senior or a graduate student in contact with students early in their career as undergraduates, I would suggest to tell them about your experience, because right now we know that research experiences are normally skewed towards seniors, like people like, leaving the university, getting ready to graduate soon. And we need to recruit your op students or students for research experiences early in their university experience, so they can get all of these sort of experiences ahead of time. So I won't take up any more time. I just wanted to say a few words, and I'm really excited to see the presentations today. And I'll leave it over to Andre, I believe, to do the next. Thank you, Andre.
First off, thank you, Dr. Sylvester, for the welcome, and thank you, Dr. Dawson, for those warm remarks to open. First off, good morning, everybody. My name is Andre Lewis. I'm from the Office of Research and Economic Development here at U of M Flint, and I'm one of the co-coordinators for this event. So in a few short moments, before we start, I just wanted to go over a few quick housekeeping rules. So in a few short moments, the breakout sessions will start. So all the moderators and presenters have been pre-assigned to your respective breakout rooms. So once I open those, you'll automatically get sent over to your breakout. Now, if you don't move automatically, don't worry. As a fallback, you can actually you can follow these instructions, which I'll share in a moment, which also apply to you as our non-presenting guest. So I'll show you how to navigate to different rooms. So I'm going to share my screen really quickly. Let's see, here we are. Okay, so at some point, once I open the breakout rooms, you'll see a little breakout room button at the bottom of your screen. If you click on that button, then it opens up a separate panel, which will list all the different breakout sessions that are available. If you click on the little join button next to that session, and then click the join button again to confirm it, then you'll get redirected to that breakout room. And then at any point during your session, if you want to navigate to another room, you'll have the opportunity to do that as well. So again, the breakout room button will appear in that room. When you click on it, you can click on the choose a breakout room option, and then go back to that same panel, and then join whichever breakout you want to attend. So each of the rooms contain between 3 to 4 sessions, and so again, you'll be free to navigate throughout different sessions as you'd like. So I'm going to stop sharing that, and then go over a few more things. So each presenter will have 15 minutes to present. About 2 to 3 minutes of that should be dedicated to question and answer. And you can leave your questions in the chat for your breakout, or your moderator can also give you the option to have your mics turned on during that session. And then after the breakout rooms are concluded, then I'll close them out and then send you back to the main room. We will also send broadcast messages at around the 12 and 15 minute marks, just to give you give you a sense of time for your session. So Mary Davis and I we will remain in the main room, and if you have any questions, you can always come back to that main room, and we'll be happy to navigate those. So with that being said, if before I dismiss, are there any questions? Feel free to open up for any questions at all. Okay, well, we are actually a little early, so for the first students, for the first student presenters, if you just want to take a moment to decompress before you start, you're more than welcome to. We actually didn't have our opening presentation start until around 20 after, so we've got 10 minutes still. But if you want to jump right into your presentations as well, you are more than free. So at this time, I am going to open the breakout rooms, and you'll be dismissed in 5, 4, 3, 2, 1. And then anybody who is left in the room with us, who has not selected a breakout room, we can happily assign one for you as well.
Apoptosis, and as you increase the concentration of bicolin to 200 micromolars, you increase the percentage of cells induced for late apoptosis and for dead cells. Next slide. So after looking at apoptosis, we wanted to see specific pathways that apoptosis caused. So we started a reactive oxygen species assay, and we discovered that once you increase the concentration of bicolin from our control to 50 to 100 to 200, you increase the number of cells with reactive oxygen species. So as you increase the concentration, you go from cells with reactive oxygen species from 8% to 21% to 37 to roughly 47. And reactive oxygen species are important for the further induction of apoptosis. And as you see more reactive oxygen species, you see a further induction of apoptosis. And in the graphs on the right, you can just see a direct comparison of the control with the 200 micromolars of bicolin. So that second peak with the 200 micromolars of bicolin indicates that there are more cells with reactive oxygen species. And then the two graphs are just showing a statistical analysis of the increase of bicolin inducing reactive oxygen species in cells. Next slide. And to take that even further, once you induce reactive oxygen species, you also can start a pathway of DNA damage. And to test for that, we tested the phosphorylation of H2AX and ATM, because the phosphorylation of those two proteins indicates that DNA damage is occurring. And as you increase bicolin, you increase the phosphorylation of both those proteins, and you also start inducing DNA double-stranded breaks, which is another important factor for DNA damage and apoptosis. And as you can see on the graphs on the right, as you increase the concentration of bicolin, you increase the number of cells that get phosphorylated, and you increase the number of double-stranded DNA breaks. So you are inducing a DNA damage pathway and introducing DNA double-stranded breaks to the cell. Next slide. So this figure here is a general overview of some of the processes I have just been discussing. So DNA damage causes the phosphorylation of ATM and H2AX, which leads to phosphorylation of P53. And P53 is an important factor in apoptosis, and it actually induces apoptosis even further to induce more of the late apoptotic pathways as well, to also activate caspases and introduce apoptosis to kill the cells even further. So after this pathway occurs, Ben is going to start talking about some of the genes involved in apoptosis and some of the caspases as well.
Yep, so just as Tristan said, these are the genes that are going to be affected by the boculin exposure in our breast cancer cells. So our red dots, these are going to be our genes that are upregulated, and our green dots are going to be downregulated. So for example, HRK in the top left-hand corner is going to be upregulated at a much higher rate than TNF, for example. And in the bottom, TRAD is going to be downregulated at a much higher rate than CRAD, for example. So now we're going to look at the 5 most upregulated genes in the apoptosis pathway. As you can see, CASP5 and HRK are by far the most upregulated, with HRK being 20 plus and CASP5 being 15-fold plus. CASP5 encodes for the caspase family. Activation of caspase plays a central role in the execution phase of cell apoptosis. And HRK encodes for a member of the BCL2 family, which we will talk about in depth a little bit more in the next slide, which we're going to be talking about the downregulated genes in apoptosis pathway, which there are much more genes found to be involved in the downregulation and at a much higher fold. So first, we're going to look at TRAD and CRAD, TRAD being at 100-fold downregulation, and CRAD being the 5th most downregulated, respectively. TRAD and CRAD are associated with knockdown, significantly blocked cell invasion. Next, we're going to look at CD40 and TP73, the 2nd and 3rd most downregulated. These are associated with proliferation and invasion. Next, we'll look at TNSF25. This gene is associated with drugs designed to knock down and is currently in a phase 1 clinical trial in patients with solid tumors. Knockdown of DR3 inhibited cell growth and induced apoptosis of hepatocellular carcinoma. And then lastly, the BCL2 family, which I mentioned, these are anti-apoptotic genes, and if upregulated, the cell will survive and persist, but if downregulated, like shown here, the cell will undergo apoptosis and cell death. So as you can see, with the exposure of boculin is going to regulate to inhibit breast cancer cell proliferation, and it's going to induce apoptosis. And we'll kick it back to Loveland for the conclusion.
Thank you, Ben. So in conclusion, our research demonstrated that boculin can significantly inhibit cell growth and proliferation of the human ER-alpha positive breast cell carcinoma cell line T47D by inducing cell cycle arrest, suppressing colony formation and cell migration. We have shown that cell death is induced by regulating genes and interacting signaling pathways involved in apoptosis. And these studies clearly suggest that boculin has potent anti-cancer properties and is a great potential for cancer therapy. Next slide. Those are our references, and we want to thank our principal investigator and Professor Dr. Bessa Jabia. We would like to thank the Office of Research, Andre Lewis, and everyone else who is listed here who were pertinent to the success of this project. We're happy to take questions.
Thank you very much. Questions for the presenters? Does anyone have a question? They just unmute yourself or put a question in the chat, please.
I have a question, please. So my question is related to one of the gene it shows like down-regulated by 100-fold change. So that's like a huge amount of fold change to see a reduction of this by this amount. So do you have any reason for that? Like, yeah, the first one here, because I believe this is in a log scale, right?
Right, so TRAD is associated with knockdown, and so it was found to be at a full 100-fold increase, as you said, and is very significant.
So Ben, can you please describe what TRAD does? You went through it, you described it.
Yeah, so TRAD, it's involved in a knockdown, significantly blocked cell invasion. So
So when it is in normal condition, it's involved in cell invasion, correct? So when it is knocked down, what is it involved in? What does it do? It suppresses cell invasion, right? That's the suggestion.
Right.
So we could follow that with Yushin and then do an invasion assay to kind of answer that question as well. The importance of this is that we're identifying potential key genes that are responsible for this mechanism of basically shutting down cancer and involved in the apoptosis. The other next two genes, the CD40, they are invasion, proliferation as well, right? So this is kind of confirming the TRAD, which is a 100-fold decrease. Mm-hmm. So we're using Becklin as a way to elucidate the mechanism by which we can shut down cancer progression in ER positive luminal type A cells, right? So not necessarily use Becklin as a way to kill cancer, but potentially design new therapeutic drugs to target this mechanism.
Amazing, thank you.
Thank you.
Any other questions for the first presenters?
Maybe a follow-up question here. Like, do you guys have ever thought about using computational model for gene regulatory network, for example, to explore more this pathway for this couple of genes is going to be upregulated, or they stay as it is, or downregulated? Have you thought to use any of those computational models?
Okay, I'll answer that because I don't think they would know that. So that's where you are going to come to this project.
Well, I'm asking this.
I would love to further look into this model and find out potential other genes that could be regulated. These are targeted pathways. We didn't look, this is not RNA-seq. As you know, I want to get into that, but this is targeted pathway. So this would be 84 genes in the apoptosis pathway, which could help us identify what are the target genes. Yeah. But then if we want to look at more, I think they'll be very beneficial for this project.
Perfectly. Yeah, that would be great. And I think we should talk.
Yes, yeah. And a potential, we can also bring Yushin to the picture because an invasion assay, especially with the TRAD being knocked down 100-fold, I was in shock to see a gene 100-fold, and obviously look at it at the protein level as well, right? Yeah. That we could potentially have a novel pathway for, downregulating cancer and this type of cancer, ductal carcinoma.
Yeah, sounds great. Thank you.
Yeah, thank you.
Okay, thank you. Thanks, Benjamin. So it looks like Perez is here. Perez, do you want to come out of hiding there? Would you like to go next? Or there is another presenter.
Yeah, I could go.
And yeah, okay. Would you like to share your slides, or would you like me to share your slides for the group?
You could share, that would be good, because I think I tried sharing it yesterday in a meeting and it was kind of messing up on my laptop.
Okay, all right. I'll load it up here. Let me know if you can see it, okay? How's that?
Yeah, that looks good.
Okay. Just let me know when you want to advance them.
Yeah, just one moment, sorry. All right. Hello, everyone. My name is Sylvester. I'm a senior here at the University of Michigan-Flint. So I did this project when I was at the University of Michigan Medical School in the summer of 2019 with the Michigan Health Sciences Undergraduate Research Academy. I was working with my mentor, Dr. Seville and Dr. Blackwood. And the project that I implemented during my 8-week program was exploring treatment practices for sickle cell anemia patients visiting the ED for pain crisis. So just a little bit of background information. Sickle cell disease is an inherited genetic blood disorder caused by mutation in the hemoglobin of red blood cells. And it's most common amongst African Americans. And the major or the main symptom that patients with sickle cell experience is our pain crisis. And that's usually the biggest reason why they present to the ER. And furthermore, African Americans are often stigmatized when they're seeking opioid analgesics, which is the recommended treatment by the National Heart, Lung, and Blood Institute. So our objective of this study was to explore the pain treatment practices for those pain episodes in the ED for pediatric patients specifically, because like major barriers to effective management of pain are clinicians' limited knowledge of sickle cell disease, inadequate assessment of pain, and also biases against opioid use. And like those biases are based on ignorance about opioid tolerance and physical dependence, and also like confusion with addiction and assuming patients are opioid seekers or drug abusers. So to the next slide for symptoms of sickle cell disease. So again, the main symptom that patients experience are painful episodes. And on the right, that's kind of a picture of sickle cell, like how it presents in the blood vessel. The blood sickles, and then it blocks the blood flow, and it causes obstruction in the blood flow. And this can present as hypoxia, low oxygen in your tissues, also acute chest syndrome, which is when there's a lot of sickling in the small blood vessels in the lung. Also, there's some patients have enlarged spleens, which is also known as splenic sequestration crisis, and this happens when a lot of red blood cells become trapped in the spleen. And another symptom is dactylitis, which is painful swelling of the hands and the feet. So these are just some symptoms, and then on to the message on the next slide. So this was a retrospective cohort study examining the electronic medical records of patients who presented to Mott Children's Hospital, which is the main hospital at University of Michigan, Ann Arbor, from January 2016 to July 2019. And our patient population consisted of pediatric patients from ages 0 to 21 who presented to the ED for their pain crisis. And we analyzed treatment variables against age, gender, and insurance type. And again, the goal was to explore the current treatment practices for the pain episodes in the emergency department by looking at opioid administration as an oral opioid or an IV opioid, because those are the recommended treatment for patients. So in the next slide, we have our patient population, and we decided to divide it into the different school grades, so preschool, elementary, middle school, high school, and post-high school, just because patients are at different developmental stages in those school grades, and they may experience or present or perceive pain as differently. So for example, infants in the ER, they're not given a scale of 0 to 10. It's just kind of an observation of their behavioral experience when they're in pain. And that's how they assess how in pain they are, because the infant is not going to be able to adequately, say from a scale of 1 to 10 how severe their pain is or not. And then we had one patient who did not have insurance, and we had 40% of our patients who had private insurance and 57% who had public, and we had pretty even split between males and females. So onto our results on the next slide. So we see in the figure 1, we have the oral and IV treatment by the different age groups. And in figure 2 is the oral and IV treatment by gender and insurance type. And basically, so all patients that we looked at received treatment for their pain crisis. Males and females received an oral opioid. Males received 66%, 66% of males received an oral opioid, and 56% of females received an oral opioid, respectively. And regarding an IV opioid, it was 91% for males and 84% for females. So the gender differences were not significant. And then looking at the age groups 1 and 4 and age groups 14 and 17, they appeared less likely to receive an IV opioid than other age groups. And also there were no statistical significances between oral opioid among the different age groups. Our p-value was 0.682. So also in examining opioid administration and insurance type, oral opioids were administered for patients with public and private insurance, 64% versus 55% respectively. And IV opioids for patients with public and private insurance, that was 95% versus 82% respectively. And those differences were also not significant. And for our one patient who did not have insurance, they were treated with both an oral and an IV opioid. So in conclusion, according to our data, when patients came to the ER to seek treatment for their pain crisis, the treatment was consistent with guidelines from the National Heart, Lung, and Blood Institute, and all patients were treated for either an oral or an IV opioid or both. However, the small sample size and lack of specificity in other measures of pain and the treatments in the study limited our ability to find any differences in pain treatment because our sample size was only 49 patients. So in the future, what I would like to do is, first of all, expand the patient population, of course, and I would also like to look at adult patients in the same hospital and examine if there are any disparities between pediatric patients and adult patients, because adults are more likely to be perceived as opioid seekers or drug abusers. And lastly, me and my mentor are thinking about comparing patients who present to the ER with cystic fibrosis versus patients with sickle cell disease, because patients with cystic fibrosis are majority white and patients with sickle cell are majority African American, and just to see if there are any differences in the pain management in those two patient populations. And that is it for my presentation. I would like to acknowledge the University of Michigan Medicine Office for Health Equity and Inclusion, and a special thank you to Dr. Brandon Lucas, the program manager for the ENSURE program, for allowing me this opportunity to present my research and conduct research at the University of Michigan. Thank you everyone for listening, and feel free to ask any questions.
Thank you. Any questions for Faraz?
Hi, first, nice talk. I have a question related to the mutation for this type of sickle cells. So what type of mutation? Is that like a point mutation, and in which residue, for example?
The mutation for sickle cell, it is the valine is mutated. There's a one mutation where the glutamine changes to valine. It's an autosomal recessive disease. So both genes must be carried on from the parents to the offspring for it to be passed on to the next infant. Is that your question?
Right. Is that like a specific residue in that protein, or it happened everywhere?
What do you mean a specific residue?
Like if you look at the protein is made of a couple of amino acids, for example.
Yeah. Mm-hmm.
So is that like in one of the particular residue at one point mutation, or it happened?
Yeah, it's just one base, one nucleotide that changes the valine to glutamine. It's not like different.
Mm-hmm.
But yeah, that's the question.
Okay, can I ask an unrelated question about the program that you were involved in?
Of course.
So how did you get involved in the program at Michigan Medicine and involved in that specific project?
Yeah, it was a great program. I actually have been trying to like promote it and get more Flint students to join it. So my cousin, he went to University of Michigan, Ann Arbor, and he did the program. It's under the OHE office of the medical school. So it's basically like a pipeline program for minorities, but also like, non-minorities are allowed to participate in it as well. And it consists of about like 20 students. It's pretty competitive. I think they have like about 500 people apply to take 20 students. And then you get matched with a mentor. They pay for your housing, your meals, your research is funded. You get a $4,000 stipend, and you just get access to, shadowing opportunities, volunteer healthcare and disparities workshops. And we also actually had a professor from Flint give a speech there in one of the workshops, Dr. Seeley. So that was nice to see. She was one of my professors. But yeah, it's a really nice program. Actually, my mentor, who was the person who started the program, Dr. Blackwood, I have been in like discussion with him to kind of reserve like 3 spots for U and Flint students to get into the program. And I'm just trying to engage with the faculty at U and Flint to hopefully fund that. And I think they're supportive of it. But it's definitely like a great program. Like I learned a lot about the research world in space. Like when I went there, and a lot of things I didn't know outside of research and in research and in the process to apply to medical school, like were taught to me during that program. So it's definitely a great program. I'll actually drop the link for it in here. There's the research program, yeah, and there's also a program called CDA. The URA is the Undergraduate Research Academy. CDA is the Career Development Academy, and it's basically an MCAT prep program where they pay for your MCAT course with the Princeton Review. They also pay for your housing, give you a stipend, and allow you to, just study MCAT in a structured environment with other students. So it's definitely a program, and I encourage everyone who is, interested in healthcare or MCAT studying to definitely join.
Okay. Any further questions for Perez before we move on to the next presentation? We have lots of time because there are only 3 presentations in this session, and every other session has 4 presenters.
I have a question I could ask.
Please, Tristan.
So do you think the hospital location has an effect on how the children are treated for the sickle cell disease, such as like if a hospital was less funded or in like a higher area with more minorities or less minorities, to see how these kids would be treated?
I do think that like it does matter, like the hospital, like the demographics of patients who present to the hospital, or I guess the socioeconomic status. But I mean, at the end of the day, most patients are all African American, so it's not going to be. I'm obviously not sure of this because I haven't done research in different hospitals, but I'm assuming because most patients are African American, we should maybe see a similar results. I think the interesting area to look at would be the age groups, pediatric patients versus adult patients, because, a kid who's 5 years old, he's not going to be coming, trying to get some opioids or get high or something versus someone who's, an adult. So that's where I want to assess and look at, I'm sorry about that, and look at adult patients versus pediatric patients. Does that answer your question?
Yeah, thank you.
Okay. Yeah, any other questions before we move on to the next presentation?
I'll also drop the link if you're interested in sending it to any students or so. The application deadline did end in February, but for next summer, it's definitely something that would be good for people to get involved in.
Okay, thanks very much.
Yep.
So the next presentation is actually a recording. I believe this is Yasser. I believe this is your student. And she had a conflict. She's also today presenting at the medical school. Is that right?
Yeah, that's correct.
Okay, so she recorded this, and I'll get it set up here. Just let me share my screen and start this video presentation. Let me know if you can hear it.
I'm not able to hear it.
Can you hear it, Yasser?
No, I can't.
I can't hear it either.
Okay. So I can definitely hear it on my machine. Do you have a copy of it, Yasser?
Yeah, I'm gonna open the folder here, see if I
Okay.
open off. She put the PowerPoint presentation, but probably put the video as well. So let's see here.
Hi, everyone. My name is Quinn.
Okay, so I think I can share my screen.
Yes, you can. Please go ahead.
Yeah.
Thanks for the assist.
No, you're welcome. Okay, so here.
Faculty advisor, yes, sir. Thanks for taking the time to listen to this presentation. So today I'll be presenting on conventional and pioneering technologies in cardiovascular imaging. I'll start by going over the basics of cardiovascular imaging and what it is, why it is used. But the bulk of this presentation will be discussing common imaging technologies, explaining the functionality and the purposes of each. At the end, I'll fast forward to the future and what we can expect from these technologies. But the big picture or reason why we're developing a foundation of knowledge is to translate current technologies with innovative technologies, basically into a healthcare application to help diagnose and treat cardiovascular diseases or complications. So cardiac means heart. The process is typically the patient comes in for a scan, the radiologist technician performs the diagnostic imaging scan, and then the radiologist interprets that scan. So a radiologist is someone who is a medical doctor. They could be an MD or a DO who completed a 4-year residency in radiology. And then cardiovascular radiology is a subspecialty of that diagnostic radiology. These tests are ordered and used to screen for heart diseases, determine what is causing your heart symptoms, monitor your heart, see if treatment is working, things like that. So some imaging techniques I'll go over today are echo, X-ray, CT, nuclear scans, MRI, and catheterization. So let's get started here. Okay, so first, let me go over the anatomy because you will be viewing anatomy from various scanning techniques. So it's important that we just quickly overview it. So here we have blood coming in through the superior vena cava into the right atrium, right ventricle, out the bilateral pulmonary arteries. And then that's going to the lungs. It's getting oxygenated. It's coming back through the bilateral pulmonary veins, down to the left atrium, left ventricle, and then out to the aortic arch. So let's rotate it. This was the anterior view. Now you're seeing the posterior view, which is visualized over here. The same thing's happening, just reverse order. So it's on the other side now. It's coming in through here, right atrium, right ventricle, out to the lungs, come back through the left atrium, left ventricle, and then out to the systemic circulation through the bloodstream. So here we have echocardiogram. So it was developed as a diagnostic test to examine cardiac function, structure, and hemodynamics. So it uses ultrasound, also known as sonography, to produce high frequency sound waves which bounce from the heart to the transducer to create a visual of the heart on the computer, which is what you see here. So various types of echoes are available to examine the heart, such as transthoracic. We also have 3D, Doppler, and stress echo, each having unique benefits and uses. So overall, the advantages of echo are a more detailed picture compared to X-ray and no exposure to radiation. Blood clots, pericarditis, valvular heart disease are just a few examples of what an echo typically screens for. Next, the heart X-ray, also known as chest X-ray, creates a visual of the heart, lungs, and surrounding bones via radiation beams. So chest radiographs are primarily used to detect the anatomy of the aorta and surrounding pulmonary veins and arteries. As far as producing a visual, the body varies in depth and in thickness. So basically amounts of radiation are absorbed differently. For example, soft tissues may appear gray, where bones appear white. And then you can see the outline of the lungs there because air is appearing black. So chest x-rays are typically used when patients come in due to dyspnea, also known as shortness of breath, persistent cough, or maybe angina, which is heart pain. So it is most commonly used because the scan is very prompt, straightforward, and relatively affordable. Next, we have computed tomography, also known as CT. The first scanner was invented in the late 1960s and clinically applied in the early 1970s. So it merges a sequence of cross-sectional slices of the heart and surrounding blood vessels via X-ray images. A significant difference to chest X-ray is cardiac CT can create these beautiful 3D illustrations, and cardiac CT basically aims to provide these detailed images of blood vessels, soft tissues, and bones in less than 20 minutes, which is great. As for types of CT scans, a coronary artery calcium scan, also known as CAC scan, detects the presence and proportions of plaque in the arteries. Another type of scan called the CTA, the coronary CT angiogram, quantifies the amount of calcified and non-calcified plaque. So this could be useful for stenosis. Overall, cardiac CTs are generally ordered for pain, atherosclerosis, and coronary artery disease. Next, we have nuclear scans. So in this top image is a PET scan. The bottom is a PET scan. So a SPECT scan works by injecting a radio-labeled tracer that emits gamma rays into an individual's bloodstream. So this collects information from the gamma rays and displays visual of the heart on the CT cross sections to rule out things like perfusion, scar tissue, see if bypass is working or other cardiac surgeries are working as they should be. A particular type of test is called the stress test. And this basically looks at the functional status of the heart at rest and then under activity, also known as stress. So after SPECT was developed, PET followed. PET is a type of nuclear imaging technique that also injects a small radioactive tracer to circulate the heart. But the difference is the type of radio tracer used. So SPECT uses or measures gamma rays, whereas PET can produce positrons. But yeah, they're both very useful for examining tissue damage of the heart, viability, and abnormal substance buildup, just like calcium or plaque. Next, we have MRI. So an MRI is a large machine containing powerful magnets, which are aligned in a tube formation. So it basically looks like this, and the patient's going in the tube for 20, 30 minutes, but sometimes up to an hour. But these magnets work by forcing protons in your body to align, and then the radio frequency pulses through you, and it basically stimulates the protons in your body by pulling them out of equilibrium. So the differences in this proton activity allow the MRI sensors to detect the energy, which translates to the images on the screen. Distinct types of scans include cardiac viability, right and left ventricle function, stress perfusion, structural assessment, and angiogram. A significant advantage compared to other scans is the lack of radiation or strenuous iodine contrast. So for example, that's what CT scans use is the iodine, but MRIs use something called gadolinium, which is much less potent, and it's referred to as the gold standard, and that's one of the reasons too. But as you can see, it creates beautiful detailed images shown here on the screen. So let's move on. This one will be very short. Let's see if I can play this video. This is catheterization, and it's a type of invasive cardiac imaging. I'm sorry. Here we go. So you can see it working there. It basically works by guiding a thin, flexible tube called a catheter into a blood vessel, usually the femoral vein, and it goes all the way up to the heart, and it can help diagnose and treat heart conditions. Looking to the future, some of you may be familiar with artificial intelligence, machine learning, virtual reality, and augmented reality. So today it's used in the gaming industry and entertainment industries, but it has the potential to transform the healthcare industry. So we're super interested in this. We believe this sort of diagnostic imaging can positively influence overall population health, morbidity, and quality of life through early and effective means. So here are my references, and thank you for your attention. I believe Yasser is on the call if anyone has questions or thoughts. He's more than happy to talk to you more. So thank you.
Thank you everyone for your attention. I'm happy to answer any question.
Any questions from the students here? I noticed our numbers have dwindled a little bit, but let me ask the question about an example. Can you give an example of how machine learning might improve the kind of diagnostic that you would get from this kind of scanning?
Yeah, so that's a great question. So normally for image processing, most of the scans that we have to do segmentation using manual segmentation. So basically, we bring an expert to start to identify where is the tissue compared with the lumen or the blood regions and etc. So there might be a potential of mistakes from like human mistakes based on what is the level of the training for this expert, etc. So in my previous labs, we used to do it by assigning these tasks to different student pool, and then we compare between the results in order to just make sure that this is accurate enough for building our simulation. It looks like the machine learning or artificial intelligence can help and do automatic segmentations with more accurate. So the mistakes will be less because we build the machine learning based on algorithms that optimize the process. So it's going to save a lot of time and also more accuracy to identify especially the scar region in the heart or in the myocardium. This is also was a debate because if a person that does not have enough training and identify the scar region in the myocardium to be a larger region, then when we do appellation, then the clinician is going to burn a tissue that he's not supposed to burn or burn more tissue. So I believe that a coupling or making use of artificial intelligence and machine learning for image processing to identify with precisely what is the region for scar, that might help, avoid some, procedures that we're not supposed to do.
Okay. Thank you.
A second maybe reason is trying to use a VR, and that Kieran mentioned at the end, is we don't want to use it for just visualization. It's very nice to see more details about the anatomy, but we would like to use it to quantify some data from these images, not just for visualizing as a three-dimensional looks nice and maybe for educational purposes, but we would like to couple it with the image processing techniques so you can have immersive experience in a three-dimensional, and then you start to better quantify where is the biomarkers in the tissue at the tissue scale. So that's another maybe potential contribution of using machine learning or VR or augmented reality with some of these clinical scans.
Okay, thank you. Other questions? Everybody's being shy. Tristan, Levelin, Benjamin, any questions?
I guess can you like generate multiple images to get like a better image of the heart, like overlay images together or anything? I don't really know much about imaging, so I'm just curious.
Yeah, so normally for MRI scans, so they take multiple scans along the short axis. So just to look at it as a cross-sectional along the short axis of the heart, and then it's a stack of images, and then we also need a couple of more along the longitudinal axis or the long axis of the heart. And the whole goal is to try to build a 3D model using these images. So we go stack by stack or image by image, and we do image processing for each one, and that's going to give you kind of a ring, right, for each ventricle. And then you go up to, you end up by the atrium and so on. And then that's not going to be enough to build a 3D, just two-dimensional snapshots along the short axis. And then you also need a couple of them along the long axis. So you do that as well. And then you started to align them with the specific two reference axes, or what we call the inertial axes within your own body. So we'd like to orient the heart exactly same as what you have in your own, or for the patient. So the process requires a lot of image processing and also some of the optimization for the alignment with your body axes. And then that's going to be your computational domain, what we call it, for our simulation as a finite element. But this is kind of pre-processing before we start the simulation. It looks kind of maybe trivial, but it is not, because small mistakes for considering a tissue like the part of the blood pool or vice versa can change what we call it the pressure volume relationship, which is something that we iterate until we reach steady state for our simulation. So basically, if you account for more volume of the left ventricle and it's not supposed to be, and that's due to mistake when you do the segmentation, then that might lead to computational error.
Okay, thank you.
Yeah, you're welcome.
Okay. If there are no further questions, I'd like to thank all the presenters. This was a great session. And I guess we're free to have more time in our day. Go back to the main session. I think there were some closing remarks that were intended for shortly after 11 o'clock. And you're welcome to stick around for those. And of course, as in years past, if you wanted to make a submission based on your presentation, those are welcomed as well. Okay, so thanks very much, everyone.
Thank you, Ken. Thank you so much.
Thank you.
Thank you.
Thank you. Bye-bye.
That still has a fourth presentation to complete. So it could be a few more minutes before we close all of the breakout rooms. If you want to stand by, that's great. If not, I understand as well.
For an app, we found that healthcare providers, what they wanted in an app fell into a few different categories, the main one being patient education. So things like providing resources and recommendations for patients, like we talked about earlier, addressing misconceptions around speech language therapists can encourage people with Parkinson's to seek help for speech therapy. Also being able to show how to differentiate between Parkinson's symptoms and things like aging, and establishing routines so that patients can help themselves in between sessions. They also recognize a need for reimbursement. So a lot of times, doctors, they don't have that long of appointment times with their patients. So being able to be reimbursed for the time spent reviewing the app would be very helpful. And then also having a way to represent the data. So using graphs and either scatter plots or bar graphs to be able to easily see the data, and then condensing the data into snapshots to also accommodate for these small appointment times. So, and then also recommendations for monitoring symptoms, things such as passive tracking. Basically, the less that people with Parkinson's have to do, the better. So things like tracking motor symptoms with sensors in the phone, also being able to track how patients move in diverse environments, monitoring exercise, being able to self-manage symptoms between sessions, and having patient diaries that can help to reveal patterns that weren't evident before. Slide. So in the future, we want to continue to collect and analyze data on what features would be most useful. We're hoping to collect from healthcare professionals in underserved areas and across different disciplines. And ultimately, we want to actually develop an app and deploy that into the field to continue collecting usability research on the application. Next slide. So this is our research team that was working on this project. We'd like to acknowledge U of M M-Cubed funding and the U of M Flint UROP and GSRA programs. So this concludes our presentation. Thank you so much for listening. We'll now open the floor up for any questions you guys may have.
Great work, guys. Does anybody have any questions? Any questions at all? And also, this would be a good time in the last couple minutes that we have if anybody has any more broader questions. So unfortunately, Dr. Tang had to leave, and she thanked everybody for their time, and also said everybody was doing an excellent job. And so I think to the Dr. York team, that's a conversation that maybe we should have some time, or at least reaching out to Dr. York and seeing about the possibility of maybe getting some of the power participants perspectives on if they'd want to participate in a study, and see if there might be a nice interface between the power program and the app. But any other kind of bigger picture questions? It was nice in this session that there was kind of a unifying theme around Parkinson's disease or other neurological disorders. So awesome. Well, it doesn't sound like we have any questions, and it looks like we're at time. So I achieved my goal of actually getting things in time, which, if you know me at all, is a major goal, because that never happens. But thank you everybody for your presentations. Really great job today. It was great hearing all this, and continue working on this great and amazing research that you guys are doing. So. Awesome. And then Heather, I noticed that you're in the room. So will we just get kicked back in the main room session then?
Yeah, I think the idea is that everybody will be moved back at 11:20, and Ken can confirm that. That's.
Oh man, was I actually early? That never happens. That
Yeah,
never happens.
You are early, Nathan. Yeah.
That never happens. So for the PT students that presented today, I know one of you had mentioned you were a first year. I believe it was Jessica, that you were a first year student. What about you, Emma and Anna?
We're both first years as well.
Okay. And it's usually roughly a 3-year program then?
Yep, 3 years, and then we do summers as well too.
Okay, cool. So if there was the possibility for a research study, then that seems like something that you guys would be reasonably well timed for. So awesome, very cool.
I do have a question. It just popped back in my head for that last group. I know they mentioned like having difficulty recruiting participants, having only 5 participants. What, maybe you guys can speak to like what barriers, if you didn't touch on it, I apologize if it was already mentioned, but like what barriers you guys came across with recruiting participants?
A lot of that has to do mainly with the healthcare providers being so busy. And I know that was one of the main ones, but I guess there could also be, we actually had an interesting point of data where it said that there's, for example, only having 3 movement disorder specialists to treat 10,000 people with Parkinson's disease. So maybe even just a shortage of availability of healthcare providers to reach out to in the first place. Could anyone else speak on this actually? In my group?
Primarily it was time limitation, I think, with a lot of the providers. I remember one interview with a physical therapist who was meeting with us on his lunch break, and he was very rushed. He had limited time. So even though we are compensating participants for their time, I think that they just have so little time available. And yes, there are not that many movement disorder specialists to reach out to in the first place.
Yeah, I mean, what everybody was saying here was really the main reason. We ran into a lot of issues, and it's the healthcare providers kind of across all the different disciplines. So we reached out to speech language pathologists, physical therapists, occupational therapists, movement disorder specialists. I think even neurologists to some extent. There were some limitations of who we could contact, but it was just time. They didn't have the time, or they're already being hit up by a bunch of other companies or businesses to try and get their insight. And so they really are just that time swamped. I mean, I think the ones that we were able to recruit, a lot of them actually worked in community-based places and kind of had a research component to their position, or there were some academic speech language pathologists and whatnot. And so yeah, I mean, that was the biggest barrier, is time and people being open to research and willing to take the time to help with it, and having the time to help with it.
I think that's an important point. If the clinician is not involved in research or academia already, they're a little less likely to want to participate in something like this.
It kind of reminds me of Jessica, I believe it was you that said it, but just the difficulties of trying to get measures during clinical sessions highlights this. There's just not enough time to do everything, and you're trying to squeeze in everything as best you can. So yeah, a great question, Jessica.
Some of the barriers that we found to this intervention were there were very limited studies on pediatric care. The primary focus was on both adult care and also the opinions of providers, not necessarily parents. The objective of this study was to investigate challenges with the use of telehealth and various recommended strategies for promoting telehealth among parents, specifically during the COVID-19 pandemic. This qualitative study investigates the benefits of and the barriers to telehealth use and the recommended strategies for promoting telehealth use among families during the pandemic. Our recruitment methods included using convenience sampling in various pediatric offices, farmers markets, and the University of Michigan-Flint. We primarily used flyers that were distributed in all of these locations. Those who were eligible for participation in this study had to be responsible for one child under the age of 18. Now, parents could complete either the interview portion or the survey portion, not both of them. So on this slide, I just included the 2 flyers that we used for the entirety of this study. The flyer on the left was used for the majority of the study, and this has information on both the survey and the interview. This was the flyer that was posted in pediatrician offices, the farmers market, and various locations around U of M Flint. The flyer on the right, this was sent to all U of M Flint students via email, and this was only for the interview portion of our study. For the survey,
Participation was completely voluntary. And the caregiver's name was not linked to the survey results, so it was anonymous. In this survey, we included 4 open-ended questions, which included: how would you define telehealth? What, if anything, keeps you from using telehealth for your child during the coronavirus pandemic? And if you could design a societal program to make telehealth more accessible, what would you design? Now, caregivers who decided to do the survey, they were reimbursed with a $25 gift card. For the interview, again, participation was completely voluntary. And prior to the interview, participants or potential participants were emailed a consent form, which described the purpose, duration of the study, the benefits, potential discomforts, and the compensation for the study, which was, in this case, a $35 gift card. These interviews were completed and conducted over Zoom, and they were transcribed by a researcher on the team. In addition to the interview itself, caregivers were required to complete an anonymous demographic survey just to analyze the data that we were receiving from the interviews. So for the results of both the survey and the interview, in the survey, we discovered that from the demographic survey that the majority of survey participants were Caucasian, the biological parents, and either single or married. In the results that their responses to the open-ended questions included the benefits and challenges of both synchronous and asynchronous telehealth, and they provided recommendations for expanding the use of telehealth. In the interview, slightly similar, but most of the interview participants were, again, Caucasian, married, and all of them, in this case, were biological parents. Those who, in their open-ended answers to the interview questions, they stated that those who did not have access to telehealth services wished it was available and would consider switching their provider to make sure that their child could get telehealth services. These caregivers also offered various recommendations on increasing the access to telehealth, including improving the technology and also offering various types of education for caregivers. These were some of the caregiver responses that we received from both the survey and interview open-ended questions. So one caregiver, a benefit that they stated was that they really appreciated being able to send a quick message and hear back from the pediatrician without having to pay for an additional visit. One challenge that a caregiver mentioned was that they believed that telehealth could potentially leave room for a misdiagnosis or a potential error just because their provider was not able to be up close and personal and directly see their child and whatever condition was happening. A suggestion that was mentioned by a caregiver was making their telehealth portal, like their patient portal, a universal platform instead of having multiple different portals and areas to find this information, having one place where the providers could share results and also so the doctors at maybe various healthcare facilities could interact with one another easier and also get the information to the parent and the patient. One potential improvement was a suggestion on having an option to upload pictures prior to the visit. So that way parents, if they had a way to take perhaps a more high definition picture, they would be able to take the picture ahead of time and send it to the doctor. So that way, in case their video camera was not as high of quality as their phone camera, perhaps, they would be able to get that picture to the doctor and hopefully get a more clear diagnosis. Some of the benefits that the caregivers mentioned of telehealth was that it was cost effective. They appreciated that it saved time, specifically travel time, and also the cost of vehicle fuel. Others appreciated the minimized exposure to COVID-19, especially at the height of the crisis. And specifically with asynchronous care, they appreciated how convenient asynchronous care was in being able to interact with their provider either through a text message or an email. Some of the challenges that caregivers stated about using telehealth was they felt that they missed the in-person interaction and being able to ask questions in an in-person format, and that in some cases, the telehealth version of care did seem slightly impersonal. Others struggled with various technology issues, being unable to upload information if needed, or even just logging into the visit, or the visit ending earlier than they had anticipated due to various Wi-Fi issues or technology issues. Others, again, were concerned about misdiagnoses and not being able to get the proper care for their child. And some had difficulty navigating the patient portal and finding information that way. And that would have been the asynchronous version of telehealth. Others, again, with the asynchronous telehealth, struggled with misinterpreting messages between both the provider and the caregiver in order to get the proper care for their child. Some of the suggestions that caregivers stated for improving access to telehealth and also just improving telehealth in general, they suggested potentially time-saving strategies like having one password that would be used for all of their accounts instead of multiple passwords, saving time and also just mental energy in trying to remember all of the various passwords for maybe their multiple children and themselves. Others mentioned starting educational programs for parents on the benefits of telehealth, especially in underserved populations, and just providing a way for parents to learn more about how to use telehealth and just the benefits overall. Others would have liked to have seen, especially during the pandemic, more telehealth appointments available through their provider, as some struggled finding appointments available, or even, and many also did not have providers that offered telehealth services. So just having the availability to have telehealth was a big issue for many parents as well. Some of the limitations of this study included our small sample size and the lack of interviews with children and adolescents. So the sample size for the interviews was under 25, and for our survey, it was under 120. But within those, within both of those survey sizes, we were able to gather quite a bit of data, and hopefully with what we were able to discover with both of those interventions, or the survey and the interview, we hope that we're able to improve some things with telehealth through their feedback. The implications for this study, we discovered that there is a need for various telehealth interventions at the individual, community, healthcare organization, and national levels to improve access to telehealth and the benefits of telehealth for everyone. Telehealth does seem to fall short in a few areas, like being accessible and equitable, but through suggestions like media campaigns that that caregivers suggested, and also interventions, we believe that telehealth does have the potential to improve. So my references. And this study was supported by an internal grant of the College of Health Sciences at the University of Michigan-Flint. Thank you.
Thank you very much for a wonderful presentation. Are there any questions?
I actually have a question. And you might not really know this because it might not have really mentioned it in the source, but I was wondering why you would ever use only one or the other, survey or interview. Like if you had the option to get both, why would you not get both?
I think our reasoning for doing that was to provide a variety of caregivers providing their opinion in this survey. So having the option to only do one allowed us to address various concerns on both sides of the, in both types of interventions.
Thank you.
Yes, and I think that we did both interviews and surveys, correct? So there were about 24 interviews and then 105 surveys. They were both with open-ended questions. So we did both. One question I have is, and you conducted many of the interviews. What was your experience conducting them? What suggestions do you have for students who may conduct interviews? And was anything said about how to involve children more in the telehealth experience? We often talk about the parents. What do you think about children?
Well, to start, I think doing the interviews, it really, the overall experience really opened my eyes to just some of the inequalities that do exist in healthcare, specifically in telehealth. And I think there's so much that we can improve in that area. And a lot of parents, they, in the times that they did suggest ways to involve their child, I believe a few of the caregivers, it was specifically in situations like therapy, like speech therapy. They said they really struggled with finding ways to involve their child in the same way that they would in an in-person situation. And they weren't exactly sure how to provide that same type of environment. So I think there are definitely ways that could be improved, especially with perhaps providers being able, in that type of situation, being able to send maybe various resources to the home or things ahead of time that would help prepare the caregivers to assist their child in that type of setting, and also just have the children be more engaged in that type of setting as well.
Thank you so much. I thought again that your study was very valuable because you did interviews and surveys and you asked open-ended questions. So you got those deeper thoughts and opinions. So thank you. And in your study, Taylor, the parents recommended strategies for improving knowledge about telehealth. And so with the next intervention, Jada and Mark, they're continuing the research that you did. And so it is a pleasure to present to you Jada and Mark. Jada is pursuing an undergraduate degree in public health here at our university. She collaborated on a study on the influence of the Flint water crisis on family health. She is a Go Blue guaranteed scholar, and she plans on pursuing a law degree once she graduates. Mark is currently pursuing his Master of Public Health degree. He is graduating in the end of the fall semester. He earned a bachelor's degree in healthcare administration here, and he has assisted in the development and implementation of an intervention to promote COVID-19 vaccination in a church. Welcome to both.
Awesome. Thank you for having us, Dr. K and Mr. Andre Lewis. It is a pleasure to be here with you all. For our presentation, it's kind of going to be a little bit like class. We're going to have some videos to go through. And, you know, I'm always a guy with dad jokes, so we're going to have a pop quiz in there as well. So well, let's go ahead and we will begin. I believe I can share my screen. Wonderful. Can everyone see this okay?
Yes.
Awesome. Wonderful. Thank you. As Dr. K was saying, we are looking at bridging the gap between telehealth and parents. Our intervention was based off of the findings that Taylor ended up initially conducting, and we wanted to help bridge that in any way possible through the feedback that Taylor had gotten. So to begin our overview slide, we are going to be giving you a definition of telehealth and telemedicine. A lot of times those are interchangeable, so we just wanted to clear that up. We're going to be giving you the benefits of telehealth from the patient and the physician point of view, because they are quite different in some aspects. We're going to give you educational videos on the importance of telehealth. These are going to be coming from sources of patients who have used them in a variety of telehealth areas. We're going to talk about confidentiality issues surrounding telehealth, the development of an online intervention to increase knowledge on the benefits of telehealth among parents, and using strategies such as the educational videos that we will go into soon. And
definitions provided by American Academy of Family Practice.
regarding telehealth versus telemedicine would be that telehealth refers broadly to electronic and telecommunications technologies and services used to provide care and services at a distance, while telemedicine is the practice of medicine using technology to deliver care at a distance. A physician in one location uses a telecommunications infrastructure to deliver care to a patient at a distant site.
And now we're going to move into the physician point of view. And with the physician point of view, for them, a lot of this has really started up with COVID. They really saw the impact that it has played from the physician side, and they have demonstrated that it's more convenient for the physicians. Physicians can see more patients in a day versus inpatient. Physicians can stay in contact with patients through patient portals. Physicians can work from the comfort of their home, and there's a lower risk of getting infectious disease. And Dr. Azim Roy, he is a physician. Out of Ohio Sleep Medicine Institute, and he's just going to talk real quick on some of his benefits he has experienced.
Probably the one thing that I like to highlight on the pandemic side that I think we've seen as a medical community that really has revolutionized or kind of modernized our approach to treating and managing patients is to be able to treat everyone virtually. It's been a huge change for us, a big positive. We have patients who used to drive, hours to see us and see us for 20 minutes in the office, and they've had to spend their whole day or half a day and take off work or whatnot. And now, it's a minimal disruption to their work day or whatever, whatever they needed to do. They can see us quickly. And we can pretty much do everything we were doing before all virtually now. So I think it's not that we want a pandemic to go through every few years, but this definitely I think has been a huge win on the medical side of being able to give more patients access and be able to handle care in a much more efficient manner.
And now we're going to be Jada's going to be sharing the patient point of view.
The pros of telehealth for patients were that patients found peace in no longer making long drives. Healthcare has become more accessible and doctors can answer questions faster. Family is also able to be involved in their loved ones' health and provide support. And the cons would be that internet affordability impacts who uses this procedure within personal economic means. Furthermore, patients appear satisfied with their experiences so far. Concerns remain from higher preference for an in-person appointment to technology not being a secured resource for healthcare to having unruly internet services.
I hope you guys have your thinking caps on because we're about to jump into the videos, and over to this left-hand side, you're going to be seeing the questions right here. So go ahead and look for those while we're going through this. So here is the first one, and this is from a veteran's point of view using telehealth.
Thank you, Mr. Kylan, for being here. I appreciate you sitting down and letting you share your experience on the telehealth and your experience with it. So can you go ahead and tell us a little bit about what it's done for you, what you enjoy about it, and your overall experience, if you were to tell others?
I sure will. Well, to start, since the pandemic and stuff, a lot of the doctors nowadays are doing it, online and the visuals and stuff. And for a lot of the different clinics and that you go to, the different areas that you go to, it seems to work pretty good because people, they don't have to get out there, they don't have to take the concern for the the virus and all that stuff in there. The it worked really good. But now you have certain different ones, I've been seeing a person for speech and memory for a long time. And I guess one of the big setbacks, and when you have something like that, is if they're asking you to say something or repeat something or write something down, whether it's the internet or however they have it set up there, you will see the visuals and stuff a lot, but what you won't see is you won't hear the audio part of it. So they might be tasking you to do something, and it does. So it kind of breaks that part in there. So if you're doing like a question and answer type thing for a doctor or a different type clinic or something, you will find out that that makes it a little harder. But if you're going to see like your primary care doctor and it does go down for some reason, you can always go back up because it's just going to be a question instead of, okay, let me give you 7 numbers. You got to repeat them back and you didn't hear the numbers to start with. So it worked out pretty good. I got a lot of friends that have also used this and they're happy with it. Like I say, one of the shortfalls is the system in place, but also it comes out there for mental health. For some people, I would just speak on that for the veterans and stuff. And myself, it's kind of hard because you don't know who's really in that room, and you're having some experiences and stuff to go with it, and so it's not a comfortable thing. So that would probably be one of the areas I would see it would not work out well. And I've had many friends have done it, and it just didn't work out because it was a comfort thing, and then you don't know who's listening. Then you don't know what. I had one buddy. I had a doctor that got on a treadmill, and it was just psychiatrist. She started working out on her treadmill like she's trying to do an appointment with him, which is it sets it up there. You're trying to have somebody doing it. But overall, it's really good. It's a lot easier for the families and stuff like that because instead of going down there, some doctor's office won't let you bring in a spouse or somebody else because the pandemic, so they have to wait outside. As you can tell, Michigan now is in the winter part of it, so that part's not good. So this way you can do it. Now your spouse be right there with you, or somebody's helping you out on your stuff, which is a great benefit. But then you can catch it. It's like, okay, got it, and you can clear up. So you can take notes while you get notes, and it makes it better because
So going through, what were a couple of medical services that Sergeant Major Pylon had stated that he used during his experience with telehealth? If you guys are shy, you can type them in the discussion area, whatever you're comfortable with.
Towards the end, I heard mental health, right?
Yes.
Yes. Okay.
And the
Speech
other.
therapy.
Yes. Yep.
Speech therapy.
That is correct. And what did he mention were 2 benefits of using telehealth?
One was that he liked being able to have his spouse with him for the meetings.
Yes. And
Lack of transportation barrier.
Yes.
Also being able to take notes.
Absolutely. Great job. We're at 100% so far. On to the next one. We are now going to see the telehealth experience from a patient's point of view, but also who is a nurse. So be able to see it as a medical professional, but also as a patient. So here is the second one.
Hello, everyone. My name is Mark Pyland, and I am sitting here with Ms. Danielle Harris. She has volunteered to tell us about her telehealth experience. She's going to tell you a little bit about herself before we begin. And before we start, Danielle, how are you feeling? Are you excited to be here?
Yes, thank you so much for having me, Joe. My name is Danielle Harris. I'm a bachelor's prepared registered nurse, and I decided to do the interview. I have not worked with telehealth as a healthcare provider, but I have been a patient when we use telehealth.
And that's what I'm excited to learn about today is knowing from your professional side with the healthcare, but then also using it as a patient. So as we begin, can you tell me a little bit about your experience as it? What type of service did you have?
Okay, yeah, so sure. So basically what happened was I had got a cough and I wanted to see my provider, but they didn't have any available appointments. And they had the telehealth appointment available, but they had no in-person appointments available. They were backed up for months. So I decided to go ahead with the telehealth. One of the things that I don't personally like about the telehealth is being a healthcare provider. I know that one of the most important things about assessing the patient is you need to have that patient there in person in front of you. So like if I'm going in and I'm telling the doctor that I'm having this cough and I'm feeling short of breath, like it's important for them to be able to listen to me, like listen to my lungs, listen to my heart, see what's going on. Telehealth takes that away. You can't do those things. So technically there's no objective information there. It's only subjective because it's all that the doctor will know is what I tell them, because he can't physically get any information on his own because I'm not there with him personally. And I feel like for a lot of patients and for a lot of people, that's a disadvantage because especially for something like shortness of breath, like if I'm telling you I'm having shortness of breath, like it's very important that you listen to me from a healthcare provider, point. You need to be able to listen and hear exactly what it sounds, because shortness of breath for me, I can say, oh, I'm having shortness of breath and I don't know what that means. But if you listen to me, you can hear that I'm wheezy or I sound like I got crackles a little bit. And those types of things are very important, because if I'm wheezy and I have a history of asthma, then you'll know, oh, she's asthmatic, maybe she needs her inhaler. But if I'm having crackles, then that can mean that some type of fluid is on my lungs. But you wouldn't know that just from me telling you that I'm short of breath, right? So that's one of the things that I don't like about it. One of the things that I do like about telehealth is it is convenient for those people that don't like to leave their home and go to the doctor. So if, for example, if I was calling, I was saying, oh, I'm having shortness of breath and I'm out of albuterol, which is my inhaler. Then the doctor would be like, okay, she has a history of asthma. She's out of her albuterol. I've seen her and I can just put the prescription in, versus me going all the way to the clinic just to get a prescription, right? So those are like a few of the things that I found from using the telehealth communication.
So the second for the second interview, was objective information available over telehealth? We got a couple ahead.
No.
There you go. Yeah, that is correct. And that is one of the downsides to it is, you can't do like a physical examination like you would as in person, but it can also be a good thing. Like Danielle was saying about, your inhaler or something, if you have to have a refill or smaller type appointments that you normally have to go in for, those can be taken and those can be done virtually. So those can help as well. And our third interview is going to be with our own Miss Caitlin. She's going to tell us about her experience when she first had her children using telehealth. And our questions are, did Caitlin use an app for a telehealth appointment? And then what did Caitlin say were her favorite parts about using telehealth? And I know, Caitlin, you might know these answers because you gave them, but let's go ahead and check this one out.
And Mark, excuse me.
Yes.
So after this interview, then we can go to the next presentation. It's wonderful. I love the videos. Thank you.
Hello, Caitlin. I would like to first thank you for volunteering to be interviewed, for telling your experience about telehealth as a new mother. We are using this for an educational course to help get a better insight as to how new parents use telehealth and telehealth services. With me, I also have Jada, who has been helping me on this project and the creation of the class. So before we begin, can you tell me, when did you use telehealth?
So I started using telehealth in late February of 2020.
Late February. Okay. So that was right before the pandemic time then, right before it started up. So were you using that, were you pregnant at the time, or did you have your child at that point?
They were a couple weeks old. I think we only got to go in person for their first and second visits. And then it switched to telehealth right after.
And leading into that one, Jada, do you want to ask her a little bit about the types of services that she used?
Yes, what services did you use?
So, okay, so do you want to know like more about the app for this question, or about the kind of care they could give me?
It could be the care as well as a little bit of the app if you want to hit on.
Okay, so they didn't actually have us use an app. They would text us a link, and then, okay, so they would call you. They'd call me. I would answer. They would kind of check in, like that was your nurse check-in. And then they would send you a link. You'd answer the link, and then you would speak to the provider.
Speak to the provider directly?
Yes.
Awesome. So it kind of sounds almost like I'm imagining it like going into a doctor's appointment, and then instead of that pre-check-in area, you got a text message, and then after they sent you back, and then you saw the physician, kind of like if you were in person.
Yeah, like it was kind of like the nurse would check me and ask about symptoms, temperature, weights, and then I would get the link, and then I would actually speak to the provider.
Oh wow, that's awesome. And then what did you enjoy most about the telehealth, the experiences and the services that you did use?
So I actually have twins, so it was really convenient to not have to pack up 2 kids and take them for something as simple as, just a check-in to make sure they're okay. And then we didn't have to expose ourselves to any more risk than was necessary. And it was honestly really convenient in general.
That's awesome. Yeah, you definitely sound like you have your hands full with twins on that one. But,
yeah.
Awesome. And I know for time, this can be the last bit of it. But what were the two areas that Kaylin said that were her favorite about using telehealth?
Not having to pack a lot of things for the children, being more convenient.
Yes.
And the exposure of pathogen to going doctor office.
Yes.
Thank you so much. Yeah, we will start moving towards the next presentation, but I really appreciated your presentation because you presented both the advantages and disadvantages of telehealth so that patients can know both and can decide what is best for them. So one question is, what are your plans with these videos that you have been collecting? What is the next step in your research?
Right now, our next step for our research is we're going to be creating an actual course on it for parents through Taylor's information that she has gotten. So we're just continuing to go through our literature, building on that class. And right now we're just in those initial stages just to pan out because there was a lack of information from the patient's point of view in terms of what currently exists within literature. We got a lot from the physicians, so we're trying to bridge it for more of the patients because, our priority population is going to be patients using it. So that is our primary objective going forward.
Thank you. Any other questions?
Yeah, Mark, excuse me, if you already addressed this, my apologies for missing it, but do insurance companies tend to look at teleconsultations differently than in-person consultations?
With those ones, as far as what I have found so far, some are on board with telehealth, and then some are not as lenient or not likely to use it. And I know that was one of the things that came up in the study that Taylor did was they wanted something where all providers could have some sort of telehealth. So a lot of it from the physician side, it still feels like it is missing for them to get on board with, if that answers your question.
It does. Thank you.
Yeah, thank you.
Thank you so much. So now that we have been talking more about the perspective of parents, we will switch to talking about what providers think about telehealth. And our next presentation is by Miss Bushra. She is currently in the Master of Public Health program. While in this program, she co-authored a publication in the Journal of Aging and Health on cognitive impairment and hospital admissions. Prior to joining U of M Flint, she worked as a dentist and a public health officer, and she has worked on many interventions to help with preventative care for dental health. Welcome.
Thank you, Dr. K. Now I'm starting my PowerPoint presentation. Good morning, everyone. My name is Busha Kausar and Dr. K already introduced me. And the presentation title is Satisfaction with Telehealth Among Medical Providers During the COVID-19 Pandemic. Before getting the presentation, we need to know what is telehealth. According to World Health Organization, telehealth is the delivery of health service where the distance is a critical factor. Providers use technologies for the taking information and diagnosis the disease and giving the treatment for prevention of the disease. Advantage of telehealth during COVID-19 pandemic. Reduce exposure to pathogen. Long wait time in the waiting room of doctor. Some germs may spread like flus, other virus. So telehealth keeps patient at home, avoiding exposure to virus and germs. Follow-up care for babies and children. Babies are more likely to get germs very quickly, and so they have fever, rash, very other things frequently. So besides rely on the internet service, we can call the doctor using telehealth and get the good result for the babies. Medical access for people in unserved urban areas. For people who live in miles from the nearest medical facilities, medical provider can an opportunity to see the doctor. And this state like Michigan, there's winter is very poor situation in driving, like high inch of snow. So this telehealth is much convenient to go better than the doctor office. Reduce medical overhead costs. For doctor, telemedicine helps lower office costs as a record of no health desk. Limitation of telehealth during the COVID-19 pandemic. Telemedicine does not allow the doctor to take blood or urine sample. Some patient who has the chronic disease, they need to do some sample test. Telehealth doesn't allow to do this. So it's the most important limitation. Doctor cannot use a stethoscope to listen to the heart
Bushra, apologies, you're breaking up just a little bit. I don't know if maybe you could be closer to the microphone but it's fine. We can still hear you though. Thank you.
Can you hear me now? I'm sorry for that. So in telemedicine, doctor has to rely on the virtual assessment. They cannot feel the patient, they cannot assess the patient. So some bias can happen in this procedure. Insurance coverage or reimbursement is not guaranteed and varies from state to state. Children who have a service like speech therapists have trouble contacting with the therapist remotely. Objective of our studies. In the COVID-19 pandemic has spread in such a way that it's created a long-lasting impact on healthcare system globally. More provider wants to do the telehealth services than the office visit. The purpose of our study is to perform a systematic literature review to understand healthcare provider satisfaction with pediatric telehealth. For these studies, the method we have chose, and that is systematic literature review, and we follow the PISMA process. We use the telehealth, the keyword telehealth, telemedicine, telehealth satisfaction, provider satisfaction, pediatric telehealth, and provider in PubMed, Cochrane, and PMC database. We located 3,254 articles. Out of those, only 7 studies focused on satisfaction with telehealth among pediatric providers. In these studies, they have done the cross-sectional studies and some descriptive surveys, and the sample size was ranged 41 to 81 providers. For the method, here's the flowchart of these studies. We in 3 steps. There is identification, screening, and included. For identification, we search in database, that is PMC, Cochrane, Google Scholar, and there's the search word pediatric and telemedicine COVID-19 and March 2022 to November 2021. And we got 3,254 results. Among this, we removed some because of some duplicate records, and some does not match this. We removed, and the screening result is 2,806. From that, records excluded after reading the abstract and title, 2,708. Full text article assessed for eligibility is 97. From the 97 studies, we excluded because of duplication, not the time frame, and not in the pandemic time, and also the not in satisfaction. The final outcome is 7 studies. Results. In this session, I'm talking about the 7 studies that we do in this for the provider satisfaction. The first paper from the Jacqueline, 69% of providers shows despite having hesitancy initially, among them 21%. I'm sorry, I'm not seeing the. Yeah, 69% of providers showed interest despite having hesitancy initially. Among them, 21% of providers would prefer pursuing virtual appointment among of the time. The second paper of PHIL shows that 88.6% providers felt prepared to utilize telehealth method to provide receive perinatal care. 83.1% provider reported being satisfied. Among them, 92.2% of providers were interested in continuing virtual care after the pandemic. The third paper shows that 72% of providers were highly satisfied with their telehealth visit. Among them, 85% found the visit to highly convenient. The fourth paper, that is Laskin 2021, the lack of physical examination and face-to-face contact seems to be major limitation to the surgeon. Result continue. The DIAS 2021, the paper shows that overall most clinicians, 81.2% want to keep using telemedicine in future without pandemic. Hall 2021, the paper shows that the percentage of physician therapists who provide virtual service rose to 47% when they have previously been around 2% as a result of pandemic. Hall 2021 paper shows that 93.3% provider with previous telehealth training felt confident, and 83% of these respondents claimed they would be interested in continuing telehealth service. There is some limitation of the studies. Some studies were conducted early in the pandemic. So when the pandemic is COVID-19 started, the telehealth is not using that much. So they couldn't understand the essentialness of the telehealth that time. There are selected group pediatricians who so that finding may be responded to all the pediatricians. All respondents were trained in technology being used and may have held more positive attitude towards the telehealth. Most of the research was conducted in USA. There are limited perspectives from other countries. Conclusion. Satisfaction with telehealth among providers was high. Barrier is ongoing utilization, including lack of in-person assessment. Technological limitation was not viewed as a barrier by respondent. Telemedicine can fully replace face-to-face appointments. Implications for practice and research. Telehealth implications strategies. The best support provider in online treatment delivery should be identified, and proper training should be provided for best practice. Studies shows identify how increased use of telehealth might change access of treatment over the time. Future work is needed to understand the implication of telehealth, patient outcome, healthcare utilization, and associated costs. Thank you. Any questions?
Thank you so much. Are there questions?
Oh, Caitlin, do you have a question?
You can go ahead first, Mark. I'll ask my next.
Are you sure? Bushra, I just wanted to start by saying amazing job on that. It was very well put together. And I did have a question in regards to the specialties. I had seen that you had found out that physical therapy went from 2% to 47%, like with the pandemic. Were you able to find like other specialties, like if they had changed as well as a result of the pandemic? Oh, yeah.
Mark, you're breaking. I'm sorry, my internet connection is not good.
Oh, I'm sorry. Did you find other specialties similar to like physical therapy that had increased with telehealth use as well?
I am in this paper, I have found only that, but we did other research for the satisfaction in that they have increased the percentage of having provider satisfaction.
Okay, awesome. Thank you.
So I have a kind of combined question for Mark and Bushra. So, Mark mentioned there's no objective care available. And so would either of you say there's probably a higher risk when it comes to infants? A first-time mother isn't going to know what this rattling in a baby's lungs is. So I like Mark mentioned, that's the downside of telehealth. So then when you consider infants in it, how could we get it? How could we help parents understand better when it's serious?
It's like I mentioned in the advantage, and also that there is when baby and babies have so much they are very prone to have disease firstly. So if you wait in the waiting room for the babies, the germ like flu and other virus can affect the babies more quickly. So besides going to the doctor first, we can use the telehealth first. And if like the providers say then telehealth, that you have to come and we have to do the physical examination, then we can go. But we have the chance to do the telehealth first, using the telehealth and says the all symptom of the babies, that baby has a rash or fever and this kind of thing. Provider can give us some medicine or some advice. And if providers think that no, it's not, we have to see the baby, so we can go. That's the advantage of the telehealth, using telehealth.
Thank you very much. Thank you so much, Bushra. I really appreciated that you focused on the providers, and it seems that even though providers are highly satisfied, we can develop interventions for providers as well to improve their comfort with telehealth. For our last presentation, and I apologize, we have probably about 9 minutes for the last presentation. I'm not sure if we will be discontinued exactly at 11:20. Our next presentation is by Miss Caitlin. She's a healthcare administration and public health student, a member of the Epsilon Phi Delta Honor Society. She has actually organized an event as part of the Epsilon Phi Delta Health Administration Honor Society, and this is her second semester in the Europe program. Welcome.
Hi, thank you. I actually haven't shared my screen yet. Can everyone see it? Yeah. Yes.
Yes, we can.
Thanks, Mark. I saw those thumbs up over there. Okay, so my presentation is on satisfaction with telehealth, but among healthcare organizations as a whole instead of providers or, patients themselves. I actually had a lot of help with this from Taylor and Bushra, especially when it comes to our sources we use. And I do know we're short on time, so I'll probably do quite a bit of paraphrasing. So the big roles of telehealth were to reduce the risk of COVID-19 for patients. And then there was a lot of different apps or methods that were used. They included phone calls, mobile health apps, like FaceTime video calls, stuff like that. And then some organizations even use a messaging system. So because of COVID-19 and how quickly it onset into the healthcare community, we had to implement systems quickly. So this first study was to analyze how it really affected healthcare organizations and providers and any lessons that were learned from the switch to telehealth. So the methods used in this study it was a qualitative study. Interviews were used on 2 to 3 patients from each organization. There were 30 participants total from 12 hospitals spread across the US. Staff members that were interviewed included nurses, administrators, physicians, and the caregivers as well. Some benefits they reported were the risk of contracting COVID. The consultations were nice for the patients who were more remote and didn't want to have to commute to the appointment. And then overall, there was an increased access to care by utilizing telehealth. So limitations were when you consider increased access, the limitations were for non-English speakers because it was difficult. It was just difficult for them to, one, use the app and then to communicate with their providers online. And then, as Mark mentioned, there is no objective care. So, you can't do physical exams, MRIs, and those things. Some patients reported being overwhelmed by excessive communication notifications, unorganized repeated text messages and emails. And this would include just sometimes they would get notifications from things that were unnecessary, and this would be frustrating to them. And then there was also lower quality communication was reported in certain incidences. So that was another limitation of using telehealth. The conclusion in this study was basically that as a result of the major shift, it was beneficial for pediatric hospital care and provided an extra form of protection for patients. And I didn't really write this down, but it did say that there would need to be adjustments made, alterations to ensure that it was used properly and adjusted well as we continue to use it past the pandemic. The objective of study 2 was to determine if the COVID-19 pandemic would result in an increased rate of use for telehealth provided care and reduce the impacts of previously reported virtual care barriers such as regulation, use, and reimbursement for pediatric diabetes. The methods in this study, this was a longitudinal study, so the first survey was administered in 2020 and it was repeated again in 2021 after they'd had some time to get used to the adjustment. The survey included questions regarding the use of telehealth, the tools required for telehealth, and reimbursement, which Dr. Andre Lewis did mention. He had a question regarding that. 49 diabetes centers were included in this study. So I just feel like I should clarify that where the study concluded this area, they did kind of have telehealth at first before the pandemic, which is why the study was already getting ready to get done. But at the beginning of the study, only 10% of the hospitals had a dedicated portal for telemedicine. The second survey concluded that this had risen to 20% by the difference between 2020 and 2021. Generic download portals were the primary tool used for telehealth appointments, followed by email, phone calls, and messaging. And back to the reimbursement, initially only 32% of centers were able to receive reimbursement for telehealth visits. But after the second survey in 2021, it had risen to 62% in some regions. Benefits of the utilization of telehealth included the elimination of in-person exposure and less strain on hospitals, which I know is also a big topic in the United States. And then providers, the limitations of the study mentioned that hospitals suffer from revenue loss when telehealth visits are not properly reimbursed. And this was probably a bigger issue in the beginning. Like I said, their reimbursement had risen by the second study. And then conditions that may have resulted in equitable care included patients having a lack of internet access, and then reimbursement. Okay, yeah, I'm gonna go into the next one today. We got 2 minutes left. As a result of the COVID-19 pandemic, the use of telehealth has evolved to a powerful and sophisticated method of healthcare delivery. Continued use of virtual care methods should be considered, as it is a valuable asset in the pediatric healthcare community. This is just my final conclusion. There's great potential for healthcare or telehealth and healthcare services. While there are some limitations, the benefits are substantial. So adjustments will just need to be made to help make sure more equitable access for everybody by the end. Here's my sources and any questions.
Thank you very much. Any questions? I really appreciated that your presentation focused on healthcare organizations, and so there was this great transition between Bushra's presentation on providers to yours, looking at the entire healthcare organization. Thank you, everybody. I learned a lot from each of you. Thank you for the thoughtful questions, and have a nice day. And we can now return to the main room.
Thank you, Dr. Tiff. Thank you, everyone.
Thank you, everybody. I really enjoyed your presentations.
Thank you, everyone.
Have a nice weekend.
Thank you.
You as well.
Thank you, everyone.
Okay, and then give me 1 second. So I'm just going to go ahead and share the screen. So I have all of the slides up on one PowerPoint. And so if you can just signal to me when to change your slides, this was just kind of the suggested way to do it. So if that works for you guys, and you should be seeing the first presentation, the I Know Super 6. Is that what everybody's seeing?
Yep, I see it.
Okay, great. So the first talk will be the I Know Super 6. And Jessica, you're the presenter on this, but it's also some work that's been done in collaboration with Sean. Pardon me on the last names, Hurlbert and Amanda Woodruff, if I'm saying those correctly. And this has been faculty sponsored by Amy York and Susan Trojanowski. So awesome, cool. Well, I will go ahead and give the floor over to you. And do you have any preferences if people hold off on questions, or do you want them to wait until the end?
If we can wait till the end of my presentation, that'd be great.
Okay.
If there's something they want to drop in the chat, that's perfectly fine. I'll just answer it at the end if that's okay.
That is absolutely perfect. Appreciate it. So go ahead, Jessica. We're excited to hear what you have to say.
All right, perfect. Thank you so much. So hello, everyone. My name is Jessica Gerke. We've already been introduced to kind of the other authors, but I'll just kind of go through. So I am a first year student in the Doctor of Physical Therapy program at the University of Michigan-Flint. I'll be presenting this research research project titled I Know Super 6. It has a much longer title, but I didn't include it all on this slide. It's actually implementing standardized outcome assessment battery at a student-run pro bono clinic for patients with neurologic conditions. So this study was conducted by myself along with a second-year DPT student, Sean Hurlber, and a third-year DPT student, Amanda Woodruff. And then, as already mentioned, our faculty sponsors are Dr. Amy York and Dr. Suzanne Trojanowski. So you can go to the next slide. Thank you. So evidence-based practice is essential in providing high-quality patient care within physical therapy practice. Although physical therapy students and often new graduates report difficulties implementing the evidence-based practice into clinical practice. Knowledge translation, or KT, assists in the uptake of evidence into practice to improve health outcomes, provide more effective health services, and strengthen the healthcare system. The American Physical Therapy Association, or APTA, supports the use of clinical practice guidelines to guide clinical decision-making and practice. The process of knowledge translation has been used to facilitate the implementation of these clinical practice guidelines into clinical practice, and student-run pro bono clinics such as HEART at the University of Michigan-Flint provides an opportunity for students to begin implementing these evidence-based clinical practice guidelines into practice. Next slide. Thanks. So in 2018, a clinical practice guideline was published to standardize the use of a core set of outcome measures for adults with neurologic conditions. The core set of outcome measures include the following tests in this table, such as the 5-time sit-to-stand, which assesses leg strength and transfer mobility, the 10-meter walk test, which assesses gait speed, the 6-minute walk test, which assesses walking endurance and aerobic capacity, the Berg Balance Scale, which assesses static and dynamic balance in sitting and standing, the Functional Gait Assessment, which assesses balance during gait, and the Activity-Specific Balance Confidence Scale, which is a self-reported assessment of balance confidence. So these 6 outcome measures are what we refer to as the Super 6. So now we'll keep it on this slide, but now we're gonna kind of get up and moving a little bit. Of course, I feel like it's not a Dr. York presentation without moving. So I'm gonna have everyone in the audience practice the 5-time sit-to-stand outcome measure. So I'm sure everybody has a phone or maybe on their watch, there's a stopwatch app. So we're gonna get those out. We're gonna stay seated until I say go. And then when I do say go, start your stopwatch, feet flat on the floor, arms crossed over your chest, and then you're gonna stand up and sit back down 5 times as fast as you can. Simple enough, right? So I'll do it with everyone, but we're gonna start seated. And then once I say go, so 3, 2, 1, go. So stand up, sit back down. 2, 3, 4, 5. And then you'd stop your stopwatch. So it's simple as that, right? So nice job, everyone. This is really as simple as it gets, but this test provides a lot of information about a patient. So like I mentioned, the 5-time sit-to-stand, it shows us, the patient's leg strength and their ability to transfer themselves. So we'll move on to the next slide. This purpose of this study was to examine the implementation of those standardized outcome measures guided by the knowledge to action framework in patients with neurologic conditions receiving physical therapy services in a student-run pro bono clinic. Next. So that knowledge to action cycle that I mentioned was implemented as the framework to guide the knowledge translation process for this study. So the knowledge to action framework has 6 key steps. The first one is to identify problems, then adapt knowledge to local context. Then we tailor and implement interventions, we monitor knowledge use, and finally evaluate the outcomes. So the first 2 steps of that knowledge to action cycle were performed earlier on in about 2019 and were the focus of a separate presentation. So the final 2 or 3 steps were implemented in the fall of 2021, and this presentation is really going to focus on those aspects. Next slide. Student physical therapists who volunteered at a university-sponsored student-run pro bono clinic called HEART participated in this knowledge translation project. Equipment, environmental modifications, knowledge tools, and faculty clinicians were made available as resources to all of the student volunteers. Implementation of the standardized outcome battery began in September of 2021, and monitoring of the student physical therapist adherence was initiated at that time. So to examine their adherence, chart audits were completed to measure the use of outcome measures in patients with neurological conditions receiving care at HEART, which is, like I mentioned, the student-run pro bono clinic. So we had 21 documents, including 14 initial evaluations and 7 progress notes for 14 patients. And these were reviewed for the use of outcome measures. Of the 14 patients, 8 of them were diagnosed with stroke, 2 were diagnosed with Parkinson's disease, and 4 were diagnosed with some other neurologic condition that didn't fit into a specific category. Next slide. All right, so here we see results from a chart audit that was conducted in 2019. So this was prior to implementing the knowledge translation process, and also before we initiated a standardized outcome measure battery at the student-run pro bono clinic. The focus of this chart audit was to assess which outcome measures were currently being utilized in the clinic. Students evaluated 15 patients at this time, each of them having a neurological condition. 13 of them had an additional progress note, and an additional 12 had a second progress note entry. So we had 40 documentation entries total that we reviewed, and we found out that 68% of those documentation entries had at least one outcome measure used. And the 5-time sit-to-stand test was the most common outcome measure used that was recommended by those clinical practice guidelines that were released in 2018. So in total, 61 outcome measures were utilized in the 40 documentation entries. And this pie chart to the right, it kind of breaks down the outcome measures that were used altogether. So it's also important to note that not all of the outcome measures recommended by the clinical practice guideline were being used at that time. Next slide. So then now we fast forward to the end of 2021. We conducted another chart audit following the implementation and adherence to the standardized outcome measure battery at the student-run pro bono clinic. First, second, and third year DPT students were evaluating 14 patients with neurological conditions that I mentioned, 7 of them having an additional progress note. So we had 21 documentation entries that we reviewed, and we found that 100% of these documentation entries had at least one outcome measure used. And on average, 4 outcome measures were used on initial evaluation and 4 were used on progress note. So we also noticed that the 6-minute walk test and 10-meter walk test were the most frequently used outcome measures that were recommended by the clinical practice guideline. And in total, 69 outcome measures were used in the 21 documentation entries reviewed. And that breakdown is also shown to the right. So it's also good to note that all of the outcome measures recommended by the clinical practice guideline were being used in 2021. Next slide. So finally, we also looked at the DPT student success in conducting and documenting the outcome measures. So the chart to the left displays student accuracy with each of the outcome measures reviewed and documentation. So the red correlates to errors in documentation, and the blue is successful documentation. So with 5 times sit to stand, for example, the test was conducted 11 times, but only 7 of those entries were documented correctly, with 4 of them containing errors in documentation. Overall, students were 84% successful in implementing the core set of neurological outcome measures in the student-run pro bono clinic. So we graded that a B. Students received a B. Next slide. So finally, what can we take away from this study? We know that using knowledge to action framework in a student-run pro bono clinic allows students to engage in knowledge translation. It provides the opportunity for students to actively implement and modify current clinical practice. Standardizing outcome measures assessment is vital to evidence-based practice because it standardizes and decreases unwanted variation in the physical therapy examination of patients with neurologic conditions. So for our next steps with this project, we plan to modify the documentation template that we use in the clinic. We want to develop a reminder flag for progress notes in the documentation system and hopefully create a Super 6 educational module for the student physical therapists that volunteer at the clinic. Next slide. Here's the references used for this presentation. Next. And then we'd like to acknowledge that funding for this research project has been received from the Michigan Physical Therapy Association, or MPTA, and their Institute for Education and Research. And of course, we would also like to acknowledge the student physical therapists who serve the patients at heart, our university-sponsored pro bono clinic, providing patient-centered evidence-based care. And that is all. So thank you so much for your time. And I guess now we can open it up for questions.
Yeah, great talk, Jessica. Thank you. And so does anybody have any questions?
I had a quick question. So were you surprised by the results of like 84%? And for you personally, what did this experience kind of help you as like a student physical therapist, and like what you gained from it?
Yeah, those are really great questions. 84%, I don't know if it was so much surprising as we were going, doing the chart audits and going through. We did notice kind of a trend with a specific like assessment that was having a lot of errors, which was the reason why we want to kind of change the documentation template to make it easier for students to document, and then also us as chart auditors to read that documentation. So I don't know if it was necessarily surprising, but I'll be excited to see, like, once we do make those changes, how that kind of changes, hopefully makes those results a little bit better. And then as far as like what I've gained and what I've learned, it's really hard to implement evidence-based practice. Like, it's not easy. And being someone that did actually volunteer at the clinic and tries to volunteer frequently, I've kind of seen that firsthand, like how difficult it can be to implement these outcome measures and stick with them, each time, and knowing when we need to reassess. So it is a learning experience, and I'm excited to kind of see that over the years. Thanks.
Perfect, thank you.
Yes, it's definitely hard to do evidence-based stuff, especially in the clinical setting. So I did have a quick question for you. I noticed that you said that you had two individuals with Parkinson's disease who showed up at the clinic. Was it through the university, or were these individuals that were kind of in Greater Flint, I believe is what PT Heart serves?
Mm-hmm. Yeah, yep. So as far as I know, they're from the community. They're not associated with the university. I don't know because I started, as a first year, I started with PT Heart in the fall of 21. So I'm not sure if they were established like prior to that, or, how long they've been with the clinic, but I do believe they're just from the Greater Flint area.
Okay, great. And yeah, well, because I know that in conversations with PT students and with Dr. York too, that they said that they don't get too many PD cases there. So I was a little surprised, but I mean, hope that it was kind of a sign of a nice thing. So it looks like Charlotte has her hand raised. So Charlotte, if you want to go ahead and speak, if you can.
Sure, yeah. Sorry, forgot your name already, Jessica. Sorry. Great talk, thank you. And I find it very, very interesting. And I have a question about the selection of the outcome measures, like what determines which measure to to deploy or to prescribe to the patients, and what kind of tools that you use. Like would the therapist or student therapist need to be reminded of the 6 outcome measures, or there is already a checklist, or there will be, are there anything in place? This is what I'm interested in finding out, whether they actually need to recall, okay, maybe I should take this, or there are already a list right there. They just have to decide what to do.
Yeah, great question. So as far as, sorry, can you remind me the first part of your question? Why these 6 assessments? Is that the first part? So the clinical practice guideline recommended these 6 tests as being, the best to assess and examine and evaluate neurological patients. So we implemented that to standardize into the clinic. So it was the clinical practice guideline that was the reason we wanted to go with these 6 measures. And then students, there is a, because we were trying to standardize it, so there is a list, there is in the documentation system, when we know it is a neurological patient that falls under, this umbrella, to be able to use these outcome measures. We do have Dr. York, or one of the faculty clinicians in the clinic, will kind of take the students aside and say, hey, there'll be the progress note that mentions to do these outcome measures, but then the faculty clinician makes sure to talk with those students to make sure that they do the outcome measures during whether it's the evaluation or the progress note. So it's kind of useful tool to have the clinician there to kind of address, what needs to happen or kind of go over a plan for the patients. And then also there's a lot of clinical tools in the clinic for the students to have as a guide for these outcome measures. So we try to make it easy, but our plan is to hopefully make it easier with a good template for the documentation, and then also having like a little reminder flag in the documentation system moving forward. And then, just making sure that we have educational modules. So all of the next steps kind of revolve around making sure, just bringing more awareness to those outcome measures.
Thank
and
you.
Then someone else says.
And yeah, it looks like Bill had a question in the chat. Did you find the errors in the documentation just at the beginning of the study or throughout?
So the only piece that I was involved in finding the errors was in when we did the chart audits in 2021, and those charts were from September through December. So we had the initial evals and then progress notes, and they were kind of scattered throughout. There wasn't like a specific time frame where we noticed those errors. I'd be curious to see the errors that we did. Well, I wasn't involved in it, but back in 2019 when they were going through those chart audits, just to see what outcome measures were used, what the error, percentage was at that point. But good question.
Awesome, great. Well, thank you, Jessica, for that great talk and great answers. And it's always weird to give applause via Zoom, so you're getting applause on my end, and it looks like throughout the room. So thank you very much, appreciate it. And so we're going to move on to our next talk. I'm going to go ahead and try to get the share screen to see if I can get it to work fluidly. Okay, and it looks like our share screen is working for presentation 2. Everybody's seeing? Okay. Cool, so our next talk is going to be designing a mobile healthcare application for people with Parkinson's disease from the user's perspective. And so this represents the work of Kayla, Matthew, Zoe, Delaney, Abby, Quinn, Taylor, Linda, Charlotte, and myself. So go ahead for speaker.
All right, we can actually go right to the next slide. All right, Parkinson's disease is a very common neurological disorder with nearly 1 million people currently living with Parkinson's disease each day. People with Parkinson's disease can experience various symptoms, especially as the disease progresses. These are generally categorized into motor and non-motor symptoms. The motor symptoms tend to be the ones easier to be seen, such as how the person walks or moves. The non-motor symptoms are the changes in mood or affect, cognition, and sleep. These are some of the common symptoms listed, but they are not, there are many other symptoms since each person's lived experience is very different. And next slide. There are existing apps for Parkinson's disease, but most have not been tested for usability, and the existing usability data is primarily from Europe and South America. In a systematic review of 111 mobile applications for the use of people with Parkinson's disease, only one study had analyzed the feasibility and usability of implementing an app. And the existing apps are not inclusive of all services desired and needed from this population and have not been utilized in clinical practice. Each person with Parkinson's experience will be different, leading to a great variation in how the symptoms are managed, including the medications, therapeutic interventions, healthcare visits, and other recommendations. The goal of the study was to develop an all-inclusive app for people with Parkinson's disease, their care partners, and other health professionals. By including the people with Parkinson's in the conversation, we are hoping to create an app that the individuals actually find usable and beneficial. Next slide.
So we asked ourselves, what do people with Parkinson's want? What would make this app usable for them, and what features would they like? So for our methods, we had 26 people with Parkinson's disease. We recruited through Parkinson's disease support groups, and also through word of mouth, and emailed. We sent out emails. We had semi-structured interviews. So these interviews were recorded through Zoom, Participants were asked questions related to medications, interactions with their healthcare providers and professionals, and their experiences with technology. All our interviews were transcribed, and we had 2 researchers verify them, and all of our data was stored on a secure platform. So when we were analyzing data, we used the affinity diagramming process. So for this, we used lucid charts, and we took the transcriptions and we sort of put them into, you know, what people said, what really stood out to the researchers. And we put these things into broad categories so we could compare across participants.
So 3 of the primary features that came up in multiple interviews were accessibility, daily reminders, and tracking. So in order to be more accessible to people with Parkinson's disease, an easy user interface, larger font and icon size, and dictation capability were highlighted. The dictation feature would need to be sensitive enough to detect voice changes. For example, softer voice or change in pitch, which often happens with Parkinson's disease. Some of these symptoms that could be tracked included both motor and non-motor symptoms, such as UTI symptoms, fall detection, tremor, activity tracking, cognitive function, and mental health. A wearable component could also be used to track symptoms and activity, as well as sleep patterns. Daily reminders were preferred in the form of sound or written push notifications, and participants preferred daily, twice daily, or weekly input. A strong desire for access to more educational resources was expressed, as well as a social networking component. Next slide. So most of our participants were open to using technology if they believed it would be helpful in managing their disease. Lower comfort levels were reported, with some being open to classes or education regarding how to use this technology. Most had access to a person within the family that was tech savvy, and one participant had actually retired from a career in technology. Seeing trends around when medications were taken, their on and off times and activity, and being able to schedule their days around these trends can lead to less disruption to activity and less symptoms. They also expressed interest in having a one-stop patient portal to access all specialist records, and expressed a desire for their healthcare providers to communicate more regularly. Next slide. Moving forward, it will be helpful to explore the feasibility of at-home symptoms testing abilities, as well as specific screening tools that can be embedded within an app to detect cognitive changes. UTI symptom monitoring and prevention may include something that looks like tracking daily water intake and a physician-designed bladder program. 30 to 40% of people with Parkinson's disease suffer from depression. So including mental health resources within an app for people with Parkinson's disease is an essential feature. Let's go to next slide. We will continue to collect and analyze the interview data, look forward to including more care partners and participants within rural and medically underserved areas as well. Our next steps include field deployment and usability testing. Next slide. So from our entire multidisciplinary research team, we would like to thank you for attending our research presentation today. We would also like to thank our mentors, as well as the M3 funding, Flint Europe, and GSRA programs for funding this research. At this time, we can move into a question and answer period.
What would you say was the hardest part about just like overall conducting the studies and like throughout data collection?
Qualitative data is very time consuming, I would say. It was difficult to find people to interview, difficult to actually just have enough people on the research team to conduct the interviews, to conduct the data analysis, so doing the actual transcription process. We had double checks with that, so we would have multiple people that would need to go through the actual transcription to make sure that it was accurately transcribed from the video. The affinity diagramming is quite time consuming as well. So being able to have a time where everyone's available to meet up to do the data analysis in the affinity diagramming session, that was a little difficult as well. And there's so many things to analyze. So it's more time consuming than my experience was with more quantitative data.
I also think it was hard because we got great information, but really fine tuning of like, it's good information, but how does it actually apply to the app? Because, you know, it's a conversation. So they add such good information, but finding a place for it, sometimes you had to cut out stuff that's like, it sounds good, but it won't fit with an app.
Great question. Any other questions? We have a couple more minutes available if anybody else has any questions.
Can I ask can I ask Jessica a question?
Sure.
It has to do things. It has to do with the outcome measures. I'm actually thinking. I'm interested in finding out what you think about integrating some outcome measures in the mobile app that the 3 students just presented, like as a way to improve adherence to the requirements.
Yeah, that would be interesting. I'm not sure, like implementation wise, how that would work. And a lot of times, especially if the patient needs assistance with standing or walking, because a lot of the outcome measures require you to, like the sit to stand or the 6-minute walk test, 10-meter walk test, that requires someone to be there if the patient needs it. So that would be my only concern with like assistance conducting the outcome measure in some of those cases, but that would be really interesting to see how that would play out, and I'd have to kind of think about that more, maybe like pick Dr. York's brain too.
Well, and also related to that, there are some of you may be familiar with the work of Faye Horrock, who has created some sensors to be able to do this, and her team has actually created, I know at least the sit to stand has been involved, and I believe there's like something equivalent to the 5-minute walk, or a shorter version of it, maybe the 1-minute walk, but also the sit to stand with walking. And so there are some ways to do it. As far as the algorithms, I don't think they're readily available. So it would take redeveloping those since they're proprietary information, but they actually, it appears as though people have been doing it, which is interesting.
Mm-hmm. One of our, Sean on our team, he's been working on a tool to kind of help with the algorithms, like as far as calculating for students to make it easier to document it correctly, because we notice a lot of errors in like logging gait speed or documenting gait speed. A lot of people will want to record it in seconds, where we need like the gait speed actually, documented. So we see that issue consistently. So he's really developed or tried to work on this tool to make it easier for students to document some of these outcome measures, but maybe like taking that and working it into the app. I don't know. I'm just trying to think of like ways that we've tried to kind of overcome some of the issues, but yeah, that's really interesting. I'm curious to see some of that stuff that's already been done.
In the clinical setting, I'm coming from a nursing perspective, but we use something called the Bedside Mobility Assessment Tool, which assesses more functional mobility, but we have 4 levels to that. So we start off by having them do a sit and shake, and then a stretch and point, and then a stand, and then a walk. And of course, it's modified based on their own ability level, so we can modify the tasks slightly. But the way it's set up, it's very, very easy to document. And of course, PT is going to be a lot more detail oriented with that stuff, but I found the documentation to be pretty easy, like in the electronic medical system, the way it's set up. So I think having like an electronic medical system that has the assessment embedded within it and kind of set up for you is really helpful in like streamlining documentation.
Great. Any last minute questions before we move on to the next presentation? Okay, well then we will go ahead and at some time in this pandemic, I will figure out the screen share function smoothly. But you guys are seeing presentation number 3. Okay, awesome. So this next talk will entitled DPT Students Supporting Exercise Behavior for People with Parkinson's Disease. This talk will be given by Anna, Emma, and Sean, and the faculty sponsor on this project is Dr. York. So without further ado, I will go ahead and hand it over to the team.
Hi, thank you for the introduction. So again, my name's Emma, and with me today presenting we have Anna. And our presentation is on DPT students. So again, that's Doctor of Physical Therapy. We're back here again, and we're supporting exercise behaviors for individuals with Parkinson's disease. So next slide. So I know that we just talked about Parkinson's disease, but just as a reminder, Parkinson's disease is a progressive neurodegenerative disorder, and it's characterized usually by a tremor, rigidity, impaired balance, and walking. And there's obviously many more symptoms that are encompassed in this disease. And as of 2017, there was over 1 million people in the US that were diagnosed with Parkinson's disease. And that number is expected to increase to nearly 1.64 million within the next 20 years. And unfortunately, Parkinson's disease right now has no cure and is managed medically through pharmaceuticals, surgery, and of course, exercise. So community-based exercise programs are available for individuals with Parkinson's disease. And of course, there's many benefits for this. This includes promoting long-term adherence to the program, reducing the disease severity, and also it improves functional and quality outcomes for quality of life. So the purpose of this presentation overall is to describe the engagement of the DPT students at the University of Michigan-Flint. We offer face-to-face and online exercise classes for individuals with Parkinson's disease.
So moving on to our methods, our students and physical therapy faculty have been offering a weekly in-person exercise class for people with Parkinson's since the year of 2016. The class is structured consistently with the POWER or Parkinson's Wellness Recovery Principles and is offered free of charge. So training is provided by both the faculty and students with POWER experience, and the students volunteered to participate and lead portions of this exercise class. Parkinson's really impacts a person's ability to speak loud. Therefore, we emphasize talking loud when exercising. We sometimes focus on multitasking by counting aloud or making lists while walking, which is an emphasis of dual tasking. And also participants are at varying levels of function. So we'll always find ways to accommodate and modify exercises to fit their abilities. Next slide.
So what is POWER? POWER stands for Parkinson's Wellness Recovery. And this is an overall program that's run throughout the country. And the purpose of the POWER is to develop and implement worldwide access to the cutting edge Parkinson's disease specific neuroplasticity principled exercise programs. And they hold a promise to slow the disease progression, improve the symptoms, restore function, and increase the longevity and quality of life. So in POWER, there are 4 basic POWER moves, and they're each designed to target a specific skill that is known to deteriorate in individuals with Parkinson's disease. So the 4 moves that we're going to talk about and do today are the Power Up, the Power Rock, the Power Twist, and the Power Step. And all of these moves can be scaled depending on the level of ability or how hard we want to make the class. So they can be scaled to sitting, standing, on your stomach, or on all fours. Next slide, please. So the first exercise that we're going to do together, if you can click one more time. Yeah, so there's a video of me, and I'm sorry if you have to keep on pressing play. if you guys can't see it, I'm going to be doing the exercise with you too. So people with Parkinson's disease often have a really stooped, bent forward posture. That's okay. We don't need the video. We'll just do it together. So they have a bent forward posture. So it's really important to get them to open big. So if I can have everybody stand up and try to look at my screen. I'm sorry if I'm small. So we're going to be doing a little squat and putting our arms out in front of us. And then we're going to be bringing our hands back and standing up. So we're going to do 5 of those. So 1, 2, 3, 4, 5. Okay, great job, everybody. I saw you all in the little corner of my screen. The next exercise we're going to be doing is called a power rock. This one really focuses on weight shifting between the lower extremities. So you're going to be doing kind of a little lunge out to the side. You're going to focus more on my legs for this one. And we're going to bring one arm up, reach over, shift your weight over that leg, reach up high to the sky. Yep, and then to the other side. So there's 2. There's the 3rd one. 4th, and there's our 5th. Okay, and Anna's going to lead us through a couple more.
Perfect. So for the power twist, this one's a great way to implement weight shifting with emphasizing trunk rotation. So what we're going to do is stand up tall. We're going to open up our hands and we're going to work on trunk stretching and flexibility here. We're going to take our arms across and then hold all throughout, including or pointing your toes as well, and then moving your head towards that direction. We're going to open up like a book and then go to the other side. So we'll do 5 of those. So 1, 2, 3, 4, and 5. Great. And then moving on to the next one here. I'll go ahead and advance. Yep. So for the power step, this one's also a great way to open up the chest and the arms with an additive move from a step. So what we're going to do is, again, stand up hips width apart. We're going to take a step back and then open up kind of like in a diagonal direction. We're going to come back to center, and then we're just going to go to the other side. And the way I think of this is kind of serving like a plate to a table behind you if you were a waitress or a server. So we'll do 5 of those. We'll do 1, 2, 3, 4, and 5. Perfect. So those are all of the moves. Go ahead and advance the slide. So just kind of as a timeline, Power at U of M Flint, in March of 2020, we had weekly exercise classes that were stopped, unfortunately, secondary to the pandemic. And then in September of 2020, we had students and faculty from both our university and Central Michigan University with the foundation beginning to offer free Saturday classes on Zoom. And then not too long ago, in September 2021, we had weekly in-person classes resume at the campus, while online Saturday classes were continued to be supported by our students and faculty. So for the online classes, there is a spreadsheet that is utilized for faculty and students to keep track of the number of the participants that attend and which university is teaching for that Saturday class. It also keeps track of which body position is focused for that session, whether it be lying on the back, all fours, side lying, or on their stomach. Next slide. So for our results, on average, 6 people with Parkinson's attended the Friday in-person class with 4 to 6 DPT students and volunteers. And then on average for our online class, we had 4 DPT students complete the activities with roughly around 30 participants with Parkinson's. So on the right here for this image, this is what a normal power class will look like in person. We'll have chairs set up if the participants get tired, and it's also a great use of exercise modifications utilizing those chairs. There's a usage of mats, encouraging the participants to get on all fours or lying on their back or stomach to promote flexibility in movements. So we often see that older individuals will oftentimes have difficulty with getting down on the floor or being positioned onto their stomach. This is really significant because grandparents will want to get down on the floor to play with their grandkids, and this is just a great way to emphasize those gross motor movements. Boom whackers, mini hurdles, agility ladders, obstacle courses, and other tools are also utilized during the in-person classes to create more of a challenging and fun session. Next slide. And then for this one, you can kind of like advance for each quote here. So DPT students who engaged in this activity have reported that it has really allowed others to be exposed to a population that they may or may not have worked with before. Also, positive feedback from the individuals with PD has created a very strong sense of service by volunteering in a setting where everyone can participate. It really reaffirms the belief that exercise is medicine and that this experience will further shape us as expert clinicians. Other student quotes, people really like hanging out with the other people. Basically, it's fun. It's a really great way to decompress from schoolwork without totally disassociating from their PT identity. It's very uplifting, energetic, and rewarding as well. Next slide. So for our conclusions, we see that students who engage in leading an exercise class, either in person or online, have the opportunity to develop the knowledge, the skills, and the attitudes to continue this once working as a PT. So people with Parkinson's benefit from community-based exercise classes, and students can really assist in providing those free classes. So again, POWER integrates principles of neuroplasticity with the recent research on Parkinson's. And not only does POWER focus on the physical aspects of the disease, it also emphasizes empowerment. POWER empowers its participants to lead active and very fulfilling lives. Next slide. These are some of our key references here. And then going on to our acknowledgements next. We would like to acknowledge the faculty and student physical therapists at the University of Michigan-Flint. And finally, thank you to the individuals with Parkinson's that do participate in POWER. And now we'd like to open up the floor to any questions. That was our presentation.
I have a question, if that's okay. So both, I guess this could be both for Anna and Emma. Which mode do you guys prefer, face-to-face or the virtual for POWER?
I know that I personally really like going face-to-face. You get to have a little bit more of a connection with like the patients that we're helping. But unfortunately, like scheduling wise, I can only do online this semester, which is a little bit sad, but I still love to participate online too.
Same with me. I do prefer the in-person a lot more. It gives you more of that hands-on, like intimate connection with the patient, and you're learning a lot more skills in person, I feel like. However, I feel like others are, it's the convenience of joining Zoom. More participants can join. It's better, it's a little easier to facilitate in terms of like being online, but yeah, definitely in person for sure. So
I have two questions. So compared to your participation numbers before the pandemic, do you find that you have more people participating now that you offer the Zoom option? And my second question, do a lot of your participants, are they primarily early stage Parkinson's, or do you have any that are mid stage, any with advanced?
sure. So I'd say our online is obviously very much more hefty in our participants. I'd say I wasn't participating before the pandemic, so I'm not sure how that was. I think more people are joining now just because the convenience of Zoom, definitely. And then what was your second question?
Are your participants primarily early stage Parkinson's disease, or do you have any that are mid stage or even advanced PD?
I would say for our in-person ones, they're a little bit more high functioning. They're still able to, you know, some of them still drive, but then also there's like mid-level, like more moderate functioning where they need a little bit more help. They have their caregivers or their spouse come in and assist them with the exercises. Similar to Zoom, I feel like a lot of them have assistance with like technology, trying to log them into Zoom to like participate. There's definitely a mix. I would say it's more high to like mid versus the more severe stages. But it kind of varies as well in the class for like the online portion. We have like our regulars for the online, and then we have our regulars for the in-person usually. So it's kind of a mix.
I have a question, Anna. First, how many times the patients are expected to do those exercises? Because I think the community exercise is only once a week, right?
Yeah, from my understanding, POWER classes are offered every day of the week. It's just U of M themselves. We offer it twice a week. But there is other Parkinson's classes that they can join online every day of the week. Also it's all completely voluntary, right? And it all really depends on how they're doing that day, on whether or not they feel up to exercising. So sometimes we won't see an individual for a while, especially if they're recovering from a fall. But it's all completely up to them. Most of the time, we get patients that just come the once a week, and they also do physical therapy on the side.
Thank you. Do you see any benefit of patients doing these exercises, like after attending maybe a few, to learn how to do those and then to actually practice them at home by themselves so that they don't have to commute? Like if that's the case, like I don't know if you have any system already that will allow monitoring the kind of exercises that the patients are doing at home, by themselves.
From my understanding, it's a lot more of self-monitoring and physician monitoring too of their exercises. But with them doing this program, I definitely have seen some improvements, especially in the individuals who come for the first time, and then the first 2 or 3 weeks they really get going with their exercise programs. I've definitely seen those symptoms start to kind of diminish a little bit more. It's also really dependent too on their medications and what sort of period they're in. If they're on their off time or on time, that really does affect exercise too. Hopefully that helps.
Thank you.
I believe there's also a Power YouTube channel where you can find supplemental resources to do different exercises. I believe they upload their Zoom sessions from other other locations that do Power as well, and then you can go back to those recordings and kind of follow along for previous exercise sessions as well. I know Dr. York probably emails some of these participants just additional resources or exercises that they're interested in, just to help with their certain issue that they're going through right now. So
yeah, particularly for exercise, it is very important that the patients will feel motivated, and to be motivated, oftentimes they want to see the benefits, or for example, the diminishing of some kind of symptoms. So I'm actually thinking, well, sorry, because I'm with the other research team, so I keep thinking about how our mobile app can be used to also serve, to serve different purposes. For example, how can it be used to motivate patients to continue to practice those exercises that they've learned at home, so that they will be able to do that more often. So, yeah. I'm actually hope that we can, do some kind of on-site observations and maybe interviews with the patients as well, if possible. But of course, with IRB approval, we don't. But yeah, that's something maybe we should talk to Dr. York.
Thank you. I definitely think she would be open to that. Yeah.
Yeah, no, I think that's a great idea, Charlotte, but just for the sake of time, we should start on this last group, and then hopefully we'll have a little bit of time to kind of answer general questions and stuff like that at the end too. But we'll go ahead and move on to the final group of the session today, and give me 1 second. So Matthew, Kayla, Zoe, Delaney, Abby, Quinn, Taylor, Linda, Charlotte, and myself will be talking about designing a mobile healthcare application for Parkinson's disease from the perspective of the healthcare professional.
All right, so you can go to the next slide. Hello, everyone. So as we talked about previously, Parkinson's disease is a progressive neurodegenerative disorder, and its symptoms can range from person to person, and this is between motor and non-motor symptoms. And this includes changes in cognition, mental health, sleep movement patterns, and people with Parkinson's disease struggle to track their symptoms, which can make it difficult for healthcare providers to accurately monitor the course of treatment and disease progression. Next slide, please.
So people with Parkinson's disease struggle to track their symptoms, which can make it difficult for healthcare professionals to accurately monitor the course of treatment and disease progression, as well as to determine the effectiveness of medication, especially medications like carbidopa, levodopa, which are very time sensitive. So we wanted to understand what features were important to healthcare professionals from a usability standpoint when designing a healthcare application to help monitor their patients with Parkinson's disease. Next slide. Our design goal is to focus on a user-centered experience that would complement existing therapies and that can be easily implemented into the clinical setting. The participants expressed a desire to monitor their patients' experience in between visits, with an emphasis on empowering them with the tools to also self-manage symptoms in between visits. Next slide.
Okay, so we had 5 participants. We have had some difficulty recruiting. We recruited through email and through word of mouth in the US. We used semi-structured interviews with healthcare professionals who work with people with Parkinson's disease, just asking open-ended questions regarding experiences working with people with Parkinson's disease. We asked about interactions, their coordination of care, and their use of technology. Our interviews were conducted and recorded through Zoom. All of our interviews were transcribed by 2 researchers. Next slide. So we used the affinity diagramming method for data analysis. So we went through the transcriptions. We pulled out some of the more important and some of the things that just stood out. We pulled those out of the transcriptions and we put them into categories so that we could compare what the healthcare providers said.
This brings us to the findings of our research. And first we have the healthcare provider's perspective on Parkinson's disease. And first we know the symptoms are ranged into non-motor and motor symptoms. The non-motor symptoms include things like depression, loss of cognition, anxiety, sleep disorders or problem sleeping, blood pressure problems, and even urinary issues. Non-motor symptoms include things like slower walking speed, worsened gait, and tremors. And one of the things that is found is that each person with Parkinson's disease will display a unique range of these symptoms. And so this presents challenges because similar symptoms will be hard to differentiate for people with Parkinson's disease. Also, recognizing when therapy is needed is difficult because, for one, some forms of therapy are not very well recognized by people with Parkinson's disease as something that would benefit them. And a good example of this is speech therapy. Another issue is when a person with Parkinson's disease develops new symptoms, it can be hard to know when therapy intervention is needed. And also, these symptoms will affect people differently depending on their lifestyle. So people who are more active may experience a much larger loss of freedom and thus be more affected by their symptoms than people who live more sedentary lifestyles. Next slide, please. So now we have the benefits of an app to healthcare providers. First is the use of electronic medical records, which provide organized and accessible information to healthcare providers. And if an app could be linked to these electronic medical records, it would allow healthcare providers to better understand actively what problems a person with Parkinson's disease is facing and how to treat that. Now, tracking is great because it can slow progression by catching symptoms before they get worse. It can also help to identify what needs to be done to help a patient. And by this, I mean if an app could help design a treatment plan by recognizing when there's a need for therapy or additional medication, because the app can sense when symptoms are getting worse and what to do about that. It can also tell what the patient could be doing and what they are not doing, such as exercising. Are they getting good night's sleep? Are they eating well? And what needs to be done about this? It was also noted that caregivers having access to this tracking would help increase the reliability of the data, because sometimes the caregivers will notice symptoms and regression that a person with Parkinson's disease will not notice themselves. Next slide, please. Also, the app could function as an educational resource because educational guidance has been noted to be lacking for people with Parkinson's disease. This is important because better educating people with Parkinson's disease and their families on the disease will help to better improve the treatment. And a good example of this, one particular fact, is that many people with Parkinson's disease may not know that consuming protein with some certain medications will lower the effectiveness of the medication in actually treating their symptoms. And also the app could function as a platform for self-advocacy, and it could also function as a social platform to better connect the Parkinson's disease community and give them a way to advocate for the things they need as their needs evolve. Next slide, please. So when using an app, compliance is actually one of the largest issues that an app faces. And this could happen for different reasons, especially for an app designed for people with Parkinson's disease, because symptoms from the disease itself make it difficult. And an example, this is tremors. If you are trying to type on a phone, having tremors is going to make that very difficult to track your symptoms because tremors make it difficult to type. And also many people with Parkinson's disease that are in advanced stages may also suffer from old age-related illnesses that make it difficult because of problems with cognition. And an example, this is dementia. It would be impossible for a person with dementia to operate an app on their own. A caregiver would need to do that for them. And then on the matter of payment, participants seem to be more likely to use an app if it is free. However, if there's a cost to the app, they are more motivated to use it. Next slide.
So this brings us into recommendations for an app. We found that healthcare providers, what they wanted in an app fell into a few different categories, the main one being patient education. So things like providing resources and recommendations for patients, like we talked about earlier, addressing misconceptions around speech language therapists can encourage people with Parkinson's to seek help for speech therapy. Also being able to show how to differentiate between Parkinson's symptoms and things like aging, and establishing routines so that patients can help themselves in between sessions. They also recognize a need for reimbursement. So a lot of times, doctors, they don't have that long of appointment times with their patients. So being able to be reimbursed for the time spent reviewing the app would be very helpful. And then also having a way to represent the data. So using graphs and either scatter plots or bar graphs to be able to easily see the data, and then condensing the data into snapshots to also accommodate for these small appointment times. And then also recommendations for monitoring symptoms, things such as passive tracking. Basically, the less that people with Parkinson's have to do, the better. So things like tracking motor symptoms with sensors in the phone, also being able to track how patients move in diverse environments, monitoring exercise, being able to self-manage symptoms between sessions, and having patient diaries that can help to reveal patterns that weren't evident before. Slide. So in the future, we want to continue to collect and analyze data on what features would be most useful. We're hoping to collect from healthcare professionals in underserved areas and across different disciplines. And ultimately, we want to actually develop an app and deploy that into the field to continue collecting usability research on the application. Next slide. So this is our research team that was working on this project. We'd like to acknowledge U of M M³ funding and the U of M Flint UROP and GSRA programs. So this concludes our presentation. Thank you so much for listening. We'll now open the floor up for any questions you guys may have.
Well, great work, guys. Does anybody have any questions? Any questions at all? And also, this would be a good time in the last couple minutes that we have if anybody has any more broader questions. So unfortunately, Dr. Tang had to leave, and she thanked everybody for their time and also said everybody was doing an excellent job. And so I think to the Dr. York team, that's a conversation that maybe we should have some time, or at least reaching out to Dr. York and seeing about the possibility of maybe getting some of the POWER participants' perspectives on if they'd want to participate in a study and see if there might be a nice interface between the POWER program and the app. But any other kind of bigger picture questions? It was nice in this session that there was kind of a unifying theme around Parkinson's disease or other neurological disorders. So. Awesome. Well, it doesn't sound like we have any questions, and it looks like we're at time. So I achieved my goal of actually getting things in time, which, if you know me at all, is a major goal because that never happens. But thank you everybody for your presentations. Really, really great job today. It was great hearing all this, and continue working on this great and amazing research that you guys are doing. So awesome.
So, hello everyone. My name is Linda Zhu. I'm the moderator for this breakout room. Let's welcome our first presentation, assessing the ecology of the Flint River in Flint, Michigan, above and below a central old dam. Will be presented by Chloe Summers, Adrian Elkins, Carson Konzer, and Dr. Hazen Dawson.
All right. Yeah. So like I said, I'm Chloe Summers in Dr. Dawson's lab.
My name is Ariana Elkins. I'm an undergraduate in wildlife biology.
I'm Kason Konzer. I've been working with Dr. Dawson through the URIDGE internship. So it's here at U of M-Flint.
And today we will be presenting the data we've collected on the assessment of the ecology of the Flint River.
Just let me know when you want me to switch a slide after this one.
All right. So what we have here is a depiction of the Flint River watershed in the map of Michigan and all of the tributaries on the right. And then you can see where Michigan is in the US on the left. And Flint is home to 81,252 residents, and the river drains portions into 7 different counties in mid-Michigan, with the watershed being more than 1,300 square miles. Go ahead. So we have 2 objectives. The first is to investigate the ecology and health of the Flint River above and below the Hamilton Dam before the rest of the dam is removed, hopefully in 2023. And then the second is to collect data now and then into the future in order to gain an understanding of baseline ecosystem data to gauge if restoration is a success. Go ahead. So a little bit of background. The Hamilton Dam was constructed in 1920 and resides in the urban city of Flint, Michigan. It was previously used for logging in the automobile industry, but has been deemed dangerous. So it no longer serves this purpose and is currently in the process of removal to revert the stream to its natural ecology. The upstream reaches consist of a wide riparian zone, whereas the downstream is composed of cemented structures that trap debris. You can see some of it, and then other parts we pull up in cast nets during our field season. And then the Great Lakes have notoriously been polluted through agriculture, urbanization, and industrialization that introduced chemicals such as PFAS, PCBs, and heavy metals, including mercury, arsenic, chromium, lead, and cadmium. And these have been found in higher concentrations in the reservoirs above the dam, as there's sedimentation that builds up right above the dam and then collects all of these in higher concentrations that are then later bioaccumulated through the food trove. Go ahead. So an introduction to the study, it's being done on the Flint River before the weir is removed in 2023. This is being done through a $40 million project in order to restore the habitat and provide better recreational utility. This data will later be compared to data collected after the weir is removed in order to assess the impact of habitat fragmentation and then later relay how restoration efforts can affect the riverine ecosystems. We've employed multiple fishing methods that were necessary above versus below as it's a varying habitat. So some methods couldn't be done above, whereas some couldn't be done below. Continue.
Okay, we'll be switching over to Ari now.
Thank you. So the Hamilton Dam and the Flint River flow right through the U of M-Flint campus. We have the opportunity of fishing 4 different sites on the river, which is depicted in the slides. And we have a permit, which this is the next slide now. We have a permit that allows us to catch fish using cast nets, which is the top left picture, gill nets, which is some of the processes seen in the top right picture, and then hoop traps and electrofishing, which are the bottom two pictures. And then of course, hook and line methods. And you can go to the next slide, please. Once fish are obtained, we identify and record the species. We measure the length in centimeters and the weight in kilograms. If fish are above a certain size, we tag them. This means we insert an identification tag into the fish with a number that can identify them if they are caught again. We use a Floyd tag gun, which is not pictured, but we also use a homemade method that Chloe created, which is the top right picture of the slide. I'm sorry, the flow tag is in the bottom left with the pike. And then that carp in the bottom right was caught using a hook and line. You can go to the next slide. So we release fish back into the river unless they are preserved for the heavy metal testing that Chloe mentioned earlier, or they're considered an invasive species in Michigan, which would be the round goby, which is in the top right picture, white perch, which is not pictured, and then also the common goldfish, which is in the bottom picture, in which these invasive species are humanely euthanized. And now to Kason for a little bit of our results.
Okay, so I'll briefly skip back to the site map. I meant to change the labels, but sites 1 and 3 are below the dam and 2 and 4 are above the dam. So in this next graphic, we're looking at the catches per species at these various locations above and below the dam. So site 1 and 3, we're below the dam. We see a wider, what we call species richness, which means we have a higher unique count of species compared to above the dam. Similarly, we have a difference in catch rates below the dam than above the dam, and this turns into a lower quote unquote diversity. With regards to the Simpson's diversity index, that's usually measuring the spread or the evenness of the distribution of species. So we actually got flipped results of what we expected, and that's because with some of the methods we used, like a cast net, we might throw out a cast net and get a large catch of gizzard shad in proportion to what you might be catching on a hook and line. So we have a large dominance of gizzard shad here on this bubble. The size is actually the number of catches with a log weighting for scale. So we also then broke this up into different feeding tropes. So we have on the left piscivores, in the middle planktivores, and on the right benthivores to further divide and compare our species across. So one thing that we might notice here is that the planktivore rate we see is much higher low stream, or at least the richness. So on to the next slide. This is some analysis of our heavy metal testing. So in this, we're looking specifically at mercury, and we're looking at the difference downstream versus upstream. So the values depicted in our heat map are going to be the mercury in milligrams per kilogram dry weight of each fish that we caught downstream minus the respective fish upstream. So a negative value will correlate to a higher mercury per kilogram rate upstream, which we see across the diagonal here. This is on the pairwise comparison of the same species upstream and downstream. And the only difference we see where we have a higher heavy metal toxin rate downstream is with bluegill. And a possible reasoning for that is to do with the catch size, catching smaller sized bluegill upstream versus downstream. And a simple note on this is that this is a small sample size due to costs incurred with testing. So I'll move this on now to the conclusion.
So in conclusion, the ecology study of the Flint River has been conducted for 2 years, taking a break in 2020 as the pandemic interrupted the process. And it will continue through the removal and restoration of the Flint River in hopes to see the pre-assessment compared to the post-assessment to gauge whether the restoration was a success. We're excited to see how it turns out.
Many people to thank for funding, Community Foundation of Greater Flint, Saginaw Bay Watershed Initiative Network, U of M Flint's multiple organizations such as the Office of Research, College of Arts and Sciences, Opportunity Fund, Urban Institute for Racial, Economic, and Environmental Justice, the students here, as well as the scholarships applied, such as the Gary and Colleen Pace Field Biology Scholarship. We also want to thank specifically Megan Heise and the Flint River Watershed Coalition. And we have our references at the end here. So at this point is I should click again, right, and have a nice big question slide, but I'll leave that to you guys. If you have anything to ask, we'd love to answer.
I had a quick question. So could you define the feeding tropes that you went over, like planktivore?
So I am a data guy. So I did the data backend for this team. To my knowledge, piscivore is more like a carnivore. So these are meat eaters. Planktivore.
Other fish.
Yeah. Okay. I'll let these specialists take care of this question for you.
So the piscivores are fish eating fish. The planktivores are fish that eat like plankton, smaller beings within the water column. And then a benthivore is typically eating the benthic macroinvertebrates, or they might be considered omnivores because they'll pick on some algae too. What's next?
Yeah, if anyone wants to reference a specific slide, I can go to it. Otherwise, I will close down this share.
I actually have a quick question for the slides you're showing right now. What is the sample size? So, I mean, how many fishes have you tested on to receive this result?
Right, so this is small sample size. Only the largemouth bass had a duplicate. Everything else is a single caught fish.
I see, thank you. But still a lot of work have done. Good job.
We're actually, we have more samples sent out and intend to do more in the future, but it is like about $300 per fish. So yeah, just hoping to get more funding. If there's any U of M Flint students that want to ask if there's opportunities to come fish, there are, and you're more than welcome to join. Right now we're fishing every Tuesday from 6 p.m. to 8 p.m. and we'll continue through the summer. So if you're interested.
All right, great. So any other questions for our first presentation?
It's so anticlimactic. We can't all like applaud. Nice job. Like the little emoji just doesn't seem to do. I don't know. Zoom conferences are weird. Great job, everybody. That was really neat, and I like your pretty figures as well.
Thank you.
Thank you.
Yeah, can we just all unmute and clap, right? We can, can't we?
Yay. Nice job. Better. Good job. We're making a breakout room choice. I like it.
All right, I guess we can move to our next presentation. Our next presentation is Calon alternatives be dare resistant? Question by Trent Adam and Dr. Rebecca Tonioto.
Hi, everybody. So I'll share my screen real quick and get that presentation up. Can everybody see that all right?
Yes, you're coming through.
Okay, awesome. Thank you. So yeah, I'm Trent Adams. I'm an undergraduate at U of M Flint studying wildlife biology. And I'm asking this question with my advisor, Dr. Rebecca Tonietto. So yeah, I'm asking, can lawn alternatives be deer resistant? So first, I'll go into the background of why we're asking this question, and then I'll get into the question itself and some of our hypotheses. And finally, I'll end with the methods and benefits of this potential future study we'll do over the summer. So first, I wanted to talk about what a lawn alternative is. So to talk about why we're making lawn alternatives, first we should talk about some downsides to our normal turf grass lawns. So anybody who has a lawn knows that you have to mow it once or twice a week, which causes carbon emissions. Typically they need to be watered a lot, and water might become a more valuable resource as our climate changes in the future. And also, when you apply a lot of herbicide to it, can damage wildlife such as like pollinators or amphibians that are in your yard, and it can also contribute to polluting the water table as well. Same with fertilizers too. If you use those on your yard, they can contribute to pollution as well. So lawn alternatives are kind of being developed in so they'll be made out of native and low growing plants and forbs, which are flowering plants. And these wouldn't require as much maintenance, and they would also provide support for pollinators and other wildlife such as migratory songbirds and small mammals as well. So over here in figure 1, I just have an image of non-natives versus natives and their root sizes. So the fescue turf, you can see that would be our normal lawn, and the roots, you can barely see that they go down at all. But with the buffalo grass and some of the other natives, you can see that the roots, they actually go down to about like 7 to 8 feet. So they can hold the soil there and prevent erosion, and they can also act as flood control, and they won't have to be maintained as much because they can access the water at a deeper water table. And yeah, so these lawn alternatives that are being developed, they can help the University of Michigan meet carbon neutrality goals as well. I'll get into it more later, but there might be some research done on campus. So just to go into the background of this lawn alternative project, so these lawn alternatives were originally tested and planted in Chicago in the summer of 21. That was done with the Chicago Parks District and the Chicago Botanical Garden. And Dr. Tonietto also went there to collect research. Over here in figure 2, we just have an image of 2 different treatments that they were testing in Chicago. So the first one, it grows about 6 inches, and the second one, the Oak Path Meadow, grows about 8 inches, and they have different flowers and different plants that they were testing out. And then figure 3 is just an aerial view of the experiment that they did in the park in Marquette Park in Chicago. And each different rectangle was a different treatment that they were testing made of different plants. And something that we found from the data that Dr. Tonietto helped collect was that pollinators such as bees, they actually prefer native plants over the introduced plants. So that's one benefit of these lawn alternatives. So we're looking to implement lawn alternatives here in Michigan. We know that this study was done in Chicago. And one question that we had was if deer herbivory would, or like Michigan has a high deer population. So we want to know if any plants would be deer resistant. I also did some background research, and I found that most of the flowers used in Chicago and other plants were native to Michigan as well, and so that they were potential ones that we could use in our Michigan lawn alternative design. So that takes me to our actual research question now. So we wanted to look and see if large-leafed aster, silky aster, and purple prairie clover can resist white-tailed deer herbivory. The reason why we chose these 3 plants is because we know that they would provide a lot of pollinator support, and they're low growing, so they'd be good for lawn alternative projects. And when I was doing the background research, I found that like some sources were saying that these plants were deer resistant, and some sources were saying that they weren't deer resistant. So that kind of made us curious, and we wanted to test to see if they actually were or not, because they would be great additions to a lawn alternative design. My hypothesis is that all three of these species might be preyed upon by white-tailed deer, just having a garden and knowing how much deer like to eat. But we're hoping that maybe some of them, if not all of them, would be deer resistant, and we could actually use them in these lawn alternative projects. As our controls, we'll be using strawberries, which we know the deer will eat, and milkweed, which we know the deer won't eat. And here in table 1, I just have like a simple table showing kind of like spatially how we might design the project. So we'll have the strawberry control on one end of the plot, and then the milkweed control on the other end. And then in the middle, we'll have all three of the flowering plants that we're testing. And then some of that will be enclosed and some of that will be outside of an enclosure. So inside the enclosure, the deer won't be able to get in there and prey upon the plants. The second question we're asking with this research, we're asking, does age of the plant affect its chances of being preyed upon? So some research has shown that at different ages, plants tend to be preyed upon more. So for my hypothesis, I was thinking that maybe mature plants will be eaten the most because they might be more nutritious, and that seeds might be preferred the least by deer. So for our future project, and now I'm kind of getting into the methods of what we might try to do this summer. So we'll use deer exclosures as a control to keep deer and other herbivores out. We'll use camera traps to figure out what type of species are preying upon the plants if they are preyed upon. And we'll be surveying plants for herbivory damage at different life stages. So we will be placing 4 replicates across southeastern Michigan. And we'll also be using a germination chamber mainly to get our plants to different life stages. So we're hoping that we can buy seeds as well as mature, already potted plants. And what we'll do with this germination chamber is we'll put seeds in the germination chamber, which is kind of like a big fridge, and you can control the light and humidity in there and the temperature as well. And hopefully we can get these seeds into small plants, and that can be like our 3rd level of life stage of the plant. So kind of getting more into the methods and summing it up. So we'll set up plants inside of a deer exclosure as a control. That way no herbivores get in. And then we'll have plants outside as well to see if that are vulnerable to herbivory. And we'll also record damage to the plants and record the specific location of the damage as well. So we'll record if the leaves are being eaten, or maybe they're eating the stems, or maybe they're just eating the flowers. So we'll make a note of that. We'll also use the trail cams to show what species are eating the plants if they are being eaten, because maybe these plants are deer resistant, but maybe rabbits are still eating them or raccoons are still eating them. And if that's the case, then it might not be a good idea to use them for lawn alternatives. We'll also be looking at the plant life stages. So yeah, we'll have the 3 different levels. We'll have seeds, we'll have the young plants, which will grow in the germination chamber, and we'll also have the mature plants, which are flowering. And then we'll just observe predation rates at each stage. So for the future outcomes, so I hope that we can conduct this research over the summer, and then I would do the data analysis in the fall, and then hopefully I could present at the Society for Ecological Restoration Midwest Great Lakes chapter and the Natural Areas Conference in 2023. So just to talk about the broader context of lawn alternatives and why this type of study might be beneficial. So all the data that we would collect on deer herbivory would be shared with the Chicago Botanical Garden and Chicago Park District. And so when they develop future lawn alternatives, they might be able to use this data, and if they want to have plants that are deer resistant. And also Dr. Tony Edo will be working with the University of Michigan-Flint this summer to do a replicate of the Chicago study on U of M Flint's campus. And if U of M Flint does decide that they want to have lawn alternatives, it might be beneficial to see what plants are resist herbivory in case there's rabbits or they see other type of herbivory damage on campus. So just going into the benefits of the research a little bit more. So yeah, it can help the university. These lawn alternatives can help the university meet their carbon neutrality goals. It would also benefit rural landowners that do want to implement these lawn alternatives because if in rural areas you tend to get more deer, and it is an investment to put these lawn alternatives in, and people wouldn't want to seed their lawn and have them be eaten right away by herbivores such as deer. And also this could benefit urban areas that have a lot of deer as well. So some cities have issues with deer in there, and if they want to have urban gardens, they might want to have deer resistant plants that they have as part of their lawn alternatives. So here's my acknowledgement slide. I'd like to thank the University of Michigan-Flint College of Arts and Sciences, where I go to school, the Chicago Botanical Garden and the Chicago Park District for doing the initial lawn alternative project over the summer, and the Undergraduate Research Opportunity Program here at U of M Flint, which I'm employed through and I'm able to do this research through. Thank you for listening to my presentation, everyone. I'm really excited. Hopefully we can get things going this summer, and I'd love to take any questions that anyone has.
Thank you, Trent. Very exciting project, and I'm looking forward to see the results. So let's give Trent. And any questions?
Lisa.
I have a question, Trent.
Yeah.
You mentioned like you know, that there'll be seeds and then there'll be mature plants, and the young plants might be in a germination chamber. Are you gonna like test the young plants out there too? Like, because I don't know a lot about deer herbivory, but I thought they liked, you know, kind of yummy little sprouts and bud kind of things.
Yeah, we would definitely be testing the young plants as well. So yeah, maybe we'll see that they prefer to eat those young sprouts. Yeah, I'm not sure, but that'd be cool to test that out and see what happens.
Perfect. Thank you. Thank you.
I had a question.
Yeah.
Do you think there's people that would want lawn alternative options that would attract deer?
Yes, I actually, so when I went to Bourdines just seeing what plants they had, and I was actually talking to a lady there, and she was like, well, she was saying that she wants to attract deer to her yard. And so that's something I've kind of been thinking about. And what I would say is that, like, if you wanted to attract deer, you could have a lawn alternative that still is deer resistant, so you know that they won't eat like your lawn. But you could also devote part of your lawn to a deer garden where you plant strawberries or other plants that would attract deer. Or you could just let another part of your lawn just grow crazy and try to go back to like a prairie type system, and that would also attract deer, I would assume.
You might have already mentioned this, but where exactly is this taking place?
We'll do 4 different sites across southeastern Michigan, and it'll mainly take place in people's lawns. So like family members' lawns, or Dr. Tonieto might let us use her yard type thing. So, but we also might try to put one at a park, maybe 4 more or something. But we're still planning all that out. So
It's cool. You should take pictures.
definitely, yeah. Hopefully, if we set up the trail cams, we could get some really cool pictures. Well, thank you, everyone. If no one has any other questions, I'll stop my share and we can keep going on. Thank you.
Thank you, Trent. Yeah, any other questions? All right, so if there's no more questions, let's move on to our next presentation. Our next presentation is a spike analysis in the auditory brain structure. Will be presented by Alex Meeker. The work done by Alex Meeker, Shelby McBridge, Vajani Mudana, and myself, Linda Zhu, is a collaboration with of UM Flint Engineering and the School of Medicine at Wayne State University. Alex, do you want to share the PowerPoint by yourself?
Yeah, I can do that.
Go ahead.
Okay, 1 second. Okay. Can everyone see that?
Yes.
There we go.
Okay, good morning, everyone. I'm Alexis Meeker. I am a molecular biology student here at the University of Michigan-Flint, and I'm presenting about spike analysis in the auditory brain structure. So here's our overall abstract for the project, but in summary, tinnitus is the ringing sound in the ears without external stimulus. The condition affects about 15 to 20% of the United States adult population. And unfortunately, there's little understanding of the neural pathways involved, which makes it difficult to discover diagnostic tools or treatments. However, we believe hyperneural activity spikes could be related. So analysis of those spikes could find patterns of hyperneural activity in the three brain regions at 4 different conditions, which are before and after noise exposure, as well as before and after auditory cortex electrical or ACE stimulation, which is the tested treatment. An algorithm was developed to identify the neural spikes and specific related patterns. So here on this slide shows the overall brain regions that we're studying, which are the left dorsal cochlear nucleus, or DCN, the left inferior colliculus, or IC, and the right auditory cortex, or AC. So the figure shows how these structures are located in relation to each other. Many electrodes are placed in each region and break down to 32 in the DCN, 32 in the IC, and 16 in the AC. So the first thing we did was to find the timing of the spikes along with the local field potential signals. We have developed an algorithm to evaluate the amplitude and location of the peaks and label them as spikes. So the blue lines are what's showing the local field potentials, which are the electrical potential around neurons. And they are the raw data from the direct measurements. The red triangles mark the spikes with the highest peak from a grouping within a small range. An example of the marked local field potentials is what's shown on the slide. Please note that this is only one example of one animal under one condition and one of the 80 channels. So when we did the analysis, spike locations and amplitudes may vary across channels and across conditions. So we studied 3 specific patterns, which are the firing rate, which counted the number of spikes over the course of 5 minutes, the average spike peak, which averaged the amplitude of all the spikes in those 5 minutes, and the average duration, which is the duration of the average spike, which is measured in seconds. The graph on the slide is an example that we tested, so it can vary across animals. And again, the amplitude is the distance from the peak to 0, and the duration is the distance between the 2 valleys. So we have data for 15 different animals, and we're still processing it. And so the data throughout this presentation is some of the preliminary results we have for one specific animal, and we're using rats under 2 of the 4 overall conditions. So the graph on the right compares the firing rate of one rat before and after noise exposure. The firing rate is the amount of stimulation each channel received along the auditory pathway. The red line, which is showing after noise exposure, has a higher firing rate compared to the blue line, which is before noise exposure. The after noise exposure shows a higher firing rate because that is when the animal has developed tinnitus. So the neurons are picking up additional stimulation, which increases the firing rate. Some channels were removed from the data as their peaks were abnormal in comparison to the rest. The abnormality of these points could be a result of poor electrode connection or extraneous background noise. So this graph compares the average spike peak of the same rat as previous. The large jumps in the average are a result of changing from one brain region to the next. The after noise exposure numbers are higher in the DNC and IC regions than before exposure because there's more stimulation once the animal had developed tinnitus. Meanwhile, we didn't notice much change in the AC area for this animal. So finally, this graph compares the duration of the spikes in the same rat as the previous two patterns. The after noise exposure graph is much more erratic than before noise exposure because of the increased stimulation from the tinnitus. And that can stimulate neurons for much longer or prevent stimulation from true external noise. So that was the last slide, but I forgot I had a conclusion slide to it. So we have developed the algorithms and receive preliminary results as I presented. In future work, we plan to process the data for more animals because this was only 1 under 2 patterns and we have 15 under 4 overall patterns for a better understanding of the spikes and how that relates to tinnitus's hyperneural activities. Additionally, we want to see if the ACE treatment stimulation actually works. And so with that concludes my presentation. Are there any questions at all?
Thank you, Alexis. So is there any questions?
What are the
my question? Yay! Nice job.
what are the plans for additional tests, Alexis?
We were just we want to, because these are just preliminary results, we want to like further break down like what our results mean and just what they mean and see if there's any developing patterns from these hyperactivity spikes.
Okay, no, that's cool. That's really, really neat. You show an obvious difference. It's very cool.
Other questions?
I have a question. What made you ask your research question?
I'm actually totally new to this project, so I joined it because I'm just interested in all the bioengineering that's going on in this. So mainly, but overall, like tinnitus, there's poor treatment options, poor diagnostic tools. So we really just want to help people who suffer with this condition because it does affect like 1/5 of the population. So since there's like a big hole in treatment and everything, we want to help try to improve the quality of life of people who have this condition.
Thank you.
Other questions? If not, thank you, Alexis. Let's move on to our last presentation. Our last presentation is the information flow in the auditory brain structures. Will be presented by Shelby McBridge, and also the work is done by Shelby McBridge, Rajani Madonna, Evan Flanagan, and myself, and also is still collaboration with the School of Medicine at Wednesday University. And I'll go ahead and share the PowerPoint. Okay, Shelby, do you want to start?
Hi, can everyone hear me okay?
Yes.
Awesome. Hello, everyone. Thank you for joining us. It's been really great seeing everybody's work so far today. I'm Shelby McBride. I am also a molecular biology student and biotechnology student here at Flint. I started this project last semester, and so I will be reiterating a lot of the things that Alexis just went over. She just described much of the work that we are doing this semester. However, today I'm going to be discussing our research on the information flows in the auditory brain structures from last semester. and presented by myself, Shelby McBride, with Rajani Madula and Evan Flanagan, and then Dr. Linda Zhu is our research lead and principal investigator, as well as the moderator for this breakout session. This research has been performed in collaboration with Dr. Luo and Dr. Zhang of Wayne State University. And next slide, please. So to begin, I'm going to first identify the goals and objectives of our research. This is detailing what we're doing and why. Our goal is to calculate the response time to process information in order to identify the similarities and differences of information flowing between specific auditory brain structures in animals both with and without tinnitus. So this is going to help us further our understanding of the information flow through the auditory structures in the brain, and also help us make progress in testing and treatment of tinnitus. Next slide, please. So what exactly is tinnitus? Tinnitus is a ringing or buzzing sound in the head without any external source of sound. It's common in people above 55 years of age, with a prevalence of 33%, and it's estimated to affect 10 to 15% of the adult population. People with tinnitus often experience difficulties in sleeping, and it may also cause irritability, anxiety, and depression. It can be induced in different ways. However, noise-induced tinnitus is thought to be the result of increased bursting events, hypersynchrony, which is increased neural synchrony, and tonotopic map reorganization along the auditory pathways. So next slide, please. So to give you a little bit of background, here we have an illustration of how the information flows through the auditory structures. If we begin at the bottom left corner of the picture where we see the letters A, N, I'll walk us through it. So first, information flows through the auditory nerve labeled AN, and then it travels to the dorsal cochlear nucleus labeled DCN, and then to the inferior colliculus labeled IC, and then into the auditory cortex labeled AC. We take electrophysiological measurements from electrode probes that are inserted into each of these structures. And the electrode probes record local field potentials, which are basically just the summed and synchronous electrical activity of the individual neurons in that tissue. So later, when we look at some of the data that we collected, the DCN activity is represented by channels 1 to 32. The IC is channels 33 through 64. And the AC is channels 65 through 80. After the local field potentials from each condition are collected, they go through pre-processing and analysis using custom MATLAB programming. Next slide, please. So what exactly was done? Multi-channel electrophysiological signals were recorded from structures in the auditory cortex in the brains of several test subjects using electrodes to gather the neural signals. We then filtered the signals according to frequency ranges using custom MATLAB programming, and then we plotted the data using color maps and histograms. So the frequency ranges that we used were 4 to 8 hertz, which is in the theta range, 8 to 13 hertz, which is the alpha range, 14 to 30 hertz, which is beta, and then 30 to 60 hertz and 60 to 80 hertz, which are in the gamma range. So this allowed us to compare information flow between the structures and identify the differences in time delay of information flow between these structures. And next slide, please. So how did we accomplish this? We collected something called local field potentials from each test subject in the different conditions. Local field potentials, or LFPs, are the electric potentials recorded directly from the neurons in the brain structures that we're assessing, and they reflect the flow of information across the neural networks. We know that the information carried by the auditory nerve is processed in steps, and as we said before, it will process from the auditory nerve to the DCN region, to the IC region, to the AC region. So the time taken for the information to flow through this network from structure to structure in these regions is noted in each of the conditions. So next slide, please. So what do this data and the time delays mean? The time delay is plotted on a histogram for the frequency ranges, and the response time is captured in various regions of the auditory structures of the brain. Animals that are tinnitus positive will show different time delays in response. So color maps are used to identify the primary time delay based on the dominant color in each region of the auditory cortex. And as you'll see in a couple of slides, the regions with the dominant blue and yellow colors show these differences in time delay, with blue showing possible hyperneural activity and yellow showing an increased time delay between the structures. Next slide, please. So when we're looking at the color maps, they will represent all 80 channels of data. And just as a quick refresher, channels 1 through 32 represent the DCN, channels 33 through 64 measure the activity in the IC, and channels 65 to 80 record the AC. So we looked at several conditions and took measurements before the noise exposure in rats that did not yet have tinnitus, and then again after the noise exposure. We also measured prior to auditory cortex electrical stimulation, or ACE, which was used as a possible treatment for the rats after confirming noise-induced tinnitus. We also took measurements after the electrical stimulation to measure its effect. Next slide, please. So here we are comparing color maps from before and after the noise exposure in the 60 to 80 hertz or gamma range, showing the time delay between the IC and the AC. On the left is block 19 before noise exposure, and on the right is block 25 after noise exposure. From these color maps, we can identify that there's a significant change in response time between the inferior colliculus and the auditory cortex regions. The blue area in the bottom right corner of the before condition on the left becomes a green shade in the after condition on the right, in which tinnitus is behaviorally confirmed. The color bar on the far right shows the time delay correlations for each color. So as we can see, a change from blue to green shows an increase in the time delay between the IC and the AC structures. Next slide, please. So here we compare the histograms of the same animal and the same conditions that we just looked at in the color maps. So for before and after noise exposure conditions, and we're looking at the time delay between the structures, again in the 60 to 80 hertz or gamma range. And again, we have our before condition on the left and our after condition on the right. Each of the three columns in each condition from left to right show the activities in the DCN, as well as the response time from the DCN to the IC, and then the response time from the DCN all the way to the AC. So from these histograms, we can see that there's an overall increase in DCN activity, and that there's a significantly slower response time from the AC to the DCN in the after noise exposure condition. Again, this is what we would expect to see in a rat that does have tinnitus. Next slide, please. So here we're comparing the color maps of before and after the auditory cortex electrical stimulation, or ACE, again in the 60 to 80 hertz gamma range. For this data, we're looking at animal number 19 to see the changes that occurred after applying the electrical stimulation. On the left, we have our before ACE condition, excuse me, and on the right, we have our after ACE condition. On the far right, again, we have the color key showing the time delays that each of the colors represent. And now from these two color maps, we can identify that there's a significant change in response time between the inferior colliculus and the auditory cortex regions, which do again include channels 33 through 80. Showing increased time delay after the stimulation. So this is suggestive of hyperneural activity in the auditory structures in tinnitus positive animals. Next slide, please. So on this slide, we're comparing histograms of before and after the ACE treatment in the same animal, animal number 19. And again, for the same frequency range of 60 to 80 hertz or the gamma range. On the left, we have our before ACE, and on the right, we have our after ACE. And again, the three columns from each condition from the left to right show the activities in the DCN, the IC response time to the DCN, and then finally the AC response time to the DCN. From these histograms, we can see that there's significantly slower response time from the AC to the DCN in the after ACE condition, which could also mean less hyperneural activities in the auditory cortex. Next slide, please. So where do we go from here? This is an ongoing project and we've got so much more to learn. So I'd like to talk about what our future work and what our current work currently entails. We have so much excellent data to continue working with, and we're going to be looking more closely at that data to investigate things like the firing rates or numbers of peaks, as well as the amplitude and duration, the shape of the waves, and the frequency distribution for every channel of data. This will be done for each animal and in all of the conditions that we discussed. We will then compare the averages of these features to more readily identify the differences. So that is all for now, but I want to say thank you to everyone for staying and listening to this, and also thank you to the University of Michigan-Flint and to the Undergraduate Research Opportunity Program for supporting this research. Also, references for this presentation are available upon request. There's the slide for them as well, and I will also leave my email address in the chat. And Dr. Zhu, how are we on time? I think you're muted.
Yeah, we have about like 14-15 minutes left, so we have enough time. So now we just for any questions.
Okay.
And clapping. Good job.
Any questions? Thank you, Shelby. I think Shelby just share her email address in our chat, so for any question, you can contact her directly.
Can I ask, like, where this is so outside of my, I mean, when it was like the plant stuff, right? Like, what made you motivated to want to study the question from like a meta perspective as you a person? How did you get into it?
For me personally, I have a really strong interest in neuroscience. I'm actually currently working on my bachelor's, but for my graduate work, I'd like to be looking at brain-computer interfaces. We don't have a neuroscience program at Flint though, and so molecular biology and biotechnology were the closest things to that would help me along my pathway. But when I was looking to get involved in the undergraduate research program, I was looking through all of the different opportunities, and this one, I couldn't believe it that there was actually somebody looking at brain structure. So I got really excited about it, and I applied for it. One of my other personal reasons for being interested in this is that I'm actually hearing impaired, and I have tinnitus myself on both sides, and it's multitonal, and it's been that way for years and years and years. So there's not a whole lot that helps with it. So I think it's really cool to be able to hopefully contribute to helping to fix that for other people eventually.
It is so cool. Thank you for sharing that.
Of course. Thank you for asking.
And good luck in grad program. You'll have no problem. Like, I went to a really small undergrad. We didn't have options. Like, you could just be bio, and it's never been an issue. Like, it doesn't matter what your undergrad, like, no, that's good. Like, the research experience as part of this entire thing, like, what your research experience matters so much more than like technically what your major like was listed. I know you already know that, but in case if other people here are also thinking that and hearing it. So I don't know, that's so cool. And I feel like most people have these stories about why they're doing what they're really doing. We don't have the opportunity to share the human side of us too sometimes. So yeah, thank you so much.
Thank you.
Dr. Tonietto, that was why I really liked your presentation that you gave, I think last year, maybe the year before. You got really personal with it, and it motivated me and kind of inspired me to finish my degree. So thank you. But I did have a question for Ms. McBride as well. How closely do you get to work with the animals?
I have never actually even been in the same room with the animals before. And maybe Dr. Zhu can help answer this question, but I believe the data was actually collected, I believe at Wayne State. Dr. Zhu, is that correct?
Yes, the data was collected at Winston University School of Medicine. There's a group of research scientists. They are working on the data collection on the animals. So they have like the clean room for like noise exposure to the animals, the behavior test, and as well as the insert electrodes to the animal and do the surgery. And on our side, because I'm more in the engineering background, do the signal processing, so we are doing the collaboration. Once they collect the data, and I will take care of the processing of all the data.
So with that, how do we know which animals have this tetanitis?
So they do behavioral tests. In each of the conditions, the animals are trained to only drink water when they hear a tone. And then after that, they're exposed to a certain level of noise at a decibel range that has been shown to cause tinnitus. And then after they're exposed to this noise, they go back and then behaviorally test to confirm if they have tinnitus or not. And so basically what that translates to is then seeing if these rats who are not being exposed to sound are willing to drink water, even without external noise. And the ones that are, it tells us, okay, yes, that noise that they were exposed to did cause tinnitus.
Okay, cool.
That is
Thank
a
you for
very
the
cool
question.
setup. Yeah, that's really interesting. Wow.
Are there any other questions?
Any other questions? I guess that is all. Thank you so much, Shelby, for sharing your experience and the research result.
Thank you for having me.
Okay. Morning, everyone. My name is Dr. Christopher Lewis. I am the Director of Graduate Programs at the University of Michigan-Flint. Really excited to have you here today. It's always exciting to be able to see research being done by our students, and I'm excited to be a part of that with you today. I'm going to be moderating things, moving things along. We're going to jump into our first presentation here in just a moment. I will introduce our speakers and turn it over to them. Because I'm controlling the screen for our presenters, just let me know if when you want me to move things forward, and I will do that. So our first presentation today is by Lindsay Fox and Danielle Marzek on physical therapist and student physical therapist attitudes and beliefs regarding chronic pain. I'm going to turn this over to both of them, and I will mute myself and allow you to move forward.
All right, so you can go to the next slide. All right, so we will be presenting 3 projects today. So we have our poster project, which is physical therapy students' knowledge, attitudes, and beliefs, pre and post education. We have a current study, a survey study, physical therapists' attitudes and beliefs concerning individuals with low back pain. And we have a scoping review, so current and emerging physical therapist attitudes and beliefs towards individuals with chronic musculoskeletal pain. Next slide.
Okay, so I'm Danielle, and before talking about our studies, I first want to define a couple of terms that you'll be hearing Lindsay and I say commonly throughout each part of our presentation. So when we're talking about attitudes and beliefs, you're going to hear us say the terms biomedical and biopsychosocial fairly often. So biomedical refers to attitudes and beliefs that are centered around a patient's physical body. This refers to having a more biomedical approach to pain indicates that a person is going to look for tissue damage and pathology as the source of pain. Biopsychosocial refers to the attitudes and beliefs about pain that encompass more of the human experience. So including things like environmental factors, psychological well-being, and social disparities, in addition to also considering pathology and damage to tissues. This biopsychosocial approach is the theoretical orientation that is currently recommended for treating patients in pain, and especially those with chronic pain. So to start off, our first study that we'll be discussing here is physical therapist students' knowledge, attitudes, and beliefs pre and post education. So next slide. So this study was finished last year, and this poster that you see here was presented at the combined section meeting in San Antonio, Texas on February 5th. CSM is the largest physical therapy gathering in the country with over 11,000 attendees who were there this year. Next slide. Thank you. So for the background and purposes of this study, pain is among the top reasons that patients seek out physical therapy treatment. So ensuring that doctor of physical therapy students have proper understanding of the nature, assessment, and management of pain can lead to increased confidence when working out in the field and have better results in patient outcomes. Current recommendations state that this biopsychosocial approach, rather than a biomedical approach, should be used when treating patients with pain. The average DPT program spends about 31 hours teaching about pain, but many of these programs are unaware of recommendations to follow when incorporating pain education into their curriculum. So the purpose of this study was to determine if hosting a 6-hour online educational session could cause meaningful changes in first and second year DPT students' knowledge, attitudes, and beliefs regarding pain. Go to the next slide. We had 33 students pre-session and 23 students post-session voluntarily participate in our survey study, which utilized 2 reliable and validated questionnaires, and also ask questions regarding demographic and background information. The pain education session included information that followed the guidelines as set by the International Association for the Study of Pain, and it was completed in a virtual environment due to the COVID-19 pandemic restrictions. The surveys were emailed out 1 week prior to the session, then immediately following the conclusion of the session, allowing for participants to reply up to 2 weeks post-session. We used univariate ANOVA and regression analysis statistics in SPSS to get our analysis. So overall, we found that students' knowledge about pain increased and that their biopsychosocial attitudes and beliefs increased after attending the pain education session. Of the background information gathered, it was determined that students who were members of pain organizations and students who had received additional outside training on pain had increased knowledge and more biopsychosocial attitudes and beliefs about pain. Factors such as chronic pain history and clinical treatment experience were not significant. So overall, our study confirmed that meaningful changes can be made to pain knowledge, attitudes, and beliefs through just a 6-hour interactive pain education session. So on to the next slide. So our second study is titled Physical Therapist Attitudes and Beliefs Concerning Individuals with Low Back Pain. So after having looked at the attitudes and beliefs of students, we naturally wanted to look at the attitudes and beliefs of physical therapists who are currently practicing out in the field, and specifically those who are treating low back and chronic low back pain. So low back pain is one of the most common physical ailments that patients will seek medical treatment for. And when that pain lasts longer than 3 months, aka becoming chronic, patients' outcomes have been shown to worsen. Also, less than a year ago, a new clinical practice guideline was published for physical therapists treating low back pain. And we wanted to know, 1, if PTs knew that this existed, and 2, whether the PTs were actively using the recommendations in the CPG. Lastly, we also wanted to look at whether PTs were assessing sleep of their patients with chronic low back pain, as poor sleep habits can impact a patient's pain. Next slide. So to accomplish this, we sent out an online survey through Qualtrics to over 5,000 physical therapists who are currently practicing in the United States. The survey consisted of 2 questionnaires, which looked at attitudes and beliefs about low back pain and chronic low back pain. We also formulated our own questions about CPG awareness and usage, sleep assessment, work background, and demographic information. All survey distributions were sent out through Qualtrics using the Dillman method. This method has been shown to gather more responses based on the timing of how emails are sent. So where are we right now with this project? We actually just closed out our survey for responses this week on Tuesday. We gathered 488 responses total, which is about a little over a 9% response rate. We did run into a small issue with Qualtrics and we were unable to send out our final email, so we might have been shorted on a few responses, but we still decided to end our survey with the original timeline. So in the coming weeks, we'll be running our analysis and we are excited to see what information we gather.
All right, and our third study was the current and emerging physical therapists' attitudes and beliefs towards individuals with chronic musculoskeletal pain, a scoping review. So first of all, I just want to talk about the purpose of the study. So sort of what Danielle already talked about, research indicates that physical therapists' beliefs do impact treatment and patient beliefs as well. Next slide. In order to search for our articles, we had our librarian, Emily Newberry, help us. She searched CINEL, PubMed, PsychoInfo, and Web of Science for many different search terms in these categories: musculoskeletal pain, physical therapist or student physical therapists, and attitudes and beliefs. And so she used a bunch of terms that go into those three themes. We came up with 1,149 articles, and we have narrowed it down to 27 by using selection criteria. So such as English language, physical therapists and student physical therapists, attitudes and beliefs, and chronic musculoskeletal pain. Different criteria we used to exclude studies were that the studies were not about physical therapists or student physical therapists. For example, they might have been more general about healthcare providers, and we couldn't make any inferences about whether it was actually like the physical therapists themselves that had those beliefs. If it was an intervention study, for example, a lot of them would talk about cognitive behavior therapy and like having education about how to conduct that pre and post. And also if it talked more about patient beliefs. So those are the main exclusion criteria. And just kind of like where we're at like right now. So we created, so you could go back a slide. So we're still like in the progress. We created a table and we're currently like working on solidifying themes from the results. But we're still like working on that right now. So there's no like solidified results to talk about yet. But, yeah, thank you for listening.
Can open it up for any questions that anyone has for our presenters. Go ahead and unmute yourself.
Hi, Danielle, you said that lower back pain, when it becomes chronic, the diagnosis worsens. So the for physical therapy, so the prognosis worsens as well. And does the course of treatment change?
So in general, like when any condition becomes a more chronic condition, it just becomes, that's like one of the factors that for physical therapists we looked at as far as for prognosis. It's not that a patient won't improve or won't have, you know, but the longer they've had the issue, typically like the longer the therapy treatment will take. So kind of with thinking of that, that's why that biopsychosocial like orientation tends to be a lot more helpful, because when things become more chronic, usually a lot more stresses that have caused in that person's life. A lot of times there's additional costs. So all those factors have also kind of been shown to play into chronic pain, where a lot of patients, especially with low back pain, imaging is very common. And a lot of times, you know, if you looked at MRIs of all of our low backs, it's probably pretty likely that we all have some kind of damage, but we're all, you know, doing fine. So the idea to move away from like, oh, well, your back is super degenerated and it's bone on bone, and because a lot of people will have those kind of imaging. So knowing that it's not always that tissue damage, it could be that you've had this low back pain and now it's costing you a lot of money out of pocket. And now, you know, it's impacting your ability to be able to sit at work all day. So taking all of those into the idea with the chronicity and how those other things come into play, focusing on that biopsychosocial aspect and addressing these other areas besides just the tissue damage that is there or is not there, is where we want to focus. As far as interventions, like that's not really where we are focusing. We're more looking at like how the physical therapist attitudes and beliefs then impact like the treatment course, stuff like that. Something that is common just from articles I know that we've read over time that have been about treatment, not that that's what we're specifically looking at, but I know that a lot of PTs who are more of that biomedical will often recommend bed rest and decreased activity, where the biopsychosocial like kind of tries to integrate more pain education into their treatments, which doesn't usually advise bed rest or, you know, decreased activity. So it's been kind of interesting to see, but so interventions between those kind of therapists may change, but so we'll see. I don't know. Interventions, like I said, is not really what we're looking at, but just from what I've read, it seems that a lot of it's focused more on pain education. So I don't know if that answered your question, hopefully.
Yeah, thank you.
You're welcome.
After looking at these studies, are you finding, were there pieces in the studies that you did not look at that after now that you have gone through these studies that if you were to continue this research that you would focus on next?
So some things that I know for our future that we have wanted to look through, I don't know if you're referring more to like the scoping review stuff or stuff that we've done, but as far as our own studies, from what our research has done, I know we are interested in looking, part of the DPT program is that students go out on clinical rotations and they have something called a clinical instructor who is like the PT who they like end up working under and they take up their schedule, is again kind of surveying the students multiple times throughout the program. And then after they come back from clinicals to see if, you know, they were with a therapist who surveying the therapist as well as the students to see like, okay, does the therapist tend to have more biomedical or biopsychosocial beliefs? And then surveying the students again and seeing after that clinical rotation, after they were with the CI for, you know, 6 weeks, 10 weeks, however long, like did that then also impact their attitudes and beliefs? So that's one direction that we're interested in going in. Lindsay, I don't know if there was anything else from like the scoping review stuff that you found interesting. That would be like another avenue for like after the scoping review.
Something like I can tentatively say about like the results is that I'll probably be talking a lot about in like the scoping review about just different factors that impact beliefs. And I think there's just so many different things you can look at: history of low back pain, even like culture, gender. Like there's a lot of different factors there that I think are also pretty interesting to look into.
Thank you. Any final questions for our first presenters? Okay, thank you very much. We're going to move to our next presentation. And sorry for moving early to this slide. So our second presentation is by Azmat Sindhu, who is a graduate student at the University of Michigan-Flint. The study is longitudinal association of post-hospitalization transfer to residential care facilities among community dwelling older adults, moderated by Fall. She had a faculty advisor of Reza Amini from the Department of Public Health and Health Sciences. I will turn it over to her.
Hi, good morning, everyone. So Dr. Lewis already told you about the topic, and I am an MPH student, and the study was mainly conducted by Dr. Reza Amini. It started a few years back, but I joined in at a later stage, and it has been a lot. So the next slide, please. Thank you. Okay, so a little background. The study was mainly done on community dwelling older adults, like the older population. 15% of all the Medicare beneficiaries aged 65% of or older live in residential care facilities, meaning they live in facilities where they are not completely independent and need assistance with everyday tasks. A majority of those reside in retirement or senior housing communities. These are the communities out of all the facilities which offer the most independence. The second one is independent facilities and assisted living. Both of these are a type of residential care facilities. In these, the level of dependence is more than the retirement homes, but is relatively less than the nursing homes, which is the last one, which has the highest level of dependency. And the senior adults living in nursing homes require a lot of assistance with everyday tasks. So community dwelling older adults possess a risk of transfer to residential care facilities due to increased dependencies to perform their everyday tasks. Now, this risk is exaggerated when due to falls, which leads them to be more dependent, and their transition to a more dependent facility, thus deteriorating their overall quality of life. Dr. Lewis, next slide, please. The background of the study was that higher associations of falls are associated with cognitively impaired community dwelling older adults. So the cognitively impaired older adults seem to have a higher incidence of falls. The frequency of hospitalization due to falls is higher in old age, which is further exaggerated by dementia. Impaired balance associated with dementia can increase risk of fall, subsequently leading to more hospital admissions. According to a study conducted with patients aged 65 or older, 1,500 hospitalized patients had to transition to a residential care facility, and among them, the 40% of them had some kind of cognitive impairment. This made them more prone to falls because cognitive impairment was undiagnosed and it is associated with impaired balance. Next slide, please, Dr. So while we are living in the community, everyone's prone to some types of falls and hospitalizations, but the hospitalization is moderated by cognitive impairment and falls. Increased cognitive impairment leads to impaired balance, which leads to a higher incidence of falls, and thus transition to a facility where you have increased dependency on someone else to perform everyday tasks, thus decreasing and deteriorating your quality of life. Next slide, please. So the aim of the study was to attempt to examine how transferring to residential care facilities is associated with hospital admissions and how this transition is mediated by falls and dementia. If impaired cognitive function increases, the incidence of falls increases, and then subsequently this increases the risk of hospitalization. This was the hypothesis the study was based on. To explain the correlation between post-hospitalization transfer to residential care facilities for community dwelling older adults, how it affects them, and what this implicates. Next slide, please. So how the data was collected for this, a secondary data analysis was done from NHATS, which is a National Health and Aging Trend Study. This is an ongoing study. It has been collecting data, I think, since 2007. For every year, it collects data from the older adults, community dwelling, as well as those residing in facilities. So for this study, 9 years of data was utilized over a period of 2011 to 2019, and each wave had 2,548 participants. So NHATS collects data in the form of waves for every participant and does a series of tests to collect those data. The transfer to residential care facilities was measured based on their residency status. There was a question in the survey regarding where they resided, in what kind of facility, or if they still resided in a community. The number of hospital admissions was inquired and collected, as well as as part of a survey question asking that how many times they had been admitted in a hospital within the past year. Dementia was included in the part of the study as 3 things. Firstly, there were clock drawing tests. Secondly, there was memory recall test, which had 2 parts: delayed and immediate memory recall, which was graded on a Likert scale, indicating that what level of dementia is a patient suffering, and if the patient had been diagnosed with dementia over the past year. The number of falls over the past year was another survey question. Then Stata was used for multiple longitudinal logistic regression to examine the association between falls, hospital admissions, while controlling it for dementia. Next slide, please. So the results of the study indicated that falling increased the risk of transition to residential care facilities by 31%. Transferring to residential care facilities was significantly associated with hospital admissions after controlling for dementia and falling events. After including falling to the model, the odds ratio or the incidences of hospital admissions increased. Dementia was a significant predictor. Why a community dwelling older adult would be transitioned to a residential care facility rather than a more dependent or independent facility where the level of dependency was low. Including the interaction between fall and dementia in the final model showed that hospital admissions increased the risk of transition to a more dependent residential care facility by 61%. Dementia, as well as a history of falls, increases the risk by 22 times, while those without having a significant history of falls, this risk was 8 times lower, which was 14 times to transition into a residential care facility. Next slide, please. What this shows us is that hospital admissions due to falling is a predictor of transition for community dwelling older adults to residential care facilities. There was a 31% increase in transition to a more dependent residential care facility due to falls, which is indicative of the fact that falls were to be prevented for the community dependent dwelling older adults for them to have a more independent living lifestyle. Although there was no noticeable increase, there was noticeable increase in the hospital transition due to falls, but this increase was further amplified when cognitive impairment was considered. Transition to RCF when considering hospital admission due to falling events associated with dementia, this rate increased from 31% to 83.5%. There was an 8 times higher incidence of transition of community dwelling older adults with cognitive impairment to residential care facilities when falls were considered. Next slide, please. So the recommendation based on the study was to value screening for balance and cognitive function in community dwelling older adults at the time of admissions and discharge. This recommendation is based on the fact that if early screenings are done for older adults during their first or second hospital visits for falls, this would lead to early diagnosis, which would place them in a level of care which is suitable for them, where they can get care for their cognitive impairment, but at the same time enjoying more independence in their everyday tasks. Increased screening and early diagnosis would lead to a better preparation and placement of older adults into the facilities, which were most suitable for the level of care required by these older adults. This may reflect the necessity of screening for balance impairment and dementia among community dwelling older adults to prevent frequent hospital admissions after falling events. If older adults were placed in facilities which provided the level of care necessary for these Medicare beneficiaries, then falling events due to cognitive impairment could be prevented and would ultimately lead to a better quality of life and longevity for older adults. As indicated, if the cognitive impairment was diagnosed in early stages, they would be in a facility which would assist them as required by their medical diagnosis and decrease their incidence of unprecedented falls which were occurring due to imbalance, which was a cause of cognitive impairment, and lead to their overall better quality of life and longevity. Thank you so much. And does anybody have any questions? Well, thank you everyone for listening.
Thank you very much.
Thank you, Dr. Lewis.
Okay, we'll move on to our third presentation of the day. Our third presentation, I'm gonna get it there, there we go, is with Matthew Carpenter, who is a student at the University of Michigan-Flint. His faculty advisor was Jennifer Blackwood. I'll let him talk about that, but his presentation is on senior programming intergenerational college experience. Matthew, I'll turn it over to you.
Thank you, Dr. Lewis. So as you mentioned, I'm Matt, and this is my presentation on the SPICE project that was hosted by the University of Michigan-Flint. So I am a part of the Physical Mobility, Social Environment, and Falls lab here at University of Michigan-Flint, and this is headed by Dr. Blackwood. Next slide, please. So the SPICE project was an adjunct effort between other faculty at the university and our staff. So other faculty included Dr. Goldberg and Dr. Show-Groden. So the SPICE project was founded on the principle of intergenerational learning. So what intergenerational learning is it occurs when intergenerational projects or activities are purposefully planned to include one or several learning aims and outcomes across generations. So intergenerational learning is important because it allows seniors to express their thoughts and feelings to younger generations. And usually their thoughts and feelings are often dismissed as important today. And similarly, intergenerational learning allows younger generations to become more cognizant and welcoming of older generations. So this SPICE project stands for Senior Programming Intergenerational College Experience. And this was an 11-week course for seniors hosted through Zoom due to the coronavirus. Each week consisted of a different topic presented by University of Michigan faculty and staff, and each presentation was subsequently followed with a group discussion with student moderators that were enlisted in the program. So here's a flyer I created for the SPICE project. As you can see, it has the date, the speaker, and the topic. And as you can see, this took place last semester. And there's a wide range of topics. So anything from green chemistry to workaholism was covered in this project. So through the SPICE project, we hope to understand the self-perception of seniors and that relation to aging, and also the connection between technology use and technophilia. And technophilia is a strong enthusiasm for technology, especially new technologies such as personal computers, the internet, mobile phones. Next slide, please. So some of the factors that influence technology use in seniors. So ageism, studies have found that many older adults reject helpful technologies or new technologies to distance themselves from ageist stereotypes and negative identities. Another factor is technology is not made with seniors in mind. So they've been effectively excluded by technology companies when these companies do their market research and testing, and they don't take their opinions or the voices of senior citizens into play when making their products unless that product is specifically targeted for seniors. Another factor is the diffusion of innovation. So this is a theory that explains how and when new technology is adopted by individuals in societies. So early adopters are more willing to use these new technologies because they have been shown it's reliable. But late adopters, which seniors generally fall into this category, are more skeptical and only switch their viewpoints when it's been widely established that these technologies are good. And this skepticalness can be due to an expressed sense of helplessness when faced with new technologies that they're not familiar with. So when we're dealing with this late majority, simple is always better, and it makes it easier for seniors to learn and adapt to new technologies. And then some other factors that influence technology used by seniors. The interest and motivation is a huge one. If they're interested, they're more likely to actively try to use new technologies. And then lack of information, affordability and accessibility, technical support and social support, and then independence and confidence with technologies all influence their use. Next slide, please. So my responsibilities in the SPICE project were the development, recruitment, and communication. So for the development phase, I created manuals for students and seniors to access Zoom meetings. I created the pre and post survey through Qualtrics, and I created advertisements such as the flyer on the previous slide. So for the pre and post assessment, we asked questions of seniors' perception on aging and their aging process, as well as questions of technology. So to understand the seniors' attitudes of aging and their aging process, we utilized this measure, the Attitudes Towards Aging Questionnaire, or the AAQ. This measure was a part of an international project on the quality of life of older adults. In this collaboration, it was a collaboration with the World Health Organization. So this assessment is a 24-item scale that incorporates the concepts of both losses and gains of aging. So through the AAQ, we can determine psychosocial loss, psychological growth, and physical changes of seniors. And to assess their seniors' attitudes towards technology, we use the 6-item assessment tool, TechPH, and this assesses the tech enthusiasm and tech anxiety of seniors. So when we administered the pre-survey, we had a total of 39 completions, And in the post survey, we had a total of 16 responses. So the AAQ responses are on 2 different 5-item Likert scales. So the senior is provided with a statement, and they are to respond 1 of 2 ways, depending on the section. The first section statement asks how much you agree or disagree with the statement. And while the second set of statements asks how true the senior believes the statement to be. In both instances, the questions are used to specifically measure the aspects of psychosocial loss, psychological growth, and physical changes of seniors. So to determine a positive attitude of aging profile, it would be higher on psychosocial growth, or excuse me, psychological growth and physical change, and lower scores on psychosocial loss. So here is a screenshot of the Qualtrics survey on the production side. You can see the introductory statement with the directions, and then the Likert scale, which entails strongly disagree, disagree, uncertain agree, and strongly agree. So the relevance of the questions when we do the data analysis is dependent on what we're asking. So sometimes lower scores mean worse outcomes, and the higher score means better outcomes. So we rated it strongly disagree as a 1 and strongly agree as a 5, but it is question dependent. So we have to look at that when we do the analysis. And here is just the other, another screenshot of asking how true and some of the questions. And then you can see the different Likert scale of not true at all, to slightly true, to moderately true, very true, and strongly true. So there were 6 text pH questions, and they're listed here, but this was rated on a 5-item Likert scale of fully disagree to fully agree. Next slide, please. So for recruitment, I visited all the 15 local senior centers in Genesee County and spoke to staff and seniors about the program, as well as administered the flyers that I had created. There was some resistance from local senior centers. Staff at senior centers, in particular in the northern part of Genesee County, were expressed their concern that seniors' internet use and connectability, because it's a more rural area. Next slide, please. So we had the greatest participation of seniors from the Flushing Senior Center. During COVID, they implemented Zoom classes for their seniors, so it was a smoother transition. And we had a total of 4 participants from local senior centers in this project. So after we exhausted our resources with local senior centers, we still needed more participants. So we found participants through the U of M Health Research Network. And this enabled us to reach a greater audience. And through the UMHR system, we had a total of 34 participants sign up for the project. So this is a screenshot of the UMHR system. It's pretty intuitive. So the way it works is that you enter criteria into the system, and it'll send out a notification to the people that meet that criteria, and they have the choice of choosing interest in the participant or in the research. And if they don't want to, they can click they're not interested, and it'll go away. But on the right hand side, you can see the message inbox and message templates and message attachments, of which the message attachments can kind of be used to send information, just kind of like an email where you'd add attachments. Message templates are used for like a quick response. So for example, a thank you for expressing interest. A person from our team will contact you in X amount of days or at such and such time. And then when the seniors express interest, they will show up under the interested participants. And then from there, I can go in. I went in and sorted through them to make sure they met the criteria. So when you have interested participants, as I mentioned, they would appear under the new tab. Where's that little star on the left-hand side? I would look at their profile and screen them. Exclusion criteria included not being in the state of Michigan. They couldn't commit to the program and meet at the designated time, which was on a Wednesday night, or they did not fit the age range of the project. And then if they were eligible, I could move them by where it says move to, I could select that and move them to the eligible section, or I could move them to the ineligible section if they didn't meet the criteria for the project. And then additionally, you can add labels to identify what tasks you've done. So for the project, the green meant that I contacted them either via the phone or through the system and screened them. And then there's some other labels that we have created there, such as not interested, or we sent the Zoom information and the attachments and the culture link to them. So this graph demonstrates the attendance rates through the SPICE lectures. So as you can see, the first 2 lectures, we had a few participants. That's before we started using the UMHR system. And then around October, our rates increased. The blue line there is the amount of students that we had in the project. So we had a total of 6 until the end, where we only had 5. And then you can see there's kind of a drop off between November 17th and December 1st. And that was just because we didn't have a presentation on Thanksgiving, and it took a little while for people to come back. So as you can see, it came back up to 14 participants at the last presentation. So, and then as I mentioned, I did communication. So I administered both the pre and post surveys and gathered together research data. And then I communicated with seniors through the UMHR system. So our next steps are to complete the analysis through SPSS and determine other analysis to summarize our findings in a manuscript that will be published. And our aim is to have a manuscript published by the Geriatrical Society of America and have a subsequent presentation during their conference next. I believe it's in October or November of this upcoming October or November. And those are my references.
We'll open it up for any questions for Matthew.
Hi, Matthew. Very nice presentation. You said that you faced resistance while recruiting some of the older patients. Any reason you feel some were more approachable and some didn't want to be included in this?
Well, I believe that it was actually mainly from the directors of the senior centers. They often expressed that, they didn't believe that a lot of people would join, that they were worried about, as I mentioned, in Northern Genesee County, they were worried about their internet connectability and reliability, or a lot of them, while some of it is true that seniors aren't as open to new technologies, it doesn't mean that they're not willing to try. And I believe that some of these directors of the senior center kind of had ageist views where they're like, oh, they're never going to be able to do this. They can't learn how to do this. So they said I'm more than welcome to talk to them. And I did get some interest from some of the seniors, but they didn't believe that the project would really go anywhere with the local seniors. So I think it was more of the directors, not so much the seniors themselves.
Thank you.
You're welcome.
Matthew, as you look at the project as a whole and think about the future, if this was to continue, what kind of things would you recommend to make changes on to be able to engage others or other additional topics that might be of interest in the future?
Well, so I wasn't directly related in or responsible for determining the topics. I think they want to add a broader range of topics just so we can get that information out there to seniors so they're actually learning in this project and learning new things and excited about engaging in the university environment. Hopefully, once things with COVID work themselves out, maybe that we'll have something where they can come on campus. That was something that a lot of them expressed that they wish they could do, is come onto campus. And it works not only in involving them in the university environment and with students and interacting with different people, but also serves as like a kind of a community thing for them as well. So they're learning at the same time. So I think that's what the future goals would be.
Just a follow-up question on one of your data slides in regards to attendance as well. As you looked at the individuals that were engaged throughout the project, was there consistency of because I saw like at the high point for older adults, it looked like 14. So was there much overlap in individuals coming to multiple or all sessions, or was there many that people were just kind of coming and going and were kind of picking and choosing?
So I was not actually present for the presentations, just because I was more of on the development side, but I believe that a majority of the participants were regulars there. And when we looked at the post-assessment, I believe that the ones that continued to answer those questions or answer the post-assessment were the ones that were consistent and at each presentation. So yes.
Great, thank you.
You're welcome.
Other questions for Matthew? Nice job, Matthew. Thank you.
Thank you.
We're going to move to our fourth presentation today and move it to the next slide here. And that is with Sindhu Muraladharan. I hope I said that correct, Sindhu. But Sindhu is talking about blood pressure monitoring in outpatient therapy clinics. Her faculty sponsor was Leslie Smith. And I'll turn it over to Sindhu.
Hello, everyone. Thank you for the introduction. My name is Sindhu Muralidharan, and I'm a second-year physical therapy student at the University of Michigan-Flint. I've been working with Dr. Leslie Smith for almost a year now on this project. Next slide, please. Hypertension is a prevalent and preventable risk factor for several medical conditions that can lead to death. The national prevalence rate of pre-hypertensive blood pressure measure have been reported at 28%, and hypertensive blood pressure have been reported at 33%. About 1 in 3 adults have been diagnosed with hypertension, and nearly half of these individuals do not have their blood pressure under control. Within the outpatient therapy settings, nearly half of the patients have been diagnosed with hypertension or pre-hypertension. However, most of these patients, most of the PTs do not regularly monitor blood pressure. The statistics here are based on several studies. About 47% in an outpatient setting were pre-hypertensive or hypertensive. 43% of PTs reported of never measuring blood pressure. 52% reported that monitoring blood pressure were not important for their patient population. They also found that PT's knowledge of normal, pre-hypertensive, and hypertensive values were poor. Next slide, please. Outpatient physical therapists have been called to routinely assess blood pressure in order to proactively address the hypertension epidemic in the United States. It has been suggested that physical therapists have an ethical duty to screen blood pressures and provide an appropriate medical referral when necessary, as well as providing information on equipment requirements, techniques, and clinical decision-making for abnormal blood pressure readings. The purpose of this study is to determine if physical therapists and outpatient clinics are taking blood pressure regularly and use of education via webinars to attempt to improve frequency of blood pressure monitoring. Next slide, please. To collect data for phase 1 of the study on whether PTs were monitoring blood pressure, an 18-item survey was developed. It consisted of questions regarding of clinicians' attitudes, knowledge, behaviors, and barriers that they had towards blood pressure monitoring. The following items listed below were what was included in the survey. This was distributed to 20 clinicians who were employed at one hospital-based clinic system. Next slide, please. Thank you. Chart audits were also performed to collect data. 508 de-identified patient charts from electronic medical records of PT initial evaluation from November 2019 were used. To analyze the data, we looked at blood pressure, age of the patient, medications the patients were using, history of hypertension, other cardiovascular diseases, and any recent surgery performed on the patient. Next slide, please. The results of phase 1 of the survey showed that 9 clinicians participated in the study. 100% reported of having access to blood pressure measuring devices. 40.3% agreed that blood pressure should be taken on all patient population during an initial evaluation or re-evaluation. 100% rarely or occasionally took blood pressure. Next slide, please. The results of the chart audit showed that 0% of the clinicians documented blood pressure during initial evaluation. The average age of the patients were 57 years. 22.2% of the patients were diagnosed with hypertension. 38.1% had one or more cardiovascular diagnoses. 10.2% had diagnosed metabolic disorder, and 11% had a recent surgery. Next slide, please. Thank you. After phase 1 of the study, a series of 4 webinar series were conducted to educate the clinicians about blood pressure monitoring. Webinar 1 included why blood pressure measurement is important for clinicians' assessment of patients and to understand the poor frequency of blood pressure management in the outpatient setting. Webinar 2 included information on American Heart Association blood pressure guideline and to learn about a blood pressure algorithm for clinical decision-making. It also included information on hypotension. The objectives for Webinar 3 were to learn about blood pressure instrumentation and how to calibrate it. The objectives for Webinar 4 were to learn how to monitor and respond to blood pressure measurements during or after exercise. Next slide, please. In phase 2 of the study, another series of chart audits were performed after the webinar series. Same as before, we used a de-identified initial PT evaluation patient data from July 2021. 251 charts were audited this time, and in this phase, 8 clinicians participated. Next slide, please. The results showed that blood pressure documentation improved by 16% during PT initial evaluation after the webinar series. The average age of the patients were 59 years. 24.7% were diagnosed with hypertension. 34.3% had one or more cardiovascular diagnoses. 6% had a metabolic disorder, and 8.3% had a recent surgery. The important discussion points for this study are that about a quarter of the patients were diagnosed with hypertension, and a third of the patients had a cardiovascular disease or risk factor. For the clinicians who participated in this study, the time commitment to watch the webinar series were low, and it was feasible to complete. During phase 2, we found out the webinar series had increased the frequency of blood pressure monitoring. A knowledge translation may be required to increase the frequency of blood pressure monitoring. According to the Canadian Institute of Health Research, a knowledge translation is a dynamic, iterative process that includes synthesis, dissemination, exchange, and ethically sound application of knowledge to improve health, provide more effective health services and products, and strengthen the healthcare system. Next slide, please. The limitations of the study are that there were only a small number of participants who participated in the study. Lower time commitment might not be enough, and so a longer and more detailed webinars in the future can help. We had decreased access to the clinic's clinicians and the clinic supervisor or manager to give us the data on time. Information was gathered from only one hospital-based clinic system, and hence the results of this study cannot be generalized. Next slide, please. For future studies, a knowledge translation will be beneficial and can be implemented into outpatient clinics to improve frequency of blood pressure monitoring. A knowledge translation can help close the gap between what we know now and what we do. It makes users aware of knowledge and facilitating their use of it to improve health and healthcare systems and transfer knowledge into practice. More frequent education can help with blood pressure monitoring during an initial or re-evaluation. Repeating the survey and chart audits with multiple outpatient therapy clinics can also be beneficial. Having outpatient therapy clinics require documentation of blood pressure can improve frequency of blood pressure monitoring in an initial eval or reevaluation. Next slide, please. To conclude, I just wanted to say that outpatient therapy clinics have been routinely asked to monitor blood pressure in order to proactively address the hypertension epidemic in the United States. This study demonstrated that providing education on hypertension and blood pressure monitoring is feasible to improve blood pressure, to improve the frequency of blood pressure taken in outpatient therapy clinics. Next slide, please. These are my references. Does anybody have any questions for me?
Hi, Sindhu, and everybody else in the room. Sindhu, I like this study. I'm excited. I didn't know you and Dr. Smith were doing this because this is really interesting. 22, maybe 22 and a half years ago, I did a study very similar to this when I was in this program. But we, at the time, the Guide to Physical Therapist Practice had just come out, and it was an observational study. So just kind of background, we would go to outpatient clinics, and we would just stand in the corner and observe whether or not the therapist took any sort of cardiovascular response, blood pressure or heart rate. Heart rate monitoring was like at about 10%, but blood pressure was like next to nothing. So what's concerning to me is 22 years after we did this, even though the Guide to PT Practice has been around for a long time, practice patterns may not have changed as much in that first measurement that you had. The webinars that they watched, then there's a change after that. And so I want to know like a couple of things relative to the study is, were the webinars live by chance or were they recorded?
They were recorded. So due to COVID, we had to do recorded webinars.
Yeah, I think that's good. I like the idea of, you know, the webinars changing practice and in this format. And because I think even if it's 16%, I mean, based on some of the results that you had, these people were hypertensive already. If they already didn't have the diagnosis, that may have been picked up. And I think that's just really commendable for you guys as a study to look at the outcome, right? Because it's always the issue of, you sit in on a course, you learn about something like this, but then what's the next step? And this obviously showed an impact. I wonder if there's a follow-up. So is there an opportunity for you guys to go 3 months or 6 months down the line, look at those same 8, I think it was 8 therapists, and look at their charts to say, has this been maintained over time? Is that a part that you guys are considering?
So yes, so what our plan is for future is to make this a knowledge translation. So with that, I think, like I said in the presentation, like more longer and detailed like webinars on how to take blood pressure would probably be helpful for like the clinicians who already watched the webinars and also for other clinicians who would want to participate. And so I was talking to Dr. Smith yesterday on it, and she was telling me that it would be really beneficial to see that if they are continuing taking blood pressure. So that is definitely one of our focus for this study. Moving on.
Thank you. And last question, sorry to monopolize the questions, but your references number 1 and 5, Sarah Arena is a PT faculty member from Oakland University. So if you already haven't done that, I'd encourage you guys to reach out to her and maybe collaborate across universities on something like this, because she's done a significant amount of work in this area, but I think it's really great to, you know, maybe have Michigan be that leading factor, not U of M, but the state itself with this change in practice pattern. So really awesome. Thanks, guys.
Thank you.
Other questions for Sindhu? Just one observation that I would throw out there too, Sindhu, is, you know, with the asynchronous learning that you were able to provide with the webinars that were created, it might be something that as you move forward or as the project moves forward, that there might be something there to offer those webinars on the website, on the PT website, offer it as something that you can continue to offer professional development and push out for others to try to encourage more to watch and to promote that type of learning.
Thank you. I appreciate that comment.
Other questions? Okay, well, we're going to be very soon getting done with our breakout rooms and going back to our main room. So I just want to say thank you to all of our presenters today. The presentations were wonderful. It's always great to be able to see and experience research from our students. And I commend you for all the hard work, because it is not always something that that is easy to do. It takes a lot of time and effort and focus, and I commend you and encourage you to push your other colleagues, your other students to follow in your footsteps, because it's a great opportunity for you and for them to be able to push themselves throughout their education while here at U of M Flint or at other institutions, to be able to encourage, to be able to learn and to grow. Okay.
I can't, so I have the timer with me. So when we hit the 10-minute mark, I'll just remind you that you hit the 10-minute mark, and then you'll have like 2 minutes to wrap up, and then remaining 3 minutes we are going to spare for the questions. Okay?
Okay.
All right. Just tell me when you are ready. You wanna go ahead and start?
So I'm ready now.
Okay, floor is yours. Go ahead.
So, hello. This is my thesis work, actually, machine learning at the edge for disaster response. It is, of course, done by me because it's my thesis. I'm a graduate student in the Department of Mathematics and Applied Sciences at U of M Flint, and my thesis advisor is Dr. Suleiman Uludağ. He's an associate professor in the same department, and his research forte is mostly networking, and he actually came to me about my thesis interests, and I accepted, and so here we are. The topics we'll cover today are the EMS problem, what challenge are we even addressing, a possible solution that we've identified that could make a difference, and then how we plan to use it. So why is this necessary at all? The problem we're trying to solve is one of climate catastrophe. There are many island nations that have voiced their concerns in recent years, talking of the threat posed by increased hurricane frequency and potency. And as you can see by this quote by Ambassador Janine Felsen from the UN, they are particularly worried about hurricanes like Dorian and Maria coming once again and threatening places such as the Caribbean and even the United States. To that end, this has real implications in people's lives. As sea level rises, communities and governments are strained. Land is changed, displacing people and destroying infrastructure they rely on. And with that, stability and faith in government is threatened, precipitating disorder throughout the world. And on this map, you can actually see a projection out to the year 2050, in which researchers believe that most of Manila will actually be underwater if we don't do something fast. So what is a possible solution, and what does it look like? First of all, the solution that we're looking at right now is called LoRa. LoRa stands for long range. While cellular towers are robust, they're still prone to destruction and outages. Other methods like Bluetooth and Wi-Fi meshes require volunteers, and it can also tax battery life of the phones quite heavily. LoRa, on the other hand, allows for low cost and low energy gathering of data from victims for EMS personnel. And as you can see in the image description here, we have phones connecting to lower nodes, which then connect to other lower nodes, basically shuffling information through the mesh network onto this small owl looking point that can optionally connect to the internet, where EMS can gather all the information they need. But what exactly does LoRa bring to the table? Well, first of all, LoRa mainly brings its range. While the data rate is quite slow, it is enough for the equivalent of SMS to get critical data about system or about victims in need. And the implementation we'll be using is actually called the cluster duck protocol and implements mesh networking. The cluster duck protocol is a project from Project OWL, which is one that I actually worked with, an open source software and hardware solution that seems to make LoRa a bit more accessible for specifically emergency response. Another perk is that the LoRa standard, the physical layer that is standardized is backed by an alliance that's simply just called the LoRa Alliance, which includes many different companies, including some you've heard of, like Cisco and Amazon, and some maybe you haven't heard of, but are still really big in the chip space, like Semtech. The system model that we're going to be using for the data collection is going to be a mesh network. The nomenclature used by the cluster duck protocol is what we'll use in this research. Mama ducks are basically relay nodes that can forward messages to other mama ducks until they reach the sink called a papa duck. This will be connected to a low power computer, maybe a laptop of some sort at a base station where the EMS is operating out of. Data payloads can be in anything communicable in text, although technically the nodes are data type agnostic, as in everything is actually communicated in bytes. So there is research showing that you can communicate images over this technology, although it's very slow. The Papa Duck can optionally connected to the internet, as I intimated before in the last slide. If that resource is available, but it shouldn't be counted on that there is an internet connection because, as I said earlier, cellular networks may be down and you may not have weather that's good enough for a satellite connection like Iridium or something like that. Going one level deeper, mobile phones are necessary for this to work. They first use Bluetooth low energy to communicate with the mama ducks, and that has about a 10-meter range. And Wi-Fi specifically is not used because of how taxing it is on the phone's battery. And we want these phones to last as long as possible so people can be reached whenever they can. Whenever the emergency responders can get to them. Once that data is communicated from the smartphone over Bluetooth to a mama duck, it then travels throughout the cluster duck network until it reaches a papa duck. And then, what exactly do we even do with this data? Well, to tie it all together, we have machine learning used in two research problems. One is cluster formation, which is basically using GPS to group people into cohorts and to make serving the most people possible more efficient. Then there's minimizing the ETA, which is paramount in EMS. Normally, you just use Google Maps, but remember, we may not have internet connectivity or even utility power in order to access Google servers. So, how do we even solve these problems? Well, that's where our really low budget computing comes in. First, the clustering is taken care of by the dbScan algorithm from the scikit-learn Python library on, say, a Raspberry Pi or Jetson Nano. Both are very low-cost computers, about $200 total once fully kitted. Travel time estimated will be using graph neural networks, which are implemented by the TensorFlow deep learning framework, which can also be run on either the Raspberry Pi or Jetson Nano. Again, we haven't figured out which one yet because we're still trying to work out costs and availability due to the pandemic, but it still shouldn't be anywhere over $250 or so. And then the lower radios that we're using, you can see an example on this slide. One is called the TT-Go T-Beam version 1.1. It has GPS and a small ESP32 processor, and each one is roughly $40 a piece, but can be significantly cheaper when buying in bulk. So if we buy 50 of these, maybe only $32, and then so on from there. And with that comes the end of this presentation. I hope you have a few questions for me because, 10 minutes, I honestly don't think is enough for a thesis project of this complexity. So do you have any for me?
Thank you, Brenton, for the nice presentation. Yeah, let's ask the audience if they have any question.
And I'm sorry about the noise in the background. My parents are getting their kitchen remodeled.
And after a while, it becomes a white noise, and we stop hearing that. So don't worry about it.
Okay.
So any questions?
If not, yeah, I was wondering, could you paint the picture of the mama ducks again? Are they mobile and papa ducks are stationary, or how do the mama ducks communicate with the papa duck?
So the mama ducks they should be stationary, but they can move if you want. There's no reason why you can't stick one on the top of a emergency medical vehicle or an emergency response vehicle like a truck or something. But the thing is that people need to be able to reach one, and so ideally you would want to blanket an area with these things, and so that wherever people congregate, there might be one within their vicinity. It is possible to get a greater range of 10 meters with Bluetooth Low Energy, but it usually requires a lot more energy in order to do that. But we want to make these radios last as long as possible, so therefore we want them consuming as little power as possible. Having said that, you could just attach bigger batteries to them. So, we can play with the lifetime of the nodes forever, but we do envision them being stationary and then attached to maybe ground level infrastructure, while maybe another subset of them are up higher, so that they don't have to travel through as much attenuating infrastructure and have their signals distorted by, say, debris and city infrastructure.
Okay.
Thank you.
All right. Let me ask another question. So I'm just curious, why did you choose dbScan over other clustering algorithms?
Hold on. I need to raise the volume of my speakers.
Okay. You hear me now? Okay.
Hold on. I'm trying to. Okay, say that again.
I was just curious why you chose DB-scan over other clustering algorithms. Yeah. Actually, I have a couple other questions, but I'm going to ask them offline later on. So let's start with the DB-scan. Why did you choose DB-scan over, let's say, k-means or some other algorithms?
So the thing about k-means is that you need to know exactly how many clusters you want beforehand.
Mm-hmm.
And that's a bit of a problem when, you know, people are distributed around an area in manners that you don't understand yet. In a disaster scenario, you're not going to know how many clusters to group people in. So you really just want to see, where are people actually? Where are they congregating? Could we perhaps group some of those smaller cohorts into super cohorts somewhere else? And in DB-scan allows us a little bit of flexibility in determining, you know, where exactly, you know, people are, and also how many clusters we're going to get. One of the nice things about DB-scan is that it only operates on the logic of the Euclidean distance, and does not try to massage anyone into one particular group based upon the fact that, oh, you specified 4 clusters, so I will give you 4 clusters, even if those clusters don't make sense right now.
But still, you'll have to deal with the epsilon and mean points. So it has its own parameters, though. So just a reminder. Okay, so we are going to move to the next presenter. Yusuf, would you like to share your screen, or do you want me to share your slides?
I can share my screen.
Okay. All right.
Can you see it?
Yep. Yeah. Yeah, you go ahead.
Hello, my name is Yusuf Korkmaz, and I am a graduate student in computer science at University of Michigan-Flint. Today, I will present you my research, protecting the smart grid against data injection attacks using deep learning. First, I will explain you fuzzer measurement unit, PMU devices, and our data set. Then I will talk about our false data injection scope, which is repeated last value. Using this attack, we created 2 different scenarios. I will give a brief about machine and deep learning methods. When we come to the results slide, we will see the differences of the scenario, and they are going to help us to choose protection method. I will finish my presentation with conclusion and future work. Fuzzer measurement units, PMUs, are used to estimate magnitude and phase angle of electrical phasor quantity in electricity grid. Also, phasor measurement units, PMUs, are used to estimate the state and operational health of the power grid. PMU data is being sent to the phasor data collector, which is PDC unit, through legacy network. They are using a specific protocol each other to send the data. Unfortunately, this protocol doesn't include the security mechanism, and it is vulnerable to cyber attacks. And in this slide, you can see the generic PMU block diagram to reading the analog inputs to the sending the data. In this picture, you can see the PMU's deployment in the United States of America. Every year, PMU's number is increasing, and the researcher thinks PMU will be the key feature of the future smart grid. And we can estimate or assume PMU's number will be much more high in a couple of years. PMU capable to send 18 different features, which is like a GPS location, latency, magnitude, some measurements. And we have 7 different PMUs in our data set, and all of them doesn't send the 18 of them, but the 7 of them is common, which you can see in the second figure, latency, frequency to the phase, We choose to use these 9 features in our model because the PMU is sending just commonly these 9 features. There are vulnerabilities during the data transfer between PMU and PDC. In one potential attack, adversaries may simply keep injecting a repeated measurement after a compromise of PMU to disrupt the monitoring system. This attack is referred to as repeated last value, which is our scope in this research. First figure shows the PMU's data stream. And if we want to apply repeated last value attack to this stream in T7 time, we should send T7 data instead of the real measurement. In the second figure, you can see the repeated last value attack applied version of the data set. For applying repeated last value, we need two different things. First of them, when you want to apply, which time you are going to apply the repeated last value attack. And second one is how many times you want to inject the false information. With these two information, you can apply the repeated last value attack to the data set. We created two different scenarios for repeated last value attack. This is our first scenario. The first figure represents 1 day's PMU data set. Each hour we get 180,000 data from PMUs. For a day, it becomes 43,200,000 data. In all scenarios, we randomly choose the attack time. And for this scenario, we set the lower bound for attack count as a 50, but we didn't set any upper bound. There is an example for this scenario in here. We choose the attack time randomly 327, and attack count is 418. And we applied for each hour this attack. And after applying for 24 hours PMU, the data set looks like the last figure. And also we have another scenario. We created a scenario closer to real attack because we think criminals, cyber criminals will not attack your PMU constantly, like more than 30 minutes. We think the criminals will attack in a couple of samples, and we created this scenario to closer to real time. The upper bound and lower bound are closer to each other. Lower bound is 10, upper one is 40. With this scenario, it becomes harder to detect the attacks. And this is the example of the second scenario. We choose the attack count, as you can see, is 28, which is lower than 300 something. And it is harder to detect for the machine learning algorithms too. In the last figure, you can see the applied attack data set. And we use frame approach in our model, which is a novel approach to solve this problem. In our data set, we have 7 PMUs, and each PMU sends 9 common features, which makes 69 features, and 1 labeled, which is spoofed or not, the repeated last value attacked or not, real measurement or spoofed data, and which makes 70 labels. And we used frame. Our frame size is 50. Each frame has 50 rows and 70 features. Also, frames can be used as an image. And this gives us to use another algorithms like image classification algorithm for detecting the spoof data. You can see here the R frames and the applied attack data set. We can use the frames as an image, and that's why we choose to use convolutional neural networks in our detection or detecting spoof data. As you know, convolutional neural network is famous with image classifications, and we think we can catch some features in our frame to detect the spoof data. And we use convolutional network, and also we use support vector machine to make the classification algorithms. SVM finds the hyperplane with maximum margin to separate points from different classes. In our data set, we have 24 hours. And in these 24 hours, we have 86,400 frames. We choose to use 6 hours consecutive data for the training and the other 18 hours for testing. In the training part, we have 21,600 frames. In the testing part, we have 64,800 frames. We created 6 different data sets according to the amount of attack count in an hour, 500 attacks to 3,000 attack. As you can see, when we increase the number of attacks, it increases the spoofed frame too. For example, in 6 hours training, 500 dataset has 83 spoofed frame. In dataset 6, 3,000 attacked, we have almost 400, 3,099 spoofed frames. And we use this training and testing frames to detect the spoofed frames. Spoofed frame numbers are too low. If we compare with unchanged data, we need a metric which will show us how much we are predicting the spoofed data. And that's why we want to use another metric instead of accuracy. And that's why we focus, we choose to use true positive rate to compare the different methods like CNN to SVM. And we focused accuracy and true positive rate for this problem. In this slide, you can see the results. Green line represents the true positive rate of SVM. Red line shows the true positive rate of CNN. This result for scenario 1, which was there is no upper bound for the attack. And accuracies are almost same. It is not fluctuation between CNN and SVM. And also SVM and CNN gives almost same results. And it is hard to choose which one is better. And that's why we use second scenario, which is closer attack to the real scenario. And in this scenario, accuracy is almost same again, but if we look at the true positive rate, if I need to say again, the green line represents the true positive rate of SVM, red line represents the true positive rate of CNN. And we can see there is a huge differences between CNN and TPR's true positive rate. We can detect spoofed data CNN better than the SVM. And as a conclusion, we came up with these things. The frame-based approach gives us so many advantages to use image classification, and it gives us better accuracy and TPR. But also frame-based SVM and CNN could be used to detect attacks with more than 90% accuracy, but CNN is better to detect challenging spoofed frames than SVM. And for our future work, we are planning to use another false data injection methods like mirror spoofing, and we would like to use other algorithms to detect the spoofed data. Thank you for listening.
Thank you, Yusuf, for the presentation. Yeah, we have 2 minutes for the questions, and then we can move to the next presentation. Any questions from the audience?
Yeah, I didn't see the whole thing because I've been hopping around, but I did have a question about your 25-75 training test split. Is this just because of compute, or why did you choose the lower training set? I think traditionally I've seen a much higher training test ratio.
Yes, in our problem, we should predict the continuous data because we are getting 50 samples in each second, and we don't have that much training. We couldn't have so many training data set, and we would like to see how we are doing good if we have just a small training data sets. It represents like to be closer to the real time. That's why we choose smaller train hours than the test hours.
So is this parameter varied at all, or is this a set only use 6 hours here?
We just use 6 hours for training and 18 hours for testing.
Yeah,
thank you.
They would like to see how far you can go. So normally you can train on 6 hours and test in the like maybe following 2 hours. So that would have been a conventional machine learning problem, right? So you have a bigger training set, and then you have a testing set which is small in size, and you can basically measure and gauge your success rate for the algorithm that you are providing. But here, what is being tried to do is the fact that can you really do this in a long run, in a long shot? Let's say you have this trained model. Can you really take care of the remaining, let's say, remaining of the day or remaining of the week? So even 6 hours is like, it's a large data set, I would say. So it is something maybe you can basically go with in this kind of scenario. But traditionally, you are right. We choose 75% versus 25%. Yeah.
Scenario, you might start your next training phase 12 hours into the testing, so that then once this testing finishes up, you have a new model to predict. So it's like a continuous update approach.
Yeah, that is the ideal scenario. But the computations, take long time. So they are in the process of optimizing this process. Yeah, because the ideal is you should make this dynamic. I believe we are running out of time. So our third presenters.
So
association. There seems to be different associations between oral hygiene and cardiovascular diseases. And so we're going to look at one particular study, one particular cohort study done in Korea that looked at many different patients and observed their oral hygiene habits, and then observed them for 10 years, technically 9.5 years, and then followed up to look at their cardiac history to find out, is there an association between different oral hygiene habits and cardiovascular events? So what I want to do right now is give you 2 definitions that we're kind of going to be, or I'll be saying over and over again, and that is, what do I mean when I say proper oral hygiene? And what I mean by that is this: it's just frequent toothbrushing, which frequent meaning at least twice a day for 2 minutes each time, and maintaining regular dental visits, which means at least once a year, but preferably twice. So when this article talks about maintaining oral hygiene or proper oral hygiene, it always means, unless it otherwise specifies, it means brushing at least twice a day for 2 minutes each time, and maintaining at least 1, but preferably 2 dental hygiene visits a year. And then what does it mean when we say major cardiac event? And that simply means it means if we have a patient that has a major cardiac event, there's either a cardiac death, a myocardial infarction or a heart attack, a stroke, or some sort of heart failure. So those are what they consider to be major cardiac events. And so what I want to do now is, right before we look at the details of the study, is look into the possible pathways that we see leading from oral hygiene and dental health to cardiovascular disease, because it seems like something that would be almost unrelated, but there's some very strong connections. And so this figure here shows us kind of what those pathways could be. And so what we see here is we see this little zoomed up part of the figure shows that you have bacteria buildup on the outside of the tooth layers, and that bacteria is promoted by these various different variables such as tooth loss, dental caries or cavities, and then periodontal disease, which are just gum inflammation and issues with the gums. And without this protection that we're looking at, there's all these different pathways that can lead to different cardiovascular diseases. And so the study that was done was a cohort study, and it looked at 240,696 individuals with no history of major cardiac events. So they looked at 58% men and 42% women, and they were all 40 years or over, but they ranged from 46 to 60 years old. And they looked at them from 2002 to 2003, all the way up until 2013. So their timeline was almost 10 years. And then they collected their data from the National Health Insurance System, which in Korea is it'll specify this later, but it is the main source of health records or the only source pretty much of health records in that country. So basically they had access to the entire continent there of Korea to look at all these individuals. So there was about a little over 500,000 individuals that were looked at, but then 247,000 were eligible because they had no major history of major cardiac events. So their research goal when looking into this was to, quote, evaluate whether oral hygiene behavior can alleviate cardiovascular risk associated with oral health status. And so that was their research goal going into this. And so here's what they did. Their preliminary data looked like this. They looked into the National Health Insurance System, which is the sole insurance provider for Korea and covers essentially every single citizen. So they got their data from this, being a cohort study. And then what they did is they had two forms of collection methods. They had an individual survey that asked people's personal questions on their amount of dental visits. Do they have any dental symptoms such as missing teeth or pain or different sensitivities? And then they had an oral health examination performed by dentists and hygienists to look for more specific issues such as periodontal disease with the gums, number of decayed teeth, are there any fillings, are there any missing teeth? And they looked at more dental specific objective data in the health of their teeth and their oral. So then what they had is they recorded 5 pieces of data that they were looking at. What they saw, they looked at periodontal disease according to that very long acronym, which is the International Statistical Classification of Diseases and Related Health Problems. So that ISCDR-HP is what they use, that standard of periodontal disease to gauge the gum health of their patient. Then what they looked at is they categorized different dental caries, or the amount of cavities in their patients, from 0 to 1 to 5, and then 6 or more cavities. And then they did the same thing with tooth loss, toothbrushing, and dental visits. They had different categories of the amounts of tooth loss, the amount of toothbrushing, and then the amount of dental visits that these patients had. So they recorded all this data right up from the front. So out of the nearly 250,000 people that were recorded, they went over and looked at the insurance health system to figure out who qualified with no major previous cardiac disease. Then they had individual surveys that were done, and then they collected data on the oral health exams. And then they began to wait. What they did is after 9.5 years of waiting and evaluating these patients every once in a while, they found that of those people, there were nearly 15,000, but more specifically, 14,893 major cardiovascular events. And so those events, that's the total number of events. And then they broke those down into categories. And so here's what we were looking at. So here's the first 3 categories, those 5 initial categories that we saw earlier in the little boxes, which were the periodontal disease, the dental caries, tooth loss, dental visits, and toothbrushing. We see here that if we want to look at figure A, the blue line measures patients that from the beginning of the time to the end of the time of the 9.5 years had no periodontal disease. So over 10 years, they started with no periodontal disease and they ended with no periodontal disease. And the red line here shows the patient that started with periodontal disease and also ended with periodontal disease. And what this is showing you is this y-axis says freedom from cardiovascular events. So if we notice this blue line here in figure A is higher on the y-axis, which means they had a higher freedom from cardiovascular events. So the risk was lower for people that started and ended with no periodontal disease. And then as we go to figure B and figure C, we look at dental caries or cavities, and we can see this red line here says when a patient started with more than 6 cavities, their freedom from cardiovascular events decreased significantly compared to people with fewer cavities. So the risk of cardiovascular events was higher for people the more cavities that they had. And then the same thing with figure 6, tooth loss. They had different, those different categories that we specified earlier. They measured people with 0 teeth loss or 0 tooth loss, 1 to 7, and then 8 to 14, 15 to 21, and then 22 to 28. And you can see as the number of missing teeth increases, the freedom from cardiovascular events decrease. So the risk is then higher for more periodontal disease, for the more amount of dental caries, and the same thing for the more amount of tooth loss. And then lastly, they had 2 more categories of brushing and then professional dental cleaning. So we'll just take a look at these 2 very briefly. And we see that for toothbrushing here in figure D, we have, if we look at this red line, we have, these are people that brushed 0 to 1 time a day at most. They began brushing at the start of this at about 0 to once a day and ended at the same 0 to once a day. And we can see that their freedom from cardiovascular events decreased more significantly than people that brushed twice a day, or people, and then even more than people that brushed 3 or more times a day. And so the same thing, the same trend that we're seeing here is with figure E as well. People that did not maintain the proper amount of professional dental cleanings had a lower freedom from cardiovascular events. So each one of these 5 areas show and give insight to the fact that with all the other areas, they looked at many different adjustments and they looked at different areas. They found that these 5 categories were consistently across every patient here, basically, that the more neglect to oral hygiene, the higher risk you have of having a major cardiac event. And so their final conclusions were this. They said, what we believe is this: even though, according to the American Heart, there's no strong data for a causal relationship even though we don't say, okay, if you don't brush your teeth, this will happen. There isn't a clear causal relationship, but what we do have is a very significant and a strong association. And so now what we get to do is we get to take this study and we get to look at, okay, if we make now simple behavioral changes in these patients, such as maintaining better dental visits and brushing more often and watching our cavities and the things that we eat and our sugars and different things, do we then have a trend where these same patients will now be free again from cardiovascular events? And can we reverse this risk by just implementing better oral hygiene habits? And so that's what their entire focus was for this study, was to determine if there's even an association, if there's any sort of relationship. And they found that there seemed to be a very strong relationship, not a causal relationship, but definitely an association between these 5 areas. And now we get to take these areas and go further into studies to figure out, okay, let's make some healthy and simple behavioral changes at home to prevent this risk. Because by doing that, we can save our health system tons of patients. We can save people tons of money, because now you don't have to go and deal with oral hygiene or surgery or periodontal disease issues. You can just, by maintaining simple oral hygiene habits, alleviate yourself and reduce the risk of major cardiovascular events. And that was the main takeaway from this study.
Thank you, Riley. Does anybody have any questions for Riley? You have time for a question or two, perhaps. I do have one for you, Riley.
Okay.
So you did talk about it being an association rather than causal, and that was one of the things that popped into my mind, because I was wondering about other factors that might come into play here, like socioeconomic factors. So somebody who maybe is not as affluent, is maybe going to have less access to dental care, is going to pay maybe less attention to dental health, as well as perhaps getting less exercise and other things that might actually contribute to cardiovascular situation. And so it sounds like they may have considered that, but I wanted to maybe ask if that was something that was addressed in the work that
Yes, they did. In the very beginning, they mentioned a few factors that they would control for, such as, like you said, physical activity. Are they a smoker? Do they drink? And things like this. But their biggest, they just touched on that briefly and just kind of moved right by it. So that was something that I also was confused about too, because they kind of said, yes, we looked at those factors, and then they never mentioned it again. Their biggest factors that they looked at was age and gender. And so they had an age, once they finished, they looked at, they had an age adjusted conclusions. And then so they would look at those different things. And so their biggest issues and that they focused on again was age and gender, but they also said they did account for physical activity, smoking, drinking, and things of that nature.
Okay. Are there any other questions for Riley? All right, well, thank you very much, Riley.
Great, thank you. Thank you, Hayden.
Okay, so next up is Ria. Did I pronounce that correctly?
Yes, you did.
Okay, my understanding is that Andre and Mary, based on a 10:20 start time for this session, may send out some kind of message for a 12-minute warning. But since we're not quite on that time schedule, if that, oh yeah, there it is right now, popping up on the screen. So, so I'll keep track of the time. I'll let you know if you start running quite a bit over. But I will go ahead and turn it over to you now.
Okay, thank you. I'm joined by my partner Harshit, so he will be pulling up our PowerPoint right now.
Can everyone see my screen? Okay, so hello, my name is Harshit Jella, and
I'm Ria Churi.
And we have made a literature review about progastrin, a potential biomarker for colorectal cancer. Colorectal cancer is the third most common cancer in both men and women. There are estimated to be around 104,000 combined cases in the United States in 2021. The graph to the right shows the estimated cases of colorectal cancer in comparison to other forms. Furthermore, cancer stem cells are accredited with being the primary drivers for resistance to cancer therapies and metastasis. The image to the right is a visual representation of how CSCs are able to lead to the recurrence of a tumor. The red cells are cancer stem cells, while the red and orange are differentiated cells. Chemotherapy impacts the predominance of the differentiated cells, but fails to eliminate CSCs, which eventually lead to recurrence and metastasis. Finally, HPG80 is the extracellular and oncogenic form of progastrin, and it's a good colorectal cancer targeting marker because it promotes the growth and survival of CSCs. Ideally, progastrin remains in the cell until it's processed into gastrin and then secreted, but in pathologic conditions, it's secreted before being fully processed. And moving on to the results of our literature study, it's been shown that progastrin is highly expressed in CSC populations in order to support the CSC growth and survival. The graphic to the right shows the staining of progastrin in a whole clone population, which has a much greater population of CSCs, in comparison to a paraclone population, which has a greater population of differentiated cells. The greater staining of the whole clone shows how CSC population and expression of progastrin go hand in hand. The bottom left image shows a qualitative measure of colonospheres in the presence and absence of progastrin. The top row shows in the presence and the bottom row shows without progastrin. As one can see, in the absence of progastrin, the growth of the spheres are severely impacted. And for a more quantitative measure, the bar graph shows how when the progastrin is depleted, the white bar, results to a much smaller percentage of spheres formed, only 50% in comparison to the control group. While when progastrin is supplemented, there's a 25% increase in the number of colonospheres. And now that it's been established that progastrin is essential for the growth and survival, the researchers looked into the biological mechanism that allows this to happen. It was found that progastrin creates a favorable growth environment for cancer stem cells through induction of JAK-STAT-mediated signaling pathways and glycolysis. The top left image shows a staining of the activation and expression of JAK2, a pathway essential to the growth and proliferation of cells. The staining of whole clones was much greater than that of the paraclones, indicating an advantage for the whole clones in terms of survival. And off to Ria.
Okay, so obviously glycolysis is crucial to fueling tumor progression and proliferation. So it's important to look at the glycolytic profile of different cells. And if you take a look at figure C here, you'll see a quantification of glycolysis in different types of cells. First, you have your black bar, which represents cells expressing progastrin. And then the middle gray bar represents progastrin depleted cells. And the white bar to the right represents cells with low glycolytic activity, which are essentially non-cancer stem cells. And if you look at the figure, you'll notice that the progastrin expressing cells, the black bars, have the highest rates of glycolysis in all areas, basal, max, and the glycolytic reserve. And glycolysis is the inefficient metabolic pathway that cancer stem cells use to generate energy to fuel their proliferation. So this increase in glycolytic activity seen in the progastrin expressing cells when compared to the progastrin depleted cells suggests that progastrin fuels CSC survival and self-renewal. And since the gray bars are lower than the black bars, it signifies that progastrin depletion reduces glycolytic activity. Okay, so now moving on to the figure on the right, we can take a closer look at the energy production in differentiated tissue versus proliferative tissue or tumors. And you'll see from the diagram that healthy cells or differentiated tissue mainly use oxidative phosphorylation. And cancer cells, on the other hand, primarily rely on aerobic glycolysis because it produces ATP at a much faster rate than oxidative phosphorylation does. And this metabolic pathway of glycolysis is faster than oxidative phosphorylation. And this rapid mechanism of instantaneous ATP generation is needed to supply these cancer cells with enough energy for proliferation. And this is because cancer is the uncontrolled growth of cells in the body. And in order to support all of these new cells and maintain this proliferation, you're going to need lots of energy. And only aerobic glycolysis provides this energy rapidly enough. And because of their heavy dependence on aerobic glycolysis, these cancer cells are classified as having a highly glycolytic profile. But the good thing is that, as we saw in figure C, progastrin downregulation or depletion inhibits the high glycolytic activity of these cancer stem cells to limit their proliferating abilities. Now moving on to the next topic, we have clinical applications. And we've seen that progastrin has the ability to serve as a very important biomarker for various types of cancers, and specifically colorectal cancer. And this allows HPG80 to be used in many potential clinical applications. One such example is the DXPG80 test from ECS progastrin. And this is an in vitro ELISA, which stands for an enzyme-linked immunosorbent assay. And this test is a sandwich ELISA. And the way that it works is that it detects and measures the concentration of HPG80 in plasma samples by sandwiching this HPG80 in between two layers of antibodies. So one will be a capture antibody and the other one will be a detection antibody. And this measurement of HPG80 aids in the early detection of colorectal cancer because increased HPG80 points to colorectal cancer. And another valuable clinical application would be the use of anti-HPG80 antibodies for HPG80 neutralization. So these antibodies will work to block or activate specific signaling pathways in order to modify the behavior of cancer stem cells. And also therapeutic antibodies have the ability to neutralize the HPG80, and this in turn decreases the frequency of cancer stem cells. And you will see this if you take a look at the figure at the bottom. You can see that there's a decrease in the formation of spheres in various cancer cell lines in the presence of HPG80 antibodies. So in this figure, the bars on the left are the control antibodies, and the ones on the right are the anti-HPG80 antibodies. So those are neutralizing the progastrin. And you'll see that the number of spheres is lower for all of these cancer cell lines when the anti-HPG80 antibodies is utilized. And this is important because it shows that cancer stem cells, it's been shown that cancer stem cells are the key drivers of tumor progression, and these CSCs have the ability to form spheres. So decreased sphere formation suggests that the HPG80 antibodies are reducing the cancer stem cell frequency and thus reducing tumor progression. Also, anti-HPG antibodies decrease cell growth and they increase chemosensitivity. So all of these factors put together greatly decrease the self-renewal abilities of these cancer stem cells, and thus they have the potential to improve the condition of cancer patients. Now going back to our clinical applications, HPG80 could also function as a useful tool for tumor localization during PET scanning, because physicians can use anti-HPG80 antibodies to locate where HPG80 is being expressed the most. And that's where the most cancer cells are located. So with this knowledge, a physician will know which locations to target. Another very valuable current application is the use of HPG80 in simple blood tests to evaluate the efficacy of surgery. So this provides useful information to both physicians and patients in follow-ups because sustained HPG80 concentrations, even after surgery, may indicate residual diseases. So this gives a physician a great tool to assess the risks of relapse and measure a patient's improvement over time. And basically, because colorectal tumors are secreting the HPG80 themselves, after surgery, after these tumors are removed, the concentration of HPG80 in the blood should be significantly reduced. But if that HPG80 concentration is sustained, even after surgery at levels that are suggestive of unhealthy patients, this allows doctors to see how effective the surgery really was and detect relapse early on. And one last important prospective application to note is the production of an HPG80 rapid test. So due to the inexpensive and relatively non-invasive nature of HBG80 detection in the blood, rapid tests are a potential future implementation. And after some future research, it's very probable that these rapid tests will be available for physicians to utilize. And this provides us with an innovative tool to easily measure HBG80 in the blood and fight colorectal cancer together. And to wrap it up, colorectal cancer is an important issue that needs to be addressed. But luckily, 68% of colorectal cancer deaths can be avoided with effective screening measures. So thus, it's essential to target cancer stem cells to develop effective therapeutic strategies. And future research on the roles of progastrin in colorectal cancer stem cells is needed to drive the development of HBG80 diagnostic and therapeutic tools used as a standard of care. We hope that this presentation has been very informative and interesting, and we thank you very much for listening. Thank you.
Thank you, Ria. And Harshit, did I pronounce that correctly?
Yeah, that's correct.
Sorry, I didn't realize there were two presenters for this one, but does anybody have any questions? Okay, well, I have one or two questions for you. First, I was wondering, is there a normal biological function for progastrin? Is that secreted as part of any normal biological function? And if so, I wondered about how the potential for an antibody therapy causing unintended side effects.
So the gas gene is expressed in order to secrete the stomach acid that we produce. And progastrin is a precursor to gastrin. And the thing is I'll show you right here. If you look at the bottom right image, it shows like progastrin is normally expressed, but the difference between an oncogenic or like a pathologic or a non-pathologic thing is that it's not secreted directly into the bloodstream. It's usually processed into gastrin, and the gastrin is what is secreted into the bloodstream in order to stimulate the cells of the stomach.
Right, and so I was just curious though if progastrin is ever secreted for some biological purpose. And so I'm familiar, for example, with molecules that are called neurotrophins, which can be secreted in either a processed or an unprocessed form, and they elicit different biological responses. And so I was just wondering if the same is potentially true of progastrin, and if it was, then using antibody therapy could have some potentially serious side effects.
To our knowledge, progastrin does not serve a much greater purpose other than like as an incomplete form of gastrin itself.
Okay.
But it does have that proliferative qualities that when secreted, it leads to like an autocrine signaling pathway.
Yeah. Okay. Progastrin is necessary for the processing of gastrin, but when it's released in its unprocessed form, that's when it can lead to this proliferation and tumorigenesis.
I did have one other quick question for you, perhaps. So you were talking about how the proliferative cells prefer to use aerobic glycolysis as opposed to oxidative phosphorylation because it generates ATP faster. I'm just curious about that because while it may generate ATP faster, it generates a heck of a lot less ATP than oxidative phosphorylation. Sure. And considering how much energy a dividing cell actually needs, I was just curious about that as to I don't know, documentation about that, or just it just struck me as a little bit interesting that the cells would prefer that pathway.
Yeah, when we were reading the paper, we found that kind of interesting too. But I think these cancer stem cells are kind of just taking like the easy pathway. They just want that, ATP super quickly so that they can proliferate. But it is smaller amounts, but it's much more rapidly. So I think that's like a trade-off that these cancer stem cells are making.
Okay.
Yeah.
All right, well, thank you both.
Thank you for your time.
So that brings us to our third presenter, Dulce Cafatelli. And so I will go ahead and turn it over to you, Dulce.
Okay. Can you see my presentation? Okay, my name is Dulce Cifetelli. I'm a grad student from Flint, and I'm going to, my research is to investigation of furin and PACE4 mediated cancer metastasis under hypoxia and hypothermia in breast cancer cells. Hold on. So what we know about cancer? So we know that cancer cells arise from a normal cell due to DNA damage. And this DNA damage is due to exposure to hazardous environmental such UV radiation, asbestos. Also, DNA damage can become from viruses, herpes, human papilloma, and other viruses. But especially DNA can be damaged by genetic mutation, especially in proto-oncogenes, tumor suppressor genes are two genes very important. I'm having trouble with the computer. Okay. Cancer is often mistaken to be a single disease, but however, in reality, there are numerous types of cancer. These cancers have common attributes and they have great difference, but however, they have two unique characteristics, uncontrolled cell division and metastasis. So we're gonna talk a bit about metastasis. Cancer metastasis is the spread of cancer cells from the primary tumor, and these cells move from the primary tumor to and make a secondary tumor. And this is what it make a tumor malignant. And this cancer metastasis is what kill the majority of people that is diagnosed with cancer. Approximately 90% die because of cancer metastasis. So an important step in metastasis is invasion. So cells go through a process known as epithelium mesenchymal transition, also known as EMT. This involves the breakdown of adhesion molecule, cell detach and gain motility, and they go and invade nearby tissue. So these motile cells secrete enzymes that degrade the extracellular matrix. Extracellular matrix is what provides support to the cells and is made up of proteoglycans and fibrous protein. So protein involved in invasion, we have two important protein families, protein convertase and matrix degrading metalloproteinases, furin, PC, for example, activate pro-MP14, and this lead to a cascade of activation of other metalloproteinases, leading to the activation of MP9. And MP9 is the one that degrades the extracellular matrix, leading to metastasis. In normal condition, PACE4 inhibit MP9. So the objectives in this investigation is to determine if a stress condition such as hypoxia and hypothermia, which is present in tumor microenvironment, are the triggers that lead cancer cells to invade. So for us to test this hypothesis, we expose breast cancer cell to stress conditions and examine this cell for the expression of key proteins responsible for invasion. These key proteins are the metalloproteinases and the PCs. So we use MCF7 and MDA and B468A. These are cancer cell lines. We culture this cancer cell into the culture, and then we transfer them to a 96 well plate where a spheroid will form. Then to analyze our protein, we use Western blot. We use gel electrophoresis with resolved protein by their molecular way, and we use primary and secondary antibodies to get the protein of interest. So this invasion assay is done after spheroid formation, and we add the invasion assay and we incubate the spheroid for 96 hours under hypoxia and normoxia conditions. So I just want to let you guys know this is an ongoing experiment, and it's a lot of things going on now, but as of now, this is what we have so far. So most cancer research utilize 2D culture, but this culture has limitation. They don't represent cell-to-cell interaction. The cell is just growing like a flat surface, and the behavior of the cells is tied to the surrounding. So if they are more cell-to-cell tie or cell to extracellular matrix, so they behave different. And 3D culture provide a more representative environment to in vivo. So here we can see 3D, how 3D looks like, and every well, it contains a spheroid. I will show you a picture of that in a minute. So this is how we culture 3D. So this is a Western blot, measure the protein level for MB468 cell growing 3D and 2D. So here we have this band. We use beta-actin as a control. We can see furin is increased as a stress condition, which is heat, hypoxia, and dual. Dual is heat and hypoxia combined. MP9 is also increased. And whereas PACE4, we can see that in normal condition is higher than when it's in stress condition. So for us to quantify this data, we utilize ImageJade, and they give us the levels of this protein. For example, furin levels in normal condition is here. And when we compare it to stress condition, we can see that furin level is increased. MP9 level also, you can see that is increased when cells are exposed to stress condition, especially dual. So MP9 is the major affected here. We got more. And here we have the level of PACE4. Normally here is the control in normal condition, is higher, where if we go to stress condition, we can see that they decrease. So here is a graph that we compare the furin, MP9, and PACE4. As we can see, normally, and with heat, hypoxia, and dual, we can see that the different amount of MP9 is the most affected. And we can see the PACE4 level decreasing. So this is the comparison between furin, MP9, and PACE4. So here is where I was talking about. We successfully grow spheroid in 3D. So in day 0, we start with a single cell. And by day 1, we get like a conglomeration of cell. And by day 4, we have a fully formed spheroid, which is very nice. And that's what was in those little wells that I showed. So, like I say, this is ongoing investigation. We have a lot of work to do, and we need to do more quantification in invasion assay quantifications. So, in conclusion, furin and MP9 are increasing under stress conditions compared to normal conditions, whereas PACE4 is decreased under stress conditions. This suggests that furin and MP9 are important for cell invasions. Our results are consistent with our hypothesis and also consistent with current literature. Like I say, it's an ongoing experiment, and we need to quantify the invasiveness of these cells. So our ultimate and desirable outcome of this research will be a better understanding of metastasis and discover of new biomarker for metastasis and prevention of metastasis. So that my reference, and that is the end.
Thank you, Dulce. Does anybody have any questions for Dulce? Because you're leaving me to handle all of the questions.
Yeah.
Well, I'll ask you one, Dulce. So if you were going to try to do some sort of therapeutic intervention for metastatic cell behavior, based on the results that you have thus far, what might you try to target?
Maybe MP9, because it's the one who shows more increase and is the one who degrades the extracellular matrix. So I believe that that will be my first approach.
Okay.
I have a question. So you mentioned PACE4 was down regulated, right?
Mm-hmm.
Did you also mention furin? Did you see it either up or down? Okay. Oh, looks like up.
Yeah.
Regulated. Do you know of like the interaction between PACE4, furin, and MP9?
Like PACE4 inhibits MP9, and according to literature, PACE4, pro-segment of PACE4 inhibits feuding. It's a connection there, but I really don't understand well how that works. And I know it's, you know, I don't know how to use this, but it's one go one way and the other one go the other way. But it's a connection there, but to tell you, I really can get it yet. So
I can add a little bit to that, Hayden. So the literature is a little bit conflicted over the role of PACE4. There are some papers that suggest PACE4 is upregulated in metastatic cancers, but particularly in breast cancer and ovarian cancer, there's information in literature that suggests the opposite is true, that it's actually downregulated. And it's been suggested that it actually inhibits MMP9, which is key for degradation of the extracellular matrix. And so in that circumstance, it would make sense that PACE4 would be downregulated, which is not exactly what is being shown in the data.
Thank you.
All right, thank you, Dulce. We'll now move on to our final presentation then, which is going to be from Hayden Norris.
There we go. Sorry. So brain-derived neurotrophic factor, Val66 Met polymorphism, fear of falling, and fast gait speed in community-dwelling older adults. So I'll be the one presenting today, Jonathan Garber. I listed him as like a second author on this. He helped with a lot of the data and like the actual research. He did everything that I did that I'm presenting here, is to get a second group of results to sort of corroborate what I got. Dr. Alan Goldberg, he helped a lot with the data analysis, and Dr. Susik helped us a lot with this too. So this is my abstract. It's a giant wall of text. I'm not going to read all of it for you for the sake of your time and sanity, but a few things I want to point out really quickly in this. So we found in association in the interactive effect between the Val-Vel polymorphism and the fear of falling and the fast gait speed. But I should note that the Val-Vel polymorphism was divided into two groups, for one being Val-Vel homozygous, and then the other group being like not Val-Vel, so either the heterozygous or homozygous met alleles. So and that's just for sake of getting enough data, because the met allele is relatively rare compared to the Val allele. So and then I'll talk a little bit more about that interactive effect between the Val polymorphism and fear of falling in a bit. But what we found is that in the non-Val-Vel subjects, there had a significantly lower fast gait speed compared to the Val-Vel subjects in the group that had a fear of falling. versus in the group that had no fear of falling, there was no statistical difference between their average fast gait speed. So the subjects we're working with are community dwelling older adults. That basically means anyone 65 years or older and independently living, so they're not institutionalized. The reason we're studying this is because certain genotypes may have an impact on physical ability, especially in older age, and may help to identify potential physical therapy targets. If we can better identify physical therapy targets, we could potentially prevent accidents that could lead to either like breaking a hip or general improvement and preventing a lack of mobility and generally improving quality of life. So that's generally why we choose to research this. And BDNF isn't the only gene we're looking at. We're looking at 2 other genes, but for this presentation, we'll just be going over the BDNF results we have. So a little bit about BDNF. Stands for brain-derived neurotrophic factor. And it is responsible for influencing cell growth, differentiation, and survival, especially in neurons. It is also found in human muscle, skeletal muscle, but mainly it's best well known for its role in neurons. So here I have a little bit of a diagram. It binds to tyrosine receptor kinase B, and then can activate a couple different pathways from there that all play pretty important roles in cell growth and survival and differentiation. So a little bit about the BDNF genotypes and phenotypic effects. So it plays an important role in regulatory function during myogenic differentiation. It plays a role in maintaining the population of muscle progenitors in adult muscle. And a little bit about the polymorphisms. Met subjects perform worse on average compared to VALO subjects in memory tests. So those are a couple of reasons why we consider this gene as far as why it may have an impact on fall risk and physical performance in the community dwelling older adults. So in Val66 MET polymorphism, the 66th amino acid is converted from like a valine to methionine, and that results in not being able to be properly sorted by the Golgi into secretory granules, and that in turn impacts secretion. And I have a little bit. So the top row of A is the valine version of the BDNF gene, and as you can see, it's more localized to those little dots, which are the secretory granules, versus the bottom row of the figure A. You see the methionine BDNF allele, and it's not nearly as localized, and it's sort of free floating around in the cytoplasm. And then in figure B, they sort of quantify that. And so this can impact secretion and have different phenotypic effects, as I mentioned before. So for our project, what we did is there were 45 of these community dwelling older adults. We collected saliva samples for DNA extraction, and we also at the same time collected physical performance data, which I'll show you in a minute. And then we used a statistical analysis to look for correlations between that data. So for the extractions, we used the Origin Discover 500 kit and protocol from DNA Genotech. And then the DNA was extracted from saliva using the protocol materials from the Origin Prep-It DNA extraction kit from DNA Genotech, and then stored it 4C. So reagent concentrations and volumes and primer sequences, as well as the thermocyclic parameters, were from USUP I'm gonna say et al. So after the PCR, we used a restriction digest. We used NLA3 endonuclease to digest after the PCR. And so a little bit about like what that does. That is able to cleave the val allele of the gene, but not the, or sorry, the other way around. It's not able to cleave the val allele of the gene, but it is able to cleave the met allele. So you see a higher molecular weight at the top. Those are all val, and then if there's 2 at the bottom, that's because the restriction, the NLA3 endonuclease was able to cleave that segment, and it was broken into 2 pieces. And then anywhere you see both a top val allele and a bottom 2 met, that's a heterozygous individual. So that's how we genotyped all of the DNA samples. So I'm going to briefly just go through this really quickly and give you a few seconds to look at these. These are the physical performance tests that were collected, and from all of our subjects. I just have a whole wall of text here, but so there were a lot of physical strength and walking tests. And then we did find, again, a statistical significant difference in the FIRA of falling group between the val-val and non-val-val subjects. And then I have a graph here that shows the difference in the mean best fast gait speed in the FIRA falling group in meters per second. And as you can see, it's significantly faster in the val-val group. And then limitations of the study, it can only show correlation for now. Then there have been some general complications with getting new samples in the pandemic, and so we're just now starting to get new samples again, and it's sort of a slow process, but we intend soon to increase the number of samples we get and analyze. And then because we only have a total of 45 samples, it's fairly limited in statistical power. So we hope to improve that by getting more subjects and data to work with. And then I have my references. And that's it.
Thank you, Hayden. Does anybody have any questions for Hayden?
I do.
Go ahead, Dulce.
Which is better to have the val allele or the M?
The val. Typically we see lower performance in the met allele, and but better performance in the val allele. So that's kind of what we're seeing here, and that's pretty typical of these studies that examine the val 66 met polymorphism. Is that it makes the MET polymorphism makes BDNF not secreted as effectively, and that could have detrimental effects usually. Not just here, but like with memory, and there are studies that have been done on cognitive performance, and they seem to agree with that, as generally the MET allele is detrimental.
Thank you.
But I should note that we did not see any difference between the val and the val and non-val subjects in the non-fear of falling group. So it may not make as a big difference there, but we're still getting new samples, so maybe that we'll see a change there, but so far no.
Okay, I just saw the message from Mary that we are supposed to be heading back into the main room. So thank you very much, Hayden. Thank all of you for your presentations. Very interesting. Nice to see all of you this morning. And so I guess we will go ahead and go back into the main room now.
All right, welcome everybody to the 2022 Flint Student Research Conference and Breakout Room 8, Social and Behavioral Examinations. We're going to have a few presentations this morning that all have kind of a theme as determined by the organizers of having some connection to social or behavioral traits in one way or another. So my name is Ron Stamper. I'm a professor of chemistry at Mott Community College. And I'm going to be the moderator today. So if you have any questions, please let me know in the chat, or unmute yourself after the presentations are finished and you can ask those questions. Okay. Let me share my screen for our first presentation. Okay. Can you all see my very busy screen right now? Yes, okay. So our first presenter today is here, Danielle Jarrett from Mott Community College, talking about how honey can change a houseplant's life. So Danny, go ahead and take it away. You should be able to unmute yourself.
Thank you. My name is Danny Jarrett. I am a student at Mott Community College. My major right now is botanical sciences, and this is my proposal on how honey can change a houseplant's life. Next slide, please, Dr. Stamper. So I actually learned this idea from my father. He took up a keen interest in botany when I was a senior in high school. And he's always shared these different tips, whether it be botany, like housework related. And he had briefly mentioned the ideas to me that he was coming up with and how he wasn't really like seeing it anywhere else, but I didn't think anything of it at the time. I was more focused on graduating and getting into school and furthering my future. But last year in a STEM 199 class, we had the opportunity to do a research project. And so I asked him about the tips that he was giving me a few years back. And he told me about how honey has really helped his plants grow. And so I did some research on that, and I wasn't able to find anything. And so I decided to put it out there myself. Next slide, please. So the proposal that I have, it's a rationale, significance, and my experimental aim. The rationale being that honey just contains the vital nutrients and minerals required for plant growth and development, and then in turn could be used in replace of synthetic fertilizers, but that's way down the line. So these vitamins and nutrients that it contains, it's very abundant in the ones that plants require the most, or at least very many general house plants. And the significance I see as like a large scale for agricultural use. It can be used to further preserve and protect the honeybee species, and then in turn pushing us into a greener future so we're not making as many synthetic fertilizers. And my goal is to just show that honey is an appropriate and abundant nutrient for these houseplants, and hopefully furthering my research to compare it to the use of synthetic fertilizers. Next slide, please. And this is my plan and design. Next slide, please. So I had originally 4 English ivy plants, and I divided them into a control and experimental group. After dividing them into that, I identified and labeled 4 major vines on each of the plants. And after identifying the vines, I recorded each initial height. And according to which group the plants were in, whether it was control or experimental, I fed them plain water or honey water, which was just like a teaspoon of honey mixed into a spray bottle, and I misted them accordingly. They were all placed under a grow light. And over a 20-day period, I recorded any changes in growth, color, and perkiness. Next slide, please. And so this is just how I identified what was a major vine. It kind of looks like a tree branch. And then ensuring that I was labeling these vines and measuring them it showed me that I was keeping an average track of the overall plant's growth and not keeping track of just the leaves that were coming off of it. And when I labeled them, I just took pieces of sticky notes and wrapped it around the vine so that way I wasn't harming the plant or preventing the growth in any way. Next slide, please. This is my control groups data. So I divided the tables by the vine number, the initial height, the height after the 20-day period, and then the recorded growth between the 2 days that I recorded. And I was able to get a recorded growth out of all of the vines, which was good news. So that way I was able to show that there was a general trend in growing for even the control group. Next slide, please. Same thing with my experimental group data, divided by the vine number, the initial height, and then after the 20-day period in my recorded growth. And I know it's a lot to look at, but even now we can see that the recorded growth on my experimental group is generally like a larger gap than it was with the control group. Next slide, please. And instead of going back and forth between the slides to make everybody look at the tables, I put it on a bar graph for you guys. And this is just the average growth taken between the vines on all 4 of the plants, put next to each other. And some of them were pretty stable. They didn't grow very much, but overall we can see that the experimental group did significantly grow. Next slide, please. Unfortunately, I did run into quite a bit of challenges that I wasn't prepared for. So being that honey has no natural insect repellent and it actually attracts them, my honey-fed plants attracted a spider mite infestation, which is just a very common mite for houseplants. Any houseplant can get it. I was not able to save them. But despite the infestation, all 4 of my plants had actually begun to wilt and outgrow their designated pots, which surprised me. They were no longer perky, they were drooping over the pots. And these changes were unexpected, mainly because they happened very quickly. It was just after 2 weeks of experimenting where they were too big and then they were infested. And I was not able to save them because the care and the recovery that was required to get rid of the spider mites would have skewed my results. Next slide, please. And so overall, my initial 20-day period of growth, all 4 of my plants did grow. And I was able to show that my experimental group had a higher frequency of large-scale growth based on the labeled vines. And unfortunately, I did have to cease the study. As said, the recovery and care would have skewed my results for both my control and experimental regardless. Next slide, please. And then my recovery proposal. I do want to restart my research in whole to record the changes in growth and development only influenced by the addition of honey into an English ivy's plant. And I am hoping to have a more controlled and stable environment to prevent infestation and overgrowing, maybe overwatering. And so I definitely have some things that I need to tweak, but I would like to further my research and restart it. And that's all I have. Thank you.
All right, thank you, Danny. We'll open it up for questions. Anybody has them?
I do.
Go ahead, Laura.
I was just curious, despite the spider mite infestation, do you still think that there is application, like if preventative measures are taken or larger pots are used to implement using the honey in plant growth?
Like, are you asking like how to prevent the spider mite infestation?
Well, I guess my question is more, do you think that it is feasible to, like, is this still viable as a potential use of plant growth despite the spider mite infestation threat? Like, is it something that can be fairly easily prevented if it is, you know, something that is taken note of early on?
Oh, for sure. Spider mites, they're really easy to get rid of. They come as easily as they go. The only thing, the reason why I wasn't able to save them is I noticed too late. All of my other houseplants too were infested. And I did the study just because it was for educational purposes and not just like caring for houseplants in general, but it is very easy to get rid of them. And so using honey as like a house care plant additive, it may attract them in the beginning, but obviously like if I'm advertising this, you know, let's say like several years down the road, I'm assuming hopefully future Danny will come up with something to be like, this is what you do before the spider mites come. And if they do come, this is what you do after.
Okay, thank you.
Mm-hmm.
I have a question.
Corey, go ahead.
Danny, what specific kind of honey did you use? Was this like local honey from by where you lived, or was it just regular supermarket honey?
My dad uses like local honey, but that's very expensive. And so I was just using like a store brand, like Meyer honey, nothing special, but it contains all the same nutrients as any other honey. So,
of course, awesome, thank you.
Mm-hmm.
Danny, I actually had a question too, if I could, and only because I don't have a green thumb like in my entire family tree. So would you expect the same sorts of results if you tried this with a different plant, or is there something special about ivy that would make it maybe more susceptible or more responsive to the honey?
I chose ivy plants because they grow very fast. So I knew that I was going to get data more quickly, as if I chose like a succulent, I probably wouldn't see results for like over a year. Mm-hmm. And that data would be probably more gradual and steady. But I needed to show that, because I had figured that with the school year ending, I needed a plant that was going to grow and adapt as quickly as I wanted to record my data. But it's like a pro and a con because they grow so fast. They're also very susceptible to outgrowing and overwatering and underwatering. And so they're really easy to take care of if you really know what you're doing, but it's like one wrong turn can kind of put the whole plant underground.
Gotcha. Thank you.
Mm-hmm.
Laura, did I see you unmute there? Do you have another question? No. Okay. All right. Well, thank you, Danny.
Thank you.
I'm going to stop this. And we're a couple of minutes early here, but since we have, at the present moment, our last presenter is not in the room, I'm going to suggest that we go ahead and move into our next presentation, albeit a couple minutes early, and probably our Zoom timing is going to be off. So that if you all choose to go to a different room for the fourth presentation slot, that will be on time to do that. So unless there are any objections, we'll go ahead and move on to our second presentation here, which is right there. Yes, can we see that okay? This is, Corey, it occurred to me I've never pronounced your last name. So Corey Martirana.
Yeah,
Yes,
that's
okay.
correct, actually.
I don't know how many classes we've had together, but we've never said your last name out loud. So this is Corey from Mott Community College talking about how COVID-19 has impacted our carbon footprint. Corey, take it away.
Hello, everybody. My name is Corey Martirana, and this is actually just my research proposal for, so it's just theoretical research. I'm a student at Mott Community College. I will be graduating in the spring and continue on majoring in environmental science and sustainability. Okay, next slide, please. So what is a carbon footprint? It's the amount of carbon emitted into the atmosphere, which is the total sum of greenhouse gases produced by an individual, organization, or event. Greenhouse gases include carbon dioxide, methane, nitrous oxide, and water vapor. These occur naturally through events such as outgassing from oceans, decomposing vegetation and other biomass, even volcanoes, wildfires, and gases from animals. Most natural causes, however, are offset by photosynthesis and other organic processes. Synthetically, however, humans are responsible for a lot of the carbon dioxide in the atmosphere due to the burning of fossil fuels. And just a fact, according to the EPA, the total US emissions for 2019 was 6,558 million metric tons of CO2. Next slide, please. And so why do we care? Well, projections highlight devastation if emissions proceed as usual, catastrophic worldwide events linked to rising annual global temperatures, extreme weather conditions and rising temperatures, wildfires, drought, biome shifts, rising sea levels and flooding, ocean temperatures, acidity, and oxygen levels would also be affected, melting permafrost and the loss of fresh water supply. The amount of CO2 has increased more than 20% in the last 40 years, owing largely to human activities and representing over 50% of total increase in atmospheric carbon emissions in the last, since the onset of the Industrial Revolution. CO2 trapped in the atmosphere creates a greenhouse effect, keeping heat on the planet. As pictured in the graph on the left there, the CO2 levels have increased a significant amount over the last 800,000 years, especially most recently to the present. The representation of sea level rise in the next graph also shows a steep increase in only 25 years' time. These graphs depict a sort of cause and effect relationship between the two. Also, actions have been taken globally to sort of slow the amount of damage. And these are seen in the Kyoto Protocol, which was adopted in December of 1997. And this treaty was signed by more than 150 countries, pledging to lower their amount of greenhouse emissions and gases produced. Similarly, the Paris Agreement was also signed in 2015 and committed 193 parties to limit the global temperature increase to less than 2 degrees Celsius, and also by lowering emissions. And most recently, President Biden has announced his administration's goals for reducing the country's greenhouse gas pollution by 50% below 2005 levels by the year 2030. Next slide, please. So my research proposal is developing research to find possible positives to environmental impacts of lockdowns and closures. This is a socio-environmental study and literature review mostly. I'm concerned with, has there been less travel, more virtual work from home atmospheres? Are we increasing our online shopping, which is leading to ride sharing, which is lessening carbon emissions because your order shares a ride with someone else? And so the picture that I've included here, the representation basically implies that in order to follow the Paris Agreement and keep global temperature increase under 2 degrees Celsius, we have to mimic the reduced carbon output of the year 2020 consistently over the next 10 years. Next slide, please. So research has shown vital influence all over the world, even in cases of higher education. When I began this research idea winter semester of 2020, there existed little to no data of this being studied at the moment. Since then, a lot has changed with the pandemic and information produced regarding the amount of carbon emissions. Here is some existing data research that support my project plan. Next slide. So this is Bournemouth University in the UK, and they did a comparative analysis of the carbon footprint. And so they showed a significant decrease in carbon footprint from April to June in 2020 as compared to previous years. The color-coded bars mostly account from study, work from home, and they're barely existent in the previous bars of 2018 and 2019. It's argued that teaching and learning fully online would actually help the climate crisis. Next slide, please. In Italy, their findings indicated that the carbon footprint during lockdown was about 20% lower than the average carbon footprint calculated for the past. And 2020 is represented by the striped figure located at the bottom of the graph there. And it's represented before and during the lockdown phase. So you can see that the shaded gray lockdown phase is significantly lower than that of the years previous. Next slide, please. So for my example, how would this be measured and demonstrated in a research method comparatively? In my case, I was going to conduct a survey, and it would be conducted differently than most of the other research that I found, because I chose to make my methods less invasive, because I figured in order to get a larger reply from people, I would have more quantity of replies if I wasn't digging into their information. So my intended target audience in my case was actually going to be the students and the faculty of Mott Community College, had I been able to go through with the actual research. And the demographics that I chose to include were going to be race, age, zip code, gender, political party affiliation, and occupation. And each question must be answered, but there was a choice for each question to choose not to answer that question. The user would attach a screenshot of their before and during pandemic carbon calculator totals along with this survey with a simple upload. And furthermore, the questionnaire will be completely anonymous for protection of privacy for the user. But at this moment, of course, my research is still theoretical. So next slide, please. So how is carbon measured? The easiest way to measure the carbon would be a carbon footprint calculator. They're all over the place, but So the individual will complete the carbon footprint calculator 2 times for their household to their best ability. The first time would be pre-pandemic, business as usual, everyday life. And then they would complete it a second time for during the pandemic when there were stay-at-home orders and quarantines. Once the amount was generated in tons, you would just take a screenshot of your total and upload it to the survey twice each time. And the link is provided below for the actual calculator that I would be using. And it's very quick and easy and takes no time at all to complete. Next slide, please. So this here is just a screen snip of what the calculator looks like and the sort of questions that it would ask. And over to the right, it would generate the your emissions in tons. And that would be uploaded into the survey as well as, or sorry, the screenshot on the right there is what would be actually uploaded into the survey before and during the pandemic. Next slide, please. So next would be to aggregate the data. So I would dig through the data to make accurate and possible correlations between the demographics and carbon use. So the secondary goal of the study would be to find the correlations between the two, such as age and carbon use, or zip code and carbon use, and so on. How do they relate? Discover connections. Is there anything else useful to be inferred from the data? What else can this show besides actual tons of carbon? I believe there's more to be found once the data is compiled. Next slide, please. So the question is, are there any positives to be found from the pandemic? Was carbon output less? Was it more? Was there no significant change? I believe that studies like this can lead to larger scale research involving more complete work and insights. So regarding data such as pollution, and that can be air, land, and noise pollution, or maybe particulate matter. But with everything, there's also negatives. So are there more negatives involved? The increased production of a new vaccine and the transportation required to produce and send worldwide, or the need for disposable masks and what that does to the environment, both in carbon output and also waste, along with other medical waste needed during this time. Ideally, I would have loved to dig much deeper in this and be more intrusive and add more questions, specifics for my survey. Given the chance in the future, I would love to navigate a full-length study of this. I'd like to have personal access to more answers regarding other variables such as electricity use or internet use, and maybe what buildings are saving on utilities during this time, just to be a lot more open with it. Next slide, please. So in summary, humankind has made such a dreadful impact on the environment due to the steep increase in greenhouse gases, mainly CO2. The simple survey will help start the conversation about the trials of climate change and the need to act now and act fast. I believe that the adaptability and reproducibility of this project seems to prove itself useful in various settings and can do considerable good. In such unprecedented times, science can find connections that allow for corrections for the future. It's important to begin the end of the climate crisis. Awareness leads to impact, and impact leads to change. Next slide. My resources. So thank you so much all for your time and consideration. I'd also like to thank my STEM 199 professors for allowing me the platform for undergraduate research, along with all of their help. And are there any questions?
Thank you, Corey. Go ahead. Yeah, Danny, go ahead.
Corey,
everybody's got questions. This is good. Danny, go ahead first.
How did you like figure out that this is what you wanted to study, like for your research project? Because this is a very in-depth, there's a lot of information to this. It's very, very in-depth. I just wanted to know how you like stumbled upon it and made the connection between quarantine and the environmental impact of it?
Yeah, so I had a lot of trouble coming up with my research topic for the class, and I just started thinking that personally myself, I was not going anywhere at the time. I stayed home, my work was closed, school moved completely to online, and I just sort of figured that if that was the case for me, it had to be the case for so many people, especially since all of classes moved to online. And I just figured, you know, everything was so bad and everything was so negative and horrible. Like, could there be a positive outlook, as in less carbon emissions? It's, you know, it's a small, minuscule thing to how horrible the pandemic was, but, you know, could we just find a little slice of something good out of it? And at the time, during that class, I really couldn't find it being studied anywhere. I couldn't find much information on it at all. So I thought this would be something to start. And now, of course, there is a lot of research on it now, but at the time, there wasn't a lot out there. So that's how I decided. I can't hear you, Dr. Stamper.
Sorry,
Okay.
Brittany, I think you were next up for a question.
Yeah, so I was curious to know, with measuring the carbon footprint in your, or not program, but in your research, how, is it an app that you utilize on your phone, or is it like a device that you purchase, and would it be given to participants, or would they have to purchase it themselves?
Sure. So the way that I was going about it was pretty simple and straightforward. If I had the opportunity, I'd like to do something more invasive. But so a carbon footprint calculator is just an online website. So if you just go to like conservation.org, which was the one I was using, it's completely free, and it just sort of asks some questions like, what type of light bulbs do you use in your house? How many miles do you drive a day? What is your mile per gallon in your car? And the website actually will just generate a physical amount of tons that you would produce by that lifestyle. So the idea is that you would complete it in your normal life, like driving to work and school every day, and then you would get a generated tons amount. And then after that, you would do it a second time of like, okay, I'm not going anywhere. I'm not traveling. I'm not going on vacation or going to school or work. And that would also generate another ton. And theoretically, we'd like to see less in the later amount than the previous amount. So it's just a free, accessible website that would just be used. Thank you.
All right. Thank you, Corey. Let's go ahead and move on to our third presentation. So over here. So we've got Brittany Jones speaking to us from University of Michigan-Flint, talking about COVID-19 beliefs and behaviors among healthcare workers. Brittany, the floor is yours.
All right, so hello everyone. My name is Brittany, and I am a Master of Public Health student at the University of Michigan-Flint. And I will be going over COVID-19 beliefs and behaviors among healthcare workers today. Next slide, please. So here's some background on vaccine hesitancy and COVID-19. Vaccine hesitancy refers to the delay in acceptance or refusal of vaccines despite availability of vaccine services. Vaccine hesitancy is not new. The World Health Organization identified vaccine hesitancy as a leading global health threat. Total COVID-19 hospitalization admissions are a little over 4.5 million. The average cost of hospital care for COVID-19 patients without insurance or who receive out-of-network care varies greatly by age, from about 51 grand for patients between 21 and 40 years of age to 78,000 for patients between 41 and 60 years of age. According to the updated cost analysis data from Fair Health. Prevalence of post-COVID conditions has been reported among persons who have had COVID-19. The range is about 5% to 80%. with differences possibly related to different study populations, case definitions, and data sources. With the increase in availability of COVID-19 vaccines, how vaccination affects post-COVID conditions still remains unclear. And the death total, and this is a pretty up-to-date number, is 963,244. Getting vaccinated against COVID-19 helps protect people from getting sick or severely ill with COVID-19 and can also help protect the people around them. CDC continues to monitor how well the vaccines are working. Clinical trials and observational studies show that all COVID-19 vaccines authorized in the United States are effective at preventing COVID-19, especially severe disease, hospitalization, and death. Next slide, please. All right, so vaccine hesitancy among nurses can increase risk of COVID-19 infection within work and community settings, as well as reduce the available healthcare workforce. Nurses are a trustworthy and credible source of vaccine-related health information. Vaccine hesitancy among nurses can negatively influence patient confidence in COVID-19 vaccination. Next slide. And the purpose of my research is to identify factors related to COVID-19 vaccine hesitancy among nursing students attending the University of Michigan-Flint. Slide. And then our methodology is, this is a cross-sectional quantitative research study. It was a 35-item online survey that we conducted, and it covered the topics of demographics, personal health status, employment status, and work conditions, COVID-19 knowledge and main sources of COVID-19 information, and vaccination behaviors, including the flu, as well as COVID-19. Next slide, please. So the study participants, nursing students were recruited by nursing faculty through classroom announcements and email. The participation criteria, you had to be a University of Michigan-Flint student, register in your RN to BSN program, and we received a total of 230 surveys, but after removing incomplete surveys, the final sample size was 194. Next slide, please. And for our statistical analysis, variables examined included race, ethnicity, political and religious affiliations, clinical working conditions, readiness for vaccination, and reasons for vaccine hesitancy. Descriptive statistics were conducted and analyzed to identify factors related to COVID-19 vaccine hesitancy. The frequencies are reported for dichotomous and categorical variables such as respondent sex, race / ethnicity, and political and religious affiliations. Means and standard deviations are also reported for continuous variables such as the respondent's age. Next slide, please. So now we're going to get into some of the results. This is our demographic characteristics of study sample. As you can see here, there's different percentages given. We had 87% of nursing students that were vaccinated, and then we had 12% that were not. The majority of the unvaccinated respondents were white at 63%, female 96%, Christian at 63%, and reported to have other political affiliation, and that was at 63%. Next slide, please. Oh, I should have told you to hit those. Sorry about that. All right, so for our clinical working conditions, most unvaccinated respondents reported providing direct care to COVID-19 patients, and that was at 81%, and that'll be the first under, or the fourth line under unvaccinated. Yes, thank you. But strongly disagreed at 62% that their employer provided adequate personal protective equipment. Less than half, at 48% of unvaccinated respondents strongly agreed that their employer had sufficient infection control. And then our next slide, please. Despite this, 71% of unvaccinated respondents reported that they have no intention of receiving the vaccination within the next 6 months. All right, next slide, please. And this is our data on reasons for vaccine hesitancy. So as you can see here, the unvaccinated portion participants believe that they already have protective antibodies, so they don't want to receive the vaccination because of this, or concerned about speed of vaccine development. Also, they're concerned about adverse side effects, and that one's sitting at 58%. Right. Next slide, please. Thank you. So our research limitations, we had a small sample of unvaccinated persons, and I think that's I think it's largely due to because it's required to be vaccinated on campus. So there was a small sample of unvaccinated persons, limited ability to conduct more complex analysis, such as identifying statistical significant differences between vaccinated and unvaccinated persons, and identification of the strongest predictors of vaccine hesitancy, response bias due to social desirability, highlights the complexity of vaccine hesitancy, that's our strength, and provides insight on how best to address vaccine hesitancy among this sample, which is a strength. Next slide, please. Okay, so we took a look at the trans-theoretical model for stages of change, which first here we have the pre-contemplation stage, which is people do not intend to take action in foreseeable future, defined as within the next 6 months. People are often unaware that their behavior is problematic or produces negative consequences. People in this stage often underestimate the pros of changing behavior and place too much emphasis on the cons of changing behavior. For the contemplation stage, in this stage, people are intending to start the healthy behavior in the foreseeable future. That's defined as within the next 6 months. People recognize that their behavior may be problematic, and a more thoughtful and practical consideration of the pros and cons of changing the behavior takes place, with equal emphasis placed on both. And then we have the preparation, which is also known as the determination stage. People are ready to take action within the next 30 days. People start to take small steps toward the behavior change, and they believe changing their behavior can lead to a healthier life. And then we have the action stage. In this stage, people have recently changed their behavior, defined as within the last 6 months, and intends to keep moving forward with the behavior change. People may exhibit this by modifying their problem behavior or acquiring new healthy behaviors. And then we have the maintenance stage. In this stage, people have sustained their behavior change for a while, defined as more than 6 months, and intends to maintain the behavior change going forward. People in this stage work to prevent relapse to earlier stages. And then our last stage is termination, and people have no desire to return to their unhealthy behaviors and are sure they will not relapse. Since this is rarely reached and people tend to stay in the maintenance stage, this stage is often not considered in health promotion programs. Moving forward. in our research implications, according to the trans-theoretical model, which aims to explain readiness for change, many unvaccinated nurses are in the pre-contemplation stage. Effective strategies for encouraging behavior change among pre-contemplators include recognizing current views, building trust by empathetically discussing specific concerns regarding vaccine hesitancy. And as you can see right there, it says conscious raising, dramatic relief, and environmental evaluation. And next slide. I feel like I got through that fairly quickly.
You did fine, Brittany.
Okay.
It's fine. Did anybody have any questions?
No, I have a question.
What made you choose this topic?
I feel like with the pandemic started, I wanted to know more. And then I started seeing a lot of hesitancy amongst maybe people that was around me. And I have like family members that's in the healthcare field. I mean, I'm pursuing the healthcare field as well, but my like family members are like nurses, and you know, even like I have like a cousin that's a doctor, and a lot of them were still like hesitant. So seeing them be hesitant about getting vaccinated kind of like made me interested in viewing this topic, because I was just kind of curious as to why people, you know, were, I mean, there's a number of reasons. And of course, you know, at first I was a bit hesitant myself, but after doing the research, it kind of, you know, puts you in a better position. So I was just interested in knowing more about it.
Danny, you had a question as well?
Is this research something that you would want to conduct at like different universities or in different settings to gather data and see if it resembled what you were able to get today?
So I worked with my mentor, Dr. L, on this, and I'm sure we would conduct it at different universities. I'm sure it would be something foreseeable in the future. You know, she kind of put this together, or she kind of presented it to me, and then after seeing vaccine hesitancy in my own family, it kind of made me more interested. So I wouldn't be hesitant to actually see it at different campuses and get more information.
This is a fascinating topic because I'm always so non-understanding of the healthcare field, or rejecting this idea and treating and seeing these patients suffering. So this is fantastic. Did your results match sort of your ideas going into it, what you were thinking you were going to get out of it in regards to your answers?
I think because the requirement on the University of Michigan-Flint campus, because there is a requirement and majority of our participants were vaccinated, I would have liked to seen a bit, you know, a heavier portion in the data, because with majority of the students being vaccinated due to the requirement, I don't know if it was something that they actually wanted to do or something that they had to do. So I do think that we got some great information out of it. And I do think furthering, going down the line, we can do some intervention work, like utilizing the trans-theoretical model. So yeah, I think it was a great turnout. At first, I didn't know what to think of like receiving participants. I didn't know how many we're going to get. So I was excited to see that, you know, we actually had 230 participate, but there was only 194 of them that actually completed the survey. So I thought that was a great amount of student participants.
Yeah, that's much higher than I would have guessed, just
Yes,
with the just with how that whole concept is. And yeah, that's fantastic. I loved it.
Thank you.
All right, with that, I think we'll have to move on. Thank you, Brittany.
Thank you.
And we've been having some technical difficulties on the back end here, but I think that's straightened out. So I'm happy to introduce our last presenter, Christina Zerka, from U of M Flint. Christina?
Yes, thank you so much. And my apologies, I was having extreme difficulty today, but thankfully that is all resolved. I'm glad I'm able to be here today. Yes, I used to be a student at the University of Michigan-Flint. I am now a student at the Ann Arbor campus, but I have been working with Dr. Kristeva Gargana, and we have been working on a systematic literature review on the behavioral and psychological concerns on the Flint water crisis. Again, this is an issue and topic that we see over and over again because of the importance that things that it's taught us and the effects that it still holds today on the community. So yeah, without further ado, let's get started. So, and most people I'm sure know some of the backgrounds, but I'm just going to mention it again. The Flint water crisis began in April of 2014. So this was not too long ago if we think about it. It was all over the national news and such. And there was basically a switch made from municipal water in order to save money. The switch was made from Lake Huron to the Flint River. And as we know, the Flint River had corrosive lead pipes in the water that it took to the homes of the community and had it contaminated. And many people were infected in the community of Flint, about 100,000 individuals. So it is important to mention that lead, the effects of it are long-term, and it specifically impacts the organs. Second of all, this was a public health emergency that was declared a year later, pretty much in October 2015. And the individuals, the 100,000 of them, hypertension, which is high blood pressure, kidney diseases, and many more issues were found because of this. Also, lead can also cause depressive symptoms, which we will talk about further in this research. Excuse me. So it is important to mention that out of the 83 countries in Michigan, Flint in Genesee County is rated the 82nd in health outcomes, which is extremely poor in the ratings, and that 42% of the city's residents are below federal poverty levels. This means that they might not have access to the best health and treatments. And Flint is ranked in the top 10 as the most violent cities in the United States. Excuse the typos in this, I'm so sorry. Also, it's a majority non-white community with 56% being African-American, and it is more prone to poverty, unemployment, and chronic disease in the research that we have found. And then all these factors are affecting individuals with previously issues with mental illnesses and barriers to treatment as well, because of the issues with them being 82nd in the state, as well as having the residents be below federal poverty levels. Next, please. So there were some measures taken by the government to help with the Flint water crisis, and we're going to explore those. Some of them were, like, the Genesee County Health Department established mobile food pantries, and that allowed for fresh fruit and vegetables to try and combat the harmfulness of the lead. Scientifically speaking, vegetables and fruits are more prone to help with the effects of lead, so that is why they decided to do that. A second thing that they did, the Genesee County Health Department, was those water tests around the city for residents to confirm that they did have lead in their water. And another thing was the free bottled water program by the state of Michigan, which actually began in 2016. So this wasn't until 2 years after the fact of the contamination. So that CDC actually responded to something called CASPER, and this was an assessment or a test in order to gauge the impacts on behavior of the public in Genesee County and specifically Flint, Michigan. And here were some of the data that was found. And we can see tremendously that mostly the aggressiveness was raised, as well as concentrations and sleep deprivation and moods. So those were the main, I would say, outcomes. And you can see that they tracked it throughout 2016, 2017, and 2018 as well. And you can see the data here, and this was for above the age of 21. Now for younger, we can also see that there was still a heavy impact and effect. A little bit less here than problems in school, like 30% and such. We can see a lot of the depressive moods was up in older adults, and it was a little bit less in younger, under the age of 21. Trouble concentrating was pretty much the same in both, and so on. This does not seem to have improved much over the years, as we can see throughout 2018. Next, please. So this again, specifically on the depression and anxiety scale, we see that. Also, they asked about rent payment and specifically money to buy nutritious meals, as this would affect their health overall. And we can see that there was definitely a tremendous amount of people who had trouble with this. So it was great that the Genesee County Health Department did offer a program for this, specifically to help with nutrition in the area. So overall, to analyze the data, we saw that over 21 saw many improvements in depression, aggressiveness, and stress. But despite the improvements, there was no use of the behavioral services. So like mental health uses, such as the ones that the county was providing, they were not used, but there were some improvements. And 50% of the residents, this was the most shocking fact of data, said that they would not see the crisis resolved in 2016, and the same was true in 2018. So it's still 50% of residents, which is a big issue that is still alive today. And that is it for this. And does anyone have any questions about the data or anything in general?
I have a question. In the previous slide, there was a fourth bar, and I was just curious as to what that represented.
Oh, those are not in correlation to the data. This was one of the templates that I used.
I see.
That does not correlate to the data.
Gotcha. Thank you.
Mm-hmm.
I had a question, if I could, Christina.
Yes, of course.
And I realize that you're using data that were taken from this large study, right?
Correct. And this study, are you going to ask how many participants are in the study?
No, I wasn't.
Oh, okay.
What I was going to ask, though, is that I know from my public health background that these longitudinal studies are sometimes challenging to get comparable results year after year. And I didn't know if you knew if they tracked the same exact people year after year, or if it was just the same study done in different years with different groups of people.
They actually did track the same people, which is beneficial. Sample size wise, I believe it was about 5,000 of the residents. So it was a pretty good sample size. Obviously, we always like more of the residents to join, but this was, I would say, pretty representative. And yes, it was the same residents, to answer your question.
Okay. Other questions from the group? Okay, I'll ask one more then, Christina. What's the next step in this kind of research? Where do you go from here?
In my opinion, where we would go from here is to do another survey and assessment on current day 2022, where the residents feel whether they are still comfortable facing issues, health concerns, behavioral concerns, to help the community even further, because this did stop in 2018. I think they should have tracked it for much longer, as lead has long-term effects, and 2018 is not a comparable time, and it should have lesson on for at least, I would say, 2 more decades, especially for those under 21 and the children that were affected in all of this.
Very interesting. Thank you. Any other questions from the group? If not, I will thank our presenters today. This was a great session. We did just get the note from Mary that breakout rooms are starting to wrap up. And so I would encourage you all to head back to the main room so that they can give us the overall sort of comments and wrap up the presentations for the day. But great job to everybody. And these recordings from this breakout room and all the other breakout rooms will be available through the conference as well later on. So if you missed any talks today while you were here, feel free to check that out later on. So with that, everybody have a great day and thanks for coming to our session.
And just as a reminder, as people come in, so we'll do formal introductions. So I'm Dr. Littleton, for those who don't know me, from psychology professor at Mott Community College. So I'll be moderating this particular breakout room. We have a total of 3 presentations that we have here today. If you have questions, I ask that you hold all your questions to the end of that presentation. You can also use the chat if you want to ask questions. You can use that as well as a way to send questions in, and I can deliver the questions over to the presenters.
And Brian, it looks like Mary and our student, is it Courtney? They're the only ones in the main breakout room. So it looks like everyone else has selected, you know, what breakout they're going to get into. So we can start, I would guess, sooner than later.
All right. And just before we get started, are there any questions anyone has?
Yes, thank you.
Yes.
My name is Jabbar Abusin. May I have the permission to record my presentation on my own computer?
I don't have those capabilities, so I don't. If you can record it, you can hit your record button. If it allows you, go ahead.
It says you got to take a permission from the session host.
Let's see here. Because I'm only a co-host, so I don't have all the, I don't have that capability on my end. That would have come from Andre, because he's the main facilitator host of this account. So I don't have those privileges.
And Jabbar, this will be recorded and released, you know, online. You'll get this recording or have access to it.
Thank you.
Thank you. Yeah, it will be recorded. Normally they send it out after they get done editing some edits, maybe about a week or 2 after. Normally it comes available for everybody to see. All right, with that, we will go ahead and begin with our first presentation. And also just ask, make sure everybody who is not presenting to mute your mic so that way it doesn't cut in. And our first presentation is by Kylie and Mallory. And so the floor is yours.
All right. Hi, I'm Kylie, and myself and my partner in this project, Mallory, present you with Securely Neurotic. Does attachment style correlate with neuroticism? So next slide, please. So our variables in this project were attachment styles and neuroticism, and specifically the 3 attachment styles that are measured in the state adult attachment measure. So attachment style is an emotional bond formed with significant others from infancy throughout life. And the 3 types are secure, which has to do with positive feelings, worthy of being loved, just safe, secure attachments. And then there are two types of insecure. The first one is anxious. This has to do with increased reactivity, generally negative, very like needing of reassurance a lot of the time. And then there's also avoidant, which has to do with the tendency to downplay or suppress emotions or avoid them. And then also neuroticism, mental distress, emotional suffering, and inability to cope effectively with the normal demands of life. And that's according to the IPIP home website, and we'll get into what that is in just a minute. So next slide, please. So why we chose this study is because we were really just curious about the possible relationship between neuroticism and insecure attachment styles. And we also found another study that supported our train of thought, and they said that these types of studies are important to better understand attachment and what it correlates with for safety, stress regulation, and adaptability. Next slide, please. So we had two hypotheses. The first one, neuroticism will be positively correlated with insecure attachment style. The second one being neuroticism will be negatively correlated with secure attachment style. Next slide, please. So our support for these hypotheses, the first being, we really just thought that this could be true because insecure attachment is typically compared with low self-esteem, negative feelings, things like that. And neuroticism is also compared with feelings of being blue and like depression and irritability. So they have very similar traits. So we kind of thought the higher you test in one, the higher you must test in the other. And then reasons we thought that secure attachment would be negatively correlated with neuroticism is because it's known to be positively correlated with various beneficial outcomes such as relational stability and satisfaction, improved emotion regulation and well-being, and increased pro-social tendencies. So next slide, please. And I will hand it off to Mallory now.
So our participants in our study were Mott Community College students. There were 21 total with an average age of 25.89. 10 of them were female with an average age of 24.73. And 6 of them were male with an average age of 28.86. And then we had one other. Next slide, please. So our measures in this study were 2 tests. The first one was the International Item Pool, and the second was the State Adult Attachment Measure. The International Personality Item Pool is a test that measures the different levels of the Big 5 personalities. Those Big 5 personalities are openness, conscientiousness, extroversion, agreeableness, and neuroticism. We took the scores from the neuroticism measure. The State Adult Attachment Measure takes the fluctuations of the different attachment styles, and we found, then they tested the 3 attachment styles, secure attachment, and then 2 insecure attachments, avoidant and anxious. We compared all 3 of these scores to the neuroticism scores. Next slide, please. Here are the differences in scores based on gender. So as we can see on the graphs, the females and the one other were lower in security, and then they were higher in avoidance and higher in anxiety than males. But then on the neuroticism graph, we can see that the females showed a much lower score than both the males and the other. Next slide, please. Our results for anxiety, attachment, and neuroticism. So we used the Pearson's product moment correlation coefficient to calculate the R. So our R was negative 0.0279 and P was 0.22. And because P was not 0.05 or less, it was not significant. So therefore we cannot reject the null hypothesis. So we found there was no correlation between anxiety, attachment, and neuroticism. Next slide, please. So our results for avoidant attachment and neuroticism. Our avoidant attachment, our score was negative 0.2851, and our P score was 0.2. And because P was not 0.05 or less, it was not significant. So therefore, we again cannot reject the null hypothesis. So we found there was also no correlation between avoidant attachment and neuroticism. Next slide, please. Our results for secure attachment and neuroticism. Our secure attachment R-score was 0.2846, and our P-score was 0.22. And again, the P was not 0.05 or less, so it was not significant. So we found there was also no correlation between secure attachment and neuroticism. Next slide, please. In conclusion, our hypotheses were not supported. We found no relationship between attachment styles and neuroticism. Although we could not reject the null hypothesis, we found there was a surprising trend toward a positive correlation between secure attachment and neuroticism, and a negative correlation between insecure attachment and neuroticism. So in the adult attachment insecurity and hippocampal cell density study, it was found that neuroticism was not significantly correlated with attachment. So we found the study later in our research that supported our no correlation. Future studies could include studying secure attachment and neuroticism, have bigger sample sizes, include other demographics, have a more evenly distributed sample size between men, women, and other. We could also address the surprising trend in future studies by researching what aspects of attachment make these findings what they are. For example, too much of a good thing can be bad. From the study of the consequences of high levels of attachment security, it was found that older adults who received high levels of help or too much support from their adult children reported a poorer mood. Similarly, it was found that well-functioning older adults who received too much support exhibited a greater risk of future disability compared to those with less support. So we found that, having too secure of an attachment style can almost be negative. Next slide, please. Thank you for listening to our research. And then next slide is our references.
All right, well, thank you. So I want to begin. Are there any questions? If you have questions, again, you can put them in the chat. If you don't, or either if you want to just, because we're a pretty small group here, you can just kind of take yourself off mute and chime in with your questions that you may have. And nobody has any question. Well, I do have a question. What was your sample size again? I don't want to kind of mess up the slide.
Our sample size was a total of 21 participants.
Okay, so yeah, okay, I was curious what the sample size was, so that probably may explain why you did not have enough variability within the responses to maybe pick up anything. So had pretty low statistical power with the 20, but good start for where you're going with it though. So hopefully,
yeah,
do you plan on?
Exactly, Brian, but you could see a couple of those correlations were in 0.28 or a negative 0.28, whatever it was. And so you can see the trend where like, hey, let's get a little bit bigger sample, it'll pick up, but it was still a surprising trend to find, the opposite of what they expected. You know what I mean?
Yeah, I'd be curious what it's going to look like when I assume you're going to continue to collect, correct? So if you need help collecting, you can always email me. Okay. There's a question. I have a question from Mindy. I hope I pronounced it right. Okay, you can go ahead.
Yeah, I just want to know, how did you select your population for your samples? How did you go about that? Especially right now, since everything's like distant. How did that work?
All of our information was from a database from a professor at Mott that he collected through his students. And so they took these tests and he recorded the data. And so we were able to use that data from him.
All right. And I see, is it Jabbar? Did I pronounce your name correct? Did you have a question? Okay, yes, you have a question?
It's typically the same question I want to ask is about the sampling procedures, and she just asked a question. So, yeah, because we are having the COVID-19 around, and I'm like, how this could happen. So, but it looks like she's using a secondary data.
Yeah, exactly right, and I can help fill that out. I'm the faculty advisor for this project, and I'm the professor whose data they're using. And so over the past couple semesters, or with each semester, I have a theories of personality class. I have a lab class. So that really gives us the opportunity to work on these different measures, apply them to different topics, talk about them as they relate to different topics in psychology. So I've been collecting data on my students' responses, and it's all in confidential and informed consent process. They know their data is going to be stored and possibly used in these reported, or in research presentations and in research. So to kind of address the sampling, even with being virtual with COVID, I can still have students, we can still have students, or participants fill out surveys online. I used Google Forms, or like the IPIP, you go to a website, you get your interpretation or responses from that website, and then students would directly report to me what their percentages were, their scores were. So it's like a self-reporting of that measure if the measure's on a different website from something I put together. So hopefully that helps.
Are there any other questions? I know we have a few minutes before the next presentation starts. If you do, you can just, because I can't see everyone on the screen, you could just jump in. All right, no other questions?
And can I just say, Kylie and Mallory, you guys did a great job. Outstanding.
All right.
Thank you.
Thank you.
So with that, I guess we can, since we have no one else, we can go ahead and proceed to the next presentation. And the next presentation here is Rob. You can take it away.
Yeah, thank you very much. This is actually, it's been established as a requirement for Dr. Wyneken with the University of Michigan-Flint in a class teaching numeracy across the curriculum. And then it looks like, and recently I was getting into the issue of disciplinary literacy while taking a class in graduate school. So the program is basically about using sales ads to foster middle schoolers mathematical literacy real world situation. That I'm like trying to put the students out of the class, and instead of bringing ready-made formula, it's just taking them out, letting them experience the world of mathematics in real situation. Next slide, please. So like I just said, this is for Dr. Wyneken in fall 2021, in which we try to enforce mathematics with other disciplines. Then, and also I was supported and mentored by Dr. Helliker at the Holmesdale Middle School Academy with Flint Community School, who was supporting with a class issues inside and try to protect, try to back me while I'm doing this project with 7th graders at Holmesdale Middle School Academy with Flint Community Schools. Next slide, please. When I was assigned to do this project, it just come to my mind, my work with my own child. His name is Abdurrahman. He's right now a 4th grader. Anytime we are doing shopping at Meijer, Walmart, Kroger, whenever Genesee Valley Mall, whenever we have the opportunity, we talk mathematics while doing shoppings. For example, he usually asks me, Baba, what does 25% means when we are trying to buy, for example, jeans or t-shirt or whatever. So people like wondering and looking at us while doing math, while shopping. So I'm like, 25% is just, you need to apply this is into the sales. And particularly when we bring pizza home and we cut it, we look at the slices and then like, okay, this pizza has 8 parts. So each one of them can be represented in a fraction or in a decimal. And then I was telling Dr. Weineken, the idea comes from my child. Next slide, please. Like I said, it's with Holmesdale Middle School Academy in the fall of 2021. Next slide, please. The main objective for this project is that students are able to understand how to do basic mathematical operations, subtraction, addition, multiplication, division in real world situation. And also the goal is that to make the students able to comprehend mathematics in real world while doing the readings, and then try to associate the numbers with text. And then the students are able, by the end of this project, to interact with the text, navigating the ads, navigating, for example, the pictures associated with the merchandise, a bag of chips, a box of candies. And also the students are able to use mathematical thinking and reasoning. And instead of just having ready-made formula in the classroom, they are able to think mathematically while they were forced, put to face a challenge. Thank you. Next slide. This project is intended to enforce this Common Core state standard in which disciplinary literacy in mathematics is the top goals. So students are able to think about mathematics and apply properties of operation and do calculation with numbers in a situation probably considered to be a real world situation. Next slide, please. I was preparing this project thinking of the students' need. Normally students in 5th to 12th grade have problems dealing with word problems. Students can be in a situation like, for example, 2 times 3 or 7/5 times 6 just being put on the board. They can discuss this. But when they have the real world problems like word problems, they're gonna try to figure out the main idea in the problem and to try to break out these problems to make sure how they can approach it. This is one of the students' need. And in addition to that, normally mathematics is put in formulas, but as Joe Bowler of Stanford University suggested that visualization is most important when teaching mathematics. And also students need to use literacy strategies. They need to do, for example, reasonings, highlighting, filling graphic organizers, or looking at these numbers, associating the numbers with the context. Next slide, please. So this work is supported by the work of many research experts, including Elizabeth Spear-Moge at the University of Michigan, and her work on disciplinary literacy. And these are a number of research enforcing and supporting the teaching of mathematical literacy while having the students doing the basic mathematical operations called the disciplinary literacy mathematics. And in this project, the students are able to look at the Kroger ads to do the visualization, reasoning, resourcing, enforcing math with literacy. Students are able to look at mathematics in real world situation. Next slide, please. So the lesson is structured in 3 parts. Part 1 is just set the introductions. Part 2 is the actual work where the students focuses in the Kroger ads. And then they have like 20 minutes. And then the students are going to look at the condition in the sales. They're going to look at these papers and try to figure out how they can associate this with the content being discussed in the classroom. Part 3 is assessment, which is formal and informal assessment. The formal assessment is in a form of word problems, and the informal assessment is teacher observation and try to make sure how the students are responding to my question while we were navigating through the Kroger ads. Next slide, please. The lesson material, like I said, is just the Kroger ads from the local store. The students are having these flyers. They look at the sales conditions. For example, buy 5, get this discount, and the expiration dates, and looking, for example, the merchandise. Next slide, please. The assessment in this lesson in structure brings 2 pages. Each page has 1 math problems. And like I said, it's a mix of formal and informal assessments. Next slide. For example, this is one of the scenario. The students are able to look at the ads and they are able to read these problems. And for example, Ms. Petlov wants to buy, for example, the Gala apples. And then she picked up 3 pounds of raspberries and strawberries and 4 pounds of strawberries. And then this is the first time to go to Kroger. And the question is that I mentioned this is the first time to go to Kroger because she doesn't have a Kroger ads. So the students are going to use their mathematical reasonings. They're going to use readings. It's not only teaching mathematics, it's also teaching the students how to read, because one of these sales says you have to have a Kroger ads. So I am giving this problem, the students will be able to think before they approach the problems. This is one of the scenario. Next slide, please. Another one is similar. This lady is shopping with her 2 kids. They want to buy chips, but one of them choose 4 bags of Doritos and the other choose 3. How much she is going to pay? So the students, those students who are not really thinking and reading and applying their reading comprehension abilities, they're going to just count 7 times the normal price. But the sales ad says, if you buy 5. So it's just like a real world problems. And I'm testing at the same time the students' literacy, not only testing the multiplication. Next slide, please. This is the Kroger ads. Next slide, please. These are what new teachers need in designing a lesson, in setting the objective, associating their lesson content with a Common Core standard. Next slide, please. What was surprising is that after the lesson, I looked and find that my students are able to apply mathematical thinking, reasoning, referencing the materials. And it's not only, they understand that it's not only doing the multiplication, it's also using their reading ability, literacy, referencing, and applying the resources that they are navigating. Next slide, please. So for example, in this question, Catherine received an email notification of 40% discounts on air tickets from Chicago O'Hare to 50 destinations around the world. She wanted to take advantage of these discounts to book a flight to travel to Indonesia. The discounts is only on adult's ticket. Catherine booked 5 tickets for herself, her husband, and the 3 children. So other students are gonna say, okay, these are 5 persons, and then they're gonna multiply this in the normal base fare. But another student, next slide, please. Next slide, please. And yeah, sorry, the one you just passed. Sorry. So another student was shouting to, I mean, just talking to the person next and saying, hey, the sales is only on adults. So the students are able to use their literacy abilities, reading and thinking, and instead of just multiplying into the base fare. So I was so happy to see my students applying literacy skills inside the mathematical problems. Next slide, please. If I was given the opportunity to do this project, I'll be able to bring a guest speaker to talk to the students on filing a tax, because this is going to help the students reasoning, asking questions, building the knowledge, taking notes inside mathematical, I mean, in a math class. Next slide, please. Right now, based on this project, I'm doing this proposal, which is a lesson to parents, how to enforce mathematics in real world situation at home. Next slide. These are the suggested work on disciplinary literacy. And then I'm so happy to have you attend my presentation. Thank you very much.
Well, thank you. Very fascinating. What was the age group again that you did this lesson?
Those are 7th graders.
7th grader, okay. So if anyone has any questions, you can chime in with questions or either through the chat.
I have a quick question. So you mentioned there was a formal or formative assessment, and then you had more of the informal, and it was great to learn more about the informal, observations and assessment of how the students were doing. In terms of the formal assessments, do you have the data on that to show like a pre-test, post-test, how they were doing on math before versus how they were doing on it after the lesson? Do you have anything like that?
Yeah, if you go back to the slides, please, a little bit to the question. This question was, yeah, a little bit more. The scenario number 1 and scenario number 2. Yeah, here. This is actually has been passed to students, and the other scenario, and the students at the same time having the Kroger ads flyer in their hands. So the students are thinking on this problem. It's a word problem. So the students are going to look. So the assessment is on literacy while doing mathematics. So it is not normal multiplication or addition or subtraction. They are going to see, for example, if you look at the Kroger sales ads, it says that if you buy 5, it says that if you have a Kroger ads, it says that if this sales is not expired. So I am testing the reading, their ability to think on the text while they are doing the multiplication, because putting the students in the literacy skills assignments is my goal, because I'm intending to develop students literacy while teaching them mathematics. And this is one question.
Yeah, I think it's a wonderful idea, and I'm a parent of an 8 and a 5-year-old, and I absolutely, I agree with this, and I try to do this. I love this idea. So what I was trying to get at was like, okay, do you give that one of those word problems or something like that before introducing the program or the information or the real world stuff, and see how they're doing with math versus after, and see if there's any improvement at all? That's what I was kind of asking, is like, how do you know there's improvement?
Yeah. Okay. So the next question on this, if you go to the next slide, please. Here you see, for example, I heard the students talking about this discounts, and one of them is applying the 40% to 5 tickets. A student on his side was correcting her, and the sales only, talking, says the sales only on 2 tickets for Catherine and her husband, as they are only. I was able to see the students learn and comprehend the numbers, because one of students says, no, the sales is only on adults. So I was able to see that the students are checking the language, because this student is reminding the other students that the sales is not on all people of the tickets. The sales is only for adults. So this is, just gives me an idea that the students are able to think on the language in the text.
Thank you, Jabbar.
And I know I'd allowed to go a little bit over time because I know we were missing one presentation, but does anyone else have any questions? I'm trying to see if I see anyone. Is there a question in the chat? So many just mentioned that great idea. Wish that my 18-year-old had the opportunity, especially the tax preparer. All right. No more questions? Yeah, I think it's very fascinating.
Thank you.
And our last presentation on the sex trade by Ethan and Avery, and you can go ahead and begin.
Hi, everybody. My name is Ethan Greenberg.
And I'm Avery.
And today we're going to be talking about the specific subdivision of human trafficking, which is the sex trade. Next slide, please. So according to the US Department of State, roughly 24.9 million people worldwide are victims of human trafficking. But a common misconception is that sexual exploitation is what human trafficking is. Although it's one of the biggest areas, human trafficking also includes forced labor, which is when people are unemployed against their will, debt bondage, which is when a person is forced to work against their will to pay off some sort of debt, domestic servitude, which is where a domestic worker is abused or unpaid and are allowed to leave their place of work, and finally, forced child labor. The most targeted age of sex trafficking is, in the United States, is age 12 to 17 years old. But sadly, victims can span all the way from newborn children to the elderly. While sex trafficking is a sensitive topic to talk about, it's vital to understand the warning signs and how to help. Next slide, please. So our objective with this research was to specifically focus on the sex trafficking subdivision of human trafficking in the United States. Next slide, please. Our methodology of how we went about this was using a literature review using the PsycInfo database, and we used the following keywords, which were human trafficking, sex trade, and sex trafficking, to get a better understanding about our topic. Next slide, please.
Okay, so human trafficking is the use of a person for the purpose of labor, domestic, and other commercial sexual activity by force. Human trafficking is also a crime and a public health concern that affects individuals, families, and other communities. However, if this activity involves a person under the age of 18, then it is automatically human trafficking. Next slide, please. Some of the characteristics of sex trafficking victims. They can be any age, although most targeted is 12 to 17 years old. You can see in the chart, women is at 54%, so they are the most targeted. Generally, the most vulnerable population is targeted victims, emotionally, physical abuse children, teens, and drug, alcohol addicted people. Some situation factors include targeting people who are disabled, homeless, and poor. Next slide, please.
So a key process that goes into the start of human trafficking and throughout the rest of the process is called the grooming process. There's 6 stages to this process. The grooming process can be defined as creating an emotional attachment with an individual in order to lower their guard with intentions of pure sexual abuse. This is often done by making false promises, fulfilling a victim's needs or wants, and instilling a false sense of love and care. So going more in depth on the 6 stages. So stage 1, a trafficker will target a victim. Typically, a trafficker is very picky with who they pick because they go after anybody. Stage 2, they gain trust. This can be on social media, in person. They can talk and overshare things just to lower the victim's guard. Stage 3 is filling a need, which could be money, love, care, like I previously discussed. Moving on to stage 4 is when they try to isolate the victim. This is when the trafficker tries to move the victim away from their friends and family. So it's pretty much just the victim and the trafficker. Stage 5 is abuse, and this can be physical or mental. And stage 6 is maintaining control, so the trafficker maintains that abuse with the victim. Next slide, please.
Okay, how social media ties in with human trafficking. Social media is used a lot nowadays in our generation. Today, it is easier than ever for human traffickers to find a victim. After the first meeting, the groomer stage takes place. The trafficker cultivates a positive relationship with the potential victim. They may say things, catfish, love interest, may shower a young girl with beautiful gifts for being beautiful. Fake school recruiters may conduct interviews with a potential student about career choices, or offer to pay travel or relocation cost. A criminal may create fake social media accounts specifically to interact with school-age children in this process. They may use the fake profiles to dialogue online for months, pretending to have common interests and building trust. Next slide.
So moving on to sex trafficking specifically, it's for the purpose of pure exploitation of somebody. It's been called a form of modern day slavery because of the way victims are forced into sexual acts, which are usually non-consensual and a form of sex slavery. Some interesting statistics we found were about 40% of sex trafficking cases involve minors. Trafficking also targets homeless children because they are all alone and isolated from parents. The average cost of a sex slave is only $90, and the sex trade earns global profits of roughly $150 billion a year. We also found that the sex trade and sex trafficking is one of the largest growing criminal enterprises in the entire history of the world. Next slide, please.
Human traffickers and risk factors. Some of the risk factors include poor neighborhoods. Traffickers take advantage of people who may be homeless, in temporary housing, and even the unemployed. Abusive homes. Children who have left abandoned by a parent, youth with a history of abuse, and teenagers who run away from home are at a higher risk. Substance use and abuse. Traffickers use addiction to keep control of their victims. Some traffickers even purposely supply the victim to break down the resistance. Most people assume that traffickers are all males. However, there are multiple cases against women traffickers. People who also have offended once have a higher chance of reoffending. Next slide. Some of the warning signs for the parents can include unwilling to tell you about their friends, not being transparent about who they hang out with and who they decide to date, having multiple social media accounts, and being defensive to certain questions.
Some potential warning signs for victims include running into strangers more often who you don't know. Strange gifts or items appearing on a vehicle or at your house. A good example of this was a couple years ago, human traffickers would often take ribbon and they would tie it around door handles on vehicles, and this was a pretty much marking of a target. People making false promises who you don't know, like things that sound too good to be true. Like, if you hang out with me, I'll pay you X amount of money. Blackmailing, if you don't send this to me, I'll do this to you. Random messages, so text, mail, etc. I know with TikTok and Snapchat, Facebook, Twitter, all the social media that we have nowadays, it's easier than ever for somebody to lie about who they are. You can pull a picture off of Google, send it to somebody and say this is who you are, and they often don't question it, which is kind of scary. And strangers oversharing personal info, tying this back to the grooming process. When they do this, it's a way to lower your guard or make you uncomfortable. Next slide, please. So what's being done? Specifically in Genesee County, the Genesee County Sheriff's Department is tackling the issue head on, and they work with joint offices and counties. The GHOST Task Force, which stands for the Genesee Human Oppression Task Strike Team, has made multiple arrests and are actively involved in the community to keep everyone safe. They have a 100% conviction rate as well. So after hearing all this, you may be wondering, how can you help? So 3 things that I know of off the top of my mind is know the warning signs and educate your children, friends, and family. So just like you have like the birds and the bees talk, the drug talk, human trafficking should also be a part of this because it's more than just stranger danger. Be there for others and report suspicious activity to law enforcement if you feel something's happening. It's better to be safe than sorry, and it's always good to have reassurance. And then finally, I encourage you all to take the Genesee County GHOST Certified program that's led by Sheriff Swanson, the links below. And this helps grow your knowledge and helps you become even more educated on the subject. Along with this are videos, and he often does town halls every other week to talk about this. Next slide, please.
Okay, for our conclusion, the sex trade is a huge problem that keeps on growing bigger and bigger. The most important thing to do is explain your knowledge of the warning signs and the characteristics of the sex trade and traffickers.
So although this can be an uncomfortable conversation to have, it's vital to stopping and reducing the problem. After all, this is happening in our community and it's happening all around the world. We thank you for listening to our presentation and hope you retain some of the information we have shared.
So, thank you. Are there any questions that we have for Ethan and Avery? And I'm trying to...
Professor Littleton, somebody's asking for the link in chat. That's one slide back.
Oh, yep. I'm sorry. Sorry.
No,
the link in the chat. Yes, I will get the link. I'm going to go actually back a slide then for the link. And are there any other? Mindy, I see you have a question.
Is there a way to go back to the slide with US on it where you got your information from?
Okay, yes, I can do
I want
that.
to see that again. Yeah. I just wanted to look at it.
That's it. That's oops.
It should be the introduction.
And is this like reported or attempted? Like what exactly is it?
So the main thing with this chart is most organizations with any crime, they can only report things that they have like solved. So as you can see, hotspots in major cities like LA, Detroit, Chicago, Miami, those are big spots for human trafficking. And this chart's from 2017. There hasn't been an updated chart. Of course, with COVID, it's hard to get resources like that out there. But these are only cases that have been solved. So realistically, this entire map would probably be read with all the things that have not been solved.
And Jabbar, yeah, have your hand.
Just I want to ask if you guys have any attempt to reach out to victims, for example, with the Genesee County jails or
like victims of people that have like victims of human trafficking. So that is extremely hard to tell because most of the time human trafficking isn't reported because the victims are often scared that abuse or other things like that will happen to them. So it's not easy to get a count on the victims because there are so many. But what we do have is the felons that have committed these crimes. I want to say there's been over 300 arrests just around the Flint area. And that's just in the past few years since the team has started.
Yeah, that's a big number. And you see, the question, the reason I asked the question because I'm thinking of, for example, a survey or like a questionnaire being distributed to gather the basic information. What are the reasons behind their cases? Why are these people become victims of these situations? because
Yeah, I agree. I think
you mentioned a teenage who ran out from a family. You mentioned someone left alone in the house. So
I think nowadays it also relies a lot on social media, like kids my age, like online dating and stuff like that. Online dating can often be a trap for people, and they're talking to somebody that is not who they say they are. There's countless victims of that. And I think once people go and they meet up with these people, then they're taken advantage of because it's easy money for the traffickers.
Thank you.
And Ethan and Avery, I have a quick question. On one of the earlier slides, you said the average cost of a sex slave was around $90. Is that like the cost? I don't know how to think of it. Is it the cost to have sex with them, or is that the cost to sell them to like someone else?
I assume that that's the cost to like engage in sexual activity with them, and that's the average cost. So what traffickers often do is they'll have like a group per se, and then that's how they make so much money so quick, because if they have their group, they're not going to hurt them or kill them because it's pretty much like a money maker for them. So if they're relatively healthy and they're obedient, then they're going to keep them more. If not, they'll probably sell them for a couple $1,000s, and then the trend just continues.
And looks like a question from Mindy again. Go ahead.
One more question. So did you see any information about international rate? I know like there's a lot more that goes on between US and other countries. Did you see anything?
Avery, do you want to take this question?
Yeah. So what I would say is, like Ethan said, that we can't know all of it because most cases are not reported. So there's a lot that we don't know about. For example, just this morning, 4 of Disneyland workers were arrested because they were part of the human trafficking. So I would definitely say it's international, but we can't know. There's no possible way to know where it's all going on and with how many people and stuff.
And Professor Littleton, if you could go to I think it's our objective slide, back to that map.
Yeah.
Okay, so just by looking at that map, the red concentrated areas, and this is from 2017, the problem's gotten a whole lot worse since then. You can tell it's on some of the key borders. Like if you look at Michigan, for example, like the Flint area and surrounding like near like Detroit and things like that, that's a direct access into Canada. So if people can get people into Canada and into Mexico and across the ocean, then that just makes the problem even bigger. I know a big hub for human trafficking is in China. China has a lot of human trafficking. Mexico has a lot. And yeah, like Avery said, it's just hard to get an exact number because it happens so much and they're so secretive about it.
And looks like another question. You can go ahead. I see your hand is up.
Yeah, sorry. I just want to follow up with a question regarding the international issue in this. I believe the airport custom and border protection personnel are able to provide with this. Did you attempt to reach out to?
We did not attempt to reach out to them per se. I didn't really actually think about doing that, which would benefit our project a lot. But I think in the future, if we represent over a topic like this and do maybe like a deeper dive, then we would definitely contact more organizations and agencies to see their numbers. For this project, we mainly focused on Genesee County, and I talked to Sheriff Swanson a lot about it. And if you go to the that Go Certified program, then there's a whole lot more information from the Sheriff's Office itself that explains more international and things like that.
Thank you.
And I do know, I'm just chiming in. I know Flint, Genesee area was a very big, one of the top spots actually in Michigan at one point. It was the highest, surprisingly, out of all the areas. And I know there's been particular paid attention on Dort Highway in the past, was a big corridor or a lot of trafficking occurred on. Some of the issues, again, when it comes to, as they were kind of mentioning in the presentation, is kind of tracking them and where they're working. Massage parlors are a big place of what they call illicit massage parlors, where it's kind of like the back door to the back door is a big area. So, any other questions? Oh, and I can go back and answer your question about, someone had a question, but so that right there is average cost for like any type of sexual favors, so to speak, was the average cost. The average sex trade person victim brings in about 100. I think last I checked, I think it's about 90, close to $100,000. And they happen to be a little bit more in the developed world. They tend to make more money for the persons doing the trafficking in the developed world compared to those in other countries. So they tend to do the victims make more money here in the United States, although it's less compared to some other countries. All right, any other questions? All right, with no other questions, we have some time left here. If you want, I think everybody's probably going to go back to the main room, be transported back there shortly. Thank all the presenters. Everybody who presented did an outstanding job. And if you probably want, you also can probably join another presentation in progress if you want as well. So.
Thank
you. Okay. So thank you all for your patience. I actually kind of blame myself for this one. I gave myself way too much time to go over the housekeeping rules to start off with. So consequently, the breakout rooms open a little earlier than we had slated. So so our timing was a little off. So I do apologize for that. But thank you for being willing to work around that and accommodate that and then to still make your breakout sessions successful. So to close out here. First, I wanted to once again thank everybody for being here. Again, my name is Andre Lewis. I'm from the Office of Research and Economic Development at the University of Michigan-Flint. Just in case I didn't have a chance to see you earlier, I'm one of the co-coordinators for the event, along with my counterpart, Mary Dybus, from the Office of Graduate Programs and Admissions, also here at U of M Flint. And I hope you found this day to be engaging and enlightening. Now, I say day in kind of an odd sense, even because our breakout sessions were only an hour, but the event included 10 different breakout sessions happening simultaneously. So that effectively gave us close to 10 hours worth of content that was shared today. So it really was a full day, even though it doesn't feel like it. But thank you for participating in this day. I'd like to take a moment first off to commend each and every one of you for an amazing and informative day of research and scholarship. And even though your presentations were only 15 minutes long or so, we do realize that it represents countless hours of work and discovery that you conducted. And I feel like the time and the effort that you put forth into this work really went a long way. So congratulations for all your hard work. And I appreciate you being able to work with us as we navigate this virtual world of disseminating research. So I commend you not only for the work you do, but also for taking the time to showcase that and then add it to the body of knowledge. So congratulations again, job well done. I would like to take a quick moment to recognize the committee members for this event who helped to bring this event into fruition. They include from U of M Flint, again, Mary Dybus, my colleague from the Office of Graduate Programs and Admissions, Dr. Ken Sylvester, the Director of Research here at the University of Michigan-Flint, and Dr. Chris Lewis, the Director of Graduate Programs and Admissions. From Mott Community College, thank you to Jason Wilson, the Vice President of Student Academic Success, and faculty members, Dr. Brian Littleton and Philip Knopf. And from Kettering University, thank you to Dr. Scott Reeve, the Dean of the Graduate School and Research. Then, in addition to the committee members, thank you to our university leadership at each of our respective institutions for their institutional support of student research. Thank you to all the individuals that helped make this program happen, namely to Dr. Heather Dawson, the Executive Director of the Office of Research and Economic Development here at U of M Flint, for delivering those opening remarks. Thank you to all of our gracious moderators who helped to navigate the student presentations. Thanks to all of our visiting guests for being here to support. And most of all, thank you to all of our presenters and their faculty sponsors. Thank you to each and every one of you. This event could not happen without your hard work and your dedication. So thank you again. I know I keep saying thank you, but I was always taught to just thank people for everything that they do. So and to close out, I just have a few quick announcements. So in the coming days and the coming weeks, you will have an opportunity to provide feedback for this event. Check your email for a link to the evaluation forms. Feel free to provide any sort of input about what we can do better to create a more robust experience for you, and we certainly appreciate that feedback. Presenters will also be contacted later as well. You'll receive your certification of recognition, and you'll also receive a courtesy t-shirt for participating. And then students who have presented today will have an opportunity to submit your papers to the annual publication for this event. So details will be provided regarding submission deadlines and submission guidelines pretty soon. So again, check your email. And then finally, the format that we followed here, as we did last year, called for us to do single breakout sessions happening simultaneously. So unfortunately, during the event, not everybody had a chance to go to different sessions. But for your benefit, we've recorded each of these sessions, and they'll be available for you to view later on. So once the video is available, we'll post it to our YouTube channel, and we'll disseminate the link to anybody who wants to visit. And in the future, so for the past 2 years, we've been enacting a virtual format, and as COVID restrictions are continuing to ease, we are considering revisiting the initial format of this event and resuming an in-person event. So we will continue to monitor the COVID environment, and we will continue to consult with local and state health officials to determine if and when in-person events will be able to be conducted again, and we will also certainly consider a hybrid version of this event as well. So stay tuned for the benefit of students who will be returning with us next year. We'd love to have you again. To any students that are graduating, congratulations, and we wish you all the best in your future endeavors. And once again, I would just like to thank you once again for your participation. This was an amazing event. And once again, I commend you for all of your hard work. So with that being said, we are going to dismiss, but if you have any questions, comments, or any kind of feedback that you would like to provide, again, we would happily solicit that. So once again, thank you all for a wonderful day. Thank you for your time commitment, and then most importantly, thank you for all the efforts that you've put forward into advancing your research agenda and sharing that with the body of knowledge. So once again, thank you all, and hopefully I will see you all soon. So take care, everyone.
Thank you, Andre.
Absolutely, thank you.
Thank you, everyone.
Thank you.
Bye-bye.
Thank you, everyone. Thank you.